Kindly be seated. Distinguished dignitaries on the dais, respected chief guests, esteemed speakers and delegates, heads of various departments, professors, researchers, scholars, students and participants, a vibrant morning to everyone present here. Sustainability is no longer about doing less harm, it's about doing more good. Reminding the words of Joe Henside, I, Princey Julia of 2nd BSc Biotechnology,
And I, Akash of 3rd BSc Biotechnology,
with immense pride and great honor, welcome you to the inaugural ceremony of the 5th International Conference on Advanced Biotechnology, Sustainable Innovation toward Viksit Bharat Vision, ICAB SIVBV 2026, hosted by the PG Department of Biotechnology, Daragadas Govindas Vaishnav College, Chennai.
a conference serves as a vibrant platform that brings together scientists, researchers, academicians, industry experts and young innovators from across the globe to share knowledge, exchange ideas and explore cutting-edge advancements and environmental sustainability. Over the course of this conference, ICAB SIVBV 2026, we will engage in thought-provoking sessions, keynote addresses, plenary lectures and panel discussions, all focused on translating scientific innovations into real-world impact for a sustainable future.
God is Tamil, ever remain Afristi alone, purity intact too. Delighted prize though, pure is Tamil, youthful forever awestruck. I request the dignitaries and the gathering to rise for the Tamil Vartha, followed by our college prayer.
நீராறும் கடலுடுத்த நிலமடந்த கிழிலொழுகும் சீராரும் வதனமென திகழ்பரத கண்டமது தெற்கணமும் அதிசிறந்த திராவிடனல் திருநாடும் தக்க சிறு பிறை நுதலும் தரித்தனரும் திலகமுமே அத்திலக வாசனை போல் அனைத்துலகும் இன்பமுற எத்திசையும் புகழ் மணக்க இருந்த பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிழமை திறம் வியந்து செயல் மறந்து வாழ்த்துதுமே வாழ்த்துதுமே வாழ்த்துதுமே.
சத்யோரதம் சத்யபரம் த்ரிசத்யம் சத்யஸ்ய யோனிம் நிஹிதம் சத்யே சத்யஸ்ய சத்யம் விதசத்யநேத்ரம் சத்யாத்மகம் த்வாம் சரணம் பிரபன்னாஹ. வாணி குணாம் கதமே ஸ்ரவணோ கதாயாம் ஹஸ்தௌ ச கர்மசு மனஸ்தவ பாதயோர்நஹ ஸ்மிருத்யாம் சிரஸ்தவ நிவாச ஜகத் பிரணாமே திருஷ்டிஸ்ததாம் दर्शने सुभवत्तनूनाम् नमो भगवते तस्मै कृष्णायाद्भुतकर्मणे रूपनामविभेदेन जगत्क्रीडति यो यतः. ॐ शान्ति शान्ति शान्ति.
In keeping with our cherished tradition, we commence this event by lighting the ceremonial lamp I kindly invite our honorable dignitaries to light the Kutuvilakku. The glowing beacon signifies our resolve to dissipate darkness and spread light, echoing the immortal word, Tamaso Ma Jyotir Kamaya, which means, from darkness, lead me to light.
A heartfelt thanks to our esteemed guests for leading this auspicious beginning. In this garden of learning, kindness blooms. I am honored to welcome you and request Dr. M.D. Balakumaran Sir, Assistant Professor and Head PG Department of Biotechnology, to set the tone with his welcome address.
Respected dignitary on the dais, distinguished guests, esteemed speaker, faculty members, dear students, and participants from across the globe. It is my profound honor and privilege to extend a warm welcome to each one of you to the 5th International Conference on Advances in Biotechnology, Sustainable Innovation Towards Viksit Bharat Vision 2047, organized by PG Department of Biotechnology, Dwarkadas Govindas Vaishnav College. This conference is not merely an academic gathering, it is a collective commitment to shaping a future where science, sustainability, and innovation converge for the progress of humanity. As India marches towards its vision of becoming a developed nation by 2047, biotechnology stands at the forefront, offering solutions for healthcare, agriculture, environment, and industry that are both sustainable and transformative. We are privileged to have with us eminent scientists, academicians, and industry leaders from India and abroad who will share their insights on cutting-edge research and practices. Their presence enriches this platform and inspires us to think beyond boundaries to translate knowledge into impactful innovation. The theme of this conference, Sustainable Innovation Toward Viksit Bharat Vision, reminds us that sustainability is not a choice but a responsibility. It is our duty to ensure that advances we make today become the foundation for a greener, healthier, and more equitable tomorrow. It is with great honor that we extend a warm welcome to our guest of honor, Dr. U. Satyanarayana, Professor of Biochemistry, Dr. Pinamanini Siddhartha Institute of Medical Science, Andhra Pradesh, and to our distinguished speaker, Dr. Joseph Selvin, Dean, School of Life Science, Pondicherry University. Professor Satyanarayana has earned enduring trust in the academic community through his pioneering contribution to biochemistry, nurturing generation of scholars, and advancing biomedical knowledge for the betterment of society. Dr. Joseph Selvin is widely respected for his impactful research in marine biotechnology and life sciences, fostering innovation and sustainable solutions that continue to inspire scientific progress and global collaboration. Your gracious presence at this inaugural ceremony add immense value and inspiration to our gathering. On behalf of the organizing committee, I extend my heartfelt gratitude to our Secretary C. Ashok Kumar Mundra, whose constant encouragement and support have made this conference possible, and to our dynamic principal, Dr. Santosh Babu Sir, whose visionary leadership has been instrumental in guiding this initiative. I also warmly welcome our international and national delegates, whose participation strengthens the spirit of collaboration and knowledge sharing. In addition, I extend my welcome to the heads of various departments, faculty members, and dear students, whose enthusiasm and commitment form the backbone of this academic endeavor. Let us use these 2 days not only to exchange ideas but also to build partnership, foster innovation, and reaffirm our collective resolve to contribute meaningfully to the mission of developed India. With immense joy and optimism, I once again welcome you all to ICAB 2026. May this conference be a milestone in our shared journey of learning, discovery, and sustainable progress. Thank you. Jai Hind.
Thank you, sir, for the warm and gracious welcome. With vision and action, we shape the future. I invite Dr. R. Ashwini, Assistant Professor, Organizing Secretary, for the opening remarks of ICAB SIVBV 2026.
A very good morning to everyone. On behalf of the organizing committee, it is my privilege to warmly welcome you all to the 5th International Conference on Advances in Biotechnology, Sustainable Innovations Towards Viksit Bharat Vision, ICAB SIVBV 2026, which is organized by the Postgraduate Department of Biotechnology, Dwarakadas Govardhan Das Vaishnav College. We are deeply honored by the presence of our chief guest, Dr. Joseph Selvin, an eminent researcher in microbiology and a contributor to advanced studies in life sciences. Our guest of honor, Dr. U. Satyanarayana, a distinguished researcher and renowned author whose textbooks have guided generations of students in biochemistry and biotechnology. Dr. Julian Adam, a passionate in agriculture and also in Tamil language. And our respected principal, Captain Dr. S. Santosh Babu, whose constant support and encouragement for academics and student initiatives inspire us all. We also extend a warm welcome to all the distinguished speakers, panelists, and delegates joining us from various academic institutions, research organizations, and industries from India and abroad. This conference is planned as a platform to share knowledge, exchange ideas, and discuss recent advances in biotechnology with a strong focus on sustainability. The theme of this conference closely aligns with the United Nations Sustainable Development Goals, particularly those related to good health and well-being, zero hunger, clean energy, climate action, and environmental protection. In today's rapidly evolving world, achieving these goals demands not only awareness but decisive scientific action. Biotechnology stands at the forefront of this transformation, enabling sustainable agriculture to feed growing populations, advancing healthcare for better quality of life, supporting renewable bioresources, and offering environmentally responsible alternatives to traditional industrial practices. At the same time, this conference reflects the spirit of Viksit Bharat Vision, which calls for innovation, self-reliance, inclusive progress, and sustainable growth. Biotechnology, again, an interdisciplinary science that integrates biology with technology, plays a vital role in realizing this vision by delivering science-based solutions that strengthen national development while safeguarding natural resources. As we move forward, it is essential to remember that true progress is not measured not only by the economic advancement but by the legacy we leave for our future generations. The responsibility lies within us, educators, researchers, and innovators, to ensure that technology becomes a force for balance between development and environment. Sustainability is no longer a choice but a scientific necessity, and biotechnology is one of the most powerful tools we possess to build a healthier planet and a resilient future. Over the next 2 days, the conference will feature keynote lectures, plenary sessions, and oral and poster presentations and panel discussions as well. We hope these sessions will encourage meaningful interaction, foster collaboration, and spark innovative thinking among the participants. I would like to sincerely thank our management, principal, advisory committee members, speakers, and sponsors for their guidance and support. I also appreciate the dedicated efforts of our faculty members and student volunteers who have worked tirelessly to make this conference possible. Once again, I warmly welcome you all and wish the conference a fruitful deliberation. Let this gathering inspire new ideas, ignite transformative research, and strengthen our collective commitment to building a sustainable world and a truly developed nation. Thank you.
Thank you, ma'am, for your insightful opening remarks. We are privileged to have our respected principal sir, whose leadership and vision have been instrumental in shaping the excellence we witness today. His firm commitment to education and tireless efforts have steered us towards greatness. Let's welcome our principal, Dr. S. Santosh Babu sir, with a big round of applause. Your insights, sir.
Jai Sri Krishna. Very warm morning to all. Today is a very wonderful day for the audience where they are going to witness the 5th International Conference in terms of life science. I always will not divide biotechnology, biochemistry, botany and microbiology as a different one. As put together, I used to call it as a life science. Since the biotechnology department is conducting, it is named as Advances in Biotechnology. And not only advances in biotechnology, for when you do something, it has to be in line with some concepts or some goals. That goals is nothing but the Sustainable Development Goal. The Sustainable Development Goal is formed by the UNESCO. And when you want to adapt to our country, there comes the Viksit Bharat. Viksit Bharat, nobody knows the meaning of Viksit Bharat because it is in Hindi. It is nothing but developed country. We will say it in English. Tamil sonna munneriya naadu. Developed country means it will go to our heart. Munneriya naadu na it is go to heart of only Tamil Nadu. But if you use the word developed or Muneri India, it will not reach the other part of the country. Most of the people are Hindi speaking people. That's why it is named as Viksit Bharat. What is the year? Staff சொல்றீங்களே ஸ்டூடெண்ட் ஒண்ணுமே சொல்ல மாட்டேன்றாங்க. 2047. Whether I am going to benefit out of this is a very big question mark. But you people are going to benefit that. 2047. So in line with this, the 5th conference is organized, and for that they have called a very good personalities, where from the old technology to new technology. Even when you go to hospitals, previously X-ray itself the method of evaluating the people, but now it is not like that. Scan have come, CT scan come, MRI have come, lot of technologies come. The people who are here is, they have adapted the technology in life science and they are going to speak to you. Who is nothing but Dr. U. Satyanarayana from Andhra Pradesh. He is going to give a lecture, he is going to give an inaugural address. And your chief guest of today's program, from Dean of Life Science, Dr. Joseph Selvin. That's why they are called a Dean of Life Science. They have not called the people from Biotechnology, Dean of Life Science, so that he knows about all the department together for growth of the students community. So both are here. I am very happy to welcome you all for our international conference. Definitely this conference will be a good platform for the researchers. But if it is a good platform, the researchers means we have to make the students to be a researcher. So you people are a student of various institution and also our institution. And you student also should know about the research in life science. Where during COVID period, where the biotechnology people and microbiology people, before the government machinery is coming to exist, they started their work using the PCR mission. I am talking about COVID-19. The first mission was operated in Tirunelveli districts in the hospital through ICMR is a research scholar of Biotechnology Department of MS University. Since I am a registrar at the time, I used to lend my device and also the students, and that student also went. Everybody is afraid of going there, but as a young chap, that fellow, he went with all dresses and he did for 1 week. After that only the government thought of, I don't know how many missions have come. See, it's a very big question mark. when you think about this life science can be used for productive and also for distraction. Usually COVID-19 I used as a lay person, I'm not belong to life science, I belong to computer science, but I used to work on application sites, so that even I can take life science and application side in computers. When I saw that, when the machine needed, immediately everybody they are able to supply the machine, means how the machines have been produced, how the vaccine have come, still is a big question mark for the government and the administrator. Only the researchers will know. So this is a very critical area, and other part of this is the life science students are less in number. When you take commerce, the outcome of commerce every year is more. Outcome of even computer science is very more. But when you go to life science programs, is very less. Because when you found out one product or one problem, and if you are able to solve it, means 1% is enough for giving a solution for 1 lakh people. So that is the case. Suppose if you are able to find out a vaccine, only 5-10 people will be working on creating a vaccine. After creating a vaccine, we need around 500 to 1000 people for production. We have to need a people for financial support. All these things put to the government support. Then it will come as a product. But base level is, who is the base? Who is the base of any product, right? Especially in vaccine and other things. You people are product. You are the base. As a student of our institution or any institution, you have now more responsibility in Viksit Bharat 2047, where you have to create without any diseases, any disaster. So this is the purpose. So I don't know in which aim this particular conference was will go, because only after your presentation and after the speech of the specialist and invited talks, at the end of the day, tomorrow evening only, we know that this conference is going on in this line. So in the beginning of the session, I could not say that this conference will give this eye-opening, that I could not say. But you people have to say. If you want to say like that, means what is the things you have to do? You can skip your regular classes. When you skip your regular classes, you can get the notes from others and you can study. But this, if you skip this conference, getting the input from experts, from various people, various paper presentation is very difficult. So this 2 days is a day for you, where it is not like seeing movie for 3 hours, attending class for 5 hours. It is a day you have to spend minimum 7 hours in this particular conference. So 7 into 2 is 14 hours. You should be vigilant, and accordingly you should sit and you can learn, understand. If you are not able to understand, make a note of it. Again you go back to your institution, you talk to the teacher, you talk to the mentor, you talk to the well-wisher and try to understand, and finally this conference outcome will be good. Otherwise, just like sitting in front of this auditorium, like watching the movie, because usually when you see movie, you should not take anything into your heart. You have to see the movie. When you go out, you have to forget and you have to go for the next job. But this is not like that. This is like spending a time here. After spending time, you have to recollect something, and based on that, you have to start working on it. Now, a lot of avenues are there. Even in our institution, we have many machines. And equipment is not a problem. Government of India and Government of Tamil Nadu, through various schemes, they are supporting the universities, they are supporting the many people. Even the private companies also ready to work with you on usage of instruments. Only thing, you need a problem. If you are able to identify a problem, for giving a solution to a problem, you need samples, you need equipment, you need interpretation, and you have to give an output. For all these things, in a current situation, 30 years before, it is very difficult. There is no name of instrumentation lab at all. Only one lab in the whole of Tamil Nadu will be there. Now it is not like that. Even if you go to MS University, there is a common instrumentation lab. During my period, I got 20 crores and I set up the lab, through which you can use the equipment. If you want to use the equipment, you can travel to Tirunelveli and you can use that equipment, you can come back. Like that IIT Madras, they have an equipment. Madras University is having an equipment. And if you are able to identify a problem, for the problem, if you want to use any instrument, we have an instruments. Only thing is, you should have a drive. Where you get the drive? As young children, where you get a drive? Sitting in our home, you could not get a drive. Sitting in a classroom, you will not be getting a drive. You will be getting a drive only when you talk to the people. Where to talk, whom to talk. That job is our institution. Our institution is creating. You know that DG Vaishnava is a 60 years old institution where we have a good faculty, where they are able to adapt to the technology, adapt to the system, and they can come up in their life. Like that, as a student, they are creating a platform like this international conference. And our management also, whenever we approach anything to the management, they will not think that conference is an expenditure. They will not think like that. They will think that conducting conference is a resource for the institution. It is resource platform for the facilitator for the students community, so that they also always supporting conducting more conference. Only they will expect that is outcome of the conference. Even as a principal of the institution, initially for the past 3 years, I made a count of the programs. But this year, we made very clearly that when the first HOD meeting was conducted, we have made clearly that whatever the program you do, that has to be aligned with the sustainable development goal, all 16 sustainable development goals. Even if their program is conducted, in that they have to mention that these are the goals they are going to cover. Once if you do this sustainable development goal effectively, Viksit Bharat 2047 is not at all a problem. Almost what are the things is happening in Viksit Bharat, the developed country, is we have to take care of whether it is climate change or alternative energy, whatever may be, EV, whatever may be health, then food industry, whatever maybe you take, everything is covered in the sustainable development goal. Only you people as youngsters, you are going to benefit from that. I am not going to benefit. I know very well that. But your people are going to benefit from that, so that you should be very clear that this conference is very important. Biotechnology always plays a vital role in this United Nations sustainable goal, like that sustainable agriculture and food security. You know how this biotechnology came? It came from botany. The botany is always think about the plant and other things. They will not think about the technology. But when you see only that, since our population is growing, once if the population is growing, food production should increase. If you want to increase the food production, you have to use some methodology in the life science, whether it is biotechnology or microbiology, you have to do something. But in that one point of time, we are happy that we are able to produce more food grains. But what is the quality of the food grains? Now the people started asking about pest control, other control, and they are talking about organic and inorganic. Organic farming plays a very important role. Even I read a newspaper a week before about usage of sugar cane. The sugar cane, what we are taking as a salt, as a sugar and other things, it will increase the glucose index. But at the same time, when you use the sugar cane, black color sugar cane, what we are using for Pongal festival, that is the actual sugar cane we have to use. I read. நம்ம படிக்கிறோம்ல இந்த பச்சை கலரில் ஒரு சுகர் கேன் வெளியிலெல்லாம் விற்கிறாங்களே, கண்டிப்பாக அந்த சுகர் கேன் குடிச்சுன்னா இட் இஸ் நாட் எனர்ஜி கொடுக்காது, உன்னுடைய கிளைக்கமெக்ஸ் இண்டெக்ஸ் இன்க்ரீஸ் பண்ணும். அதே கருப்பாக இருக்கிற ஒரு சுகர் கேன் நம்ம பொங்கல் எப்போ கடிச்சு சாப்பிடுவோமே, பல்லு சுத்தமாக எல்லாமே, அந்த சுகர் கேன் இட் இஸ் ரியல் ஒன். பட் டைம் டேக்கன் ஃபார் ப்ரொடக்ஷன் இஸ் மோர். Sugar content what we are getting from this very less, that is not correlating with the demand for the people. Adanal they went for an alternative. They went for alternative because of green revolution or food revolution. But what is the outcome? What is the side effect? Is full of pesticide, full of everything. Now we thinking about, most of the people started thinking about organic. So affordable and accessible health care, clean energy and biofuels, then environmental protection and waste management, climate resilience and biodiversity conservation. See, these are the areas. I am not a life science person, but these are the areas was predicted by the Sustainable Development Goal. Every lay person, the people from commerce know about this. So that is the purpose of DG Vaishnava College. When the student is going out of this institution, they should know about the Sustainable Development Goal. Why they should know about the Sustainable Development Goal? Tell me. எதுக்காக ஸ்டூடெண்ட் தெரிஞ்சுக்கணும்? You are the future of the country. Now we are not talking about the state. Now we are talking about the world. You are the future of the world, because even one country is not following the protocol properly means that will impact us. Because of the problem in US, our gold price is increasing. Because of the war between Russia and other things, our education system is increasing. Previously, the impact will reach the students only within the state. That went to up to the country, India. But now it is not like that. If anything happened at other part of the globe, that affects our system. So now, as a human being, now there is no Tamil Nadu, there is no India, there is no America or UK. We have to think about the human. We are all a community of human, where we are in the earth, where we have to work for the Sustainable Development Goal. So these are the things are very important. So our institution firmly believes that higher education institution must act as a catalyst for national development through interdisciplinary research. That's why I told you that interdisciplinary, like microbiology, biotechnology, biochemistry, botany, all put together. The students should interact with each other. They have to work with each other. The research scholars should work together. PG students should work together. For some courses, they should come to biotechnology. Some courses, they should go to microbiology. Next semester, we are going to revamp all the curriculum in line with implementing generative AI in the system. So people used to tell that AI, ChatGPT, all spoiling our mind. It is not like that. How we are using the chat, instead of giving a data such a way that biotechnology prepare a speech. If you give it to chat, they will give. You should not give like that. What you are going to speak, no, that you have to give it to them. So first, fifth international, whatever I speak, no, if I give it in the ChatGPT, it will give you a refined output. So ChatGPT, how to use the ChatGPT is very different. It's a technology. But technology you should not use with the two lines. You have to talk fully what you want to do, you present it. It will give you. Then only it will be a tailor-made. Tailor-made software works very well. Our LMS, e-governance software, what we are using, no, it is a tailor-made which was designed fully for DG Vaishnava College by our team. Based on the input received from the HOD and myself, it was designed. I think the students would have happy with getting your internal. Last year we would have made internal marks to be accepted. Everything you know very well that. So these are the things are very, very important. So if you ask me to speak, I will take whole day for you. I don't want to do. I conclude that. Once again, I like to appreciate the PG department, PG and research department of biotechnology. Should the university have to announce that, but anyhow, we know that our institution is meant for research, and what are the things needed for research? We have enough equipment. Recently, we added a space and we have renovated the particular lab, and the inspection commission came. They are also happy with that, and they also given some requirement. We will be fulfilling it. Definitely in the next syndicate, it will come for the next syndicate. Definitely this department will become PG and Research Department of Biotechnology. For that, I like to congratulate Dr. Murugan and his team for working out in life science. Microbiology is a research department with 52 students with 10 research scholars. Second with UG, PG. Together it is going to shortly. Biochemistry also is working on it. So if they work, the HOD is not much clever. If he is clever, definitely he will bring it in the next academic year. So with this short note, I like to congratulate the whole team, and we all met. You are all here. I will come, I will give a speech and I will go out. But you are here for the next 2 days. Spend the day with us effectively. Go with some takeaway. All the best. Jai Hind.
Thank you, sir, for your inspiring and encouraging words. We are truly honored to have our esteemed chief guest of honor, and speakers for the inauguration of ICAP 2026. As a token of our respect and gratitude, I request our dignified principal, Dr. S. Santosh Babu sir, to felicitate our guests with the ceremonial gift and memento.
Thank you everyone, kindly be seated. Memories captured today become inspiration tomorrow. A souvenir is a reminder of where we have been and what we have achieved. It's a great pleasure to launch the souvenir of the 5th International Conference on Advance in Biotechnology, Sustainable Innovation toward Viksit Bharat Vision. I request our dignitaries to release the conference proceeding, and I invite all the faculty members to come up on the stage.
Thank you everyone, kindly be seated. I would request Dr. P. Indumadi, Associate Professor, PG Department of Biotechnology, to introduce our guest of honor, Dr. U. Satyanarayana sir.
Jai Shri Krishna, happy morning to all. It's my immense pleasure in briefing today's guest of honor, Dr. Uppala Satyanarayana, Director and Professor of Biochemistry, Dr. Pinameni Siddhartha, Institute of Medical Sciences, Jnanavaram, Andhra Pradesh. Sir holds his PG degree in biochemistry from Nagpur University and was awarded PhD from National Institute of Nutrition, ICMR, Hyderabad. Dr. Uppala Satyanarayana started his academic career as an assistant professor in the Department of Biochemistry at Mahatma Gandhi Institute of Medical Sciences, Sevagraham. Later, he became associate professor and head in the Department of Biochemistry in Siddhartha Medical College, Vijayawada. His sir's passion lies in biochemistry, specifically medicinal chemistry and interdisciplinary areas like genomic medicine and personalized medicine. He published around 80 research papers in top-tier peer-reviewed journals with high impact factors and both guided the MSc and PhD scholars, ensuring the academic standards in research. Sir has also published 5 books as author in the field of biochemistry and medicinal chemistry and is highly recommended for medical and science students in India and abroad. Sir presented 75 national and international conferences and served as a project director for the research schemes of ICMR and UGC. He has received Best Teacher Award from the Siddhartha Medical College and Honorary Reviewer Award from the Journal of Molecular and and Medicine, honored with Regional Service Award by an Asian Pacific Congress of Clinical Biochemistry, Hong Kong and Japan. He is a recipient of Sundarlal Sahu Gold Medal from Nagpur University in the year 1975. Acts as a technical committee member, laboratory accreditation, Minister of Sciences and Technology in Government of India. Sir is awarded fellowship by National Academy of Clinical Biochemistry USA, and Dr. Uppala Satyanarayana is a part of the university decision-making body, contributing to academic policies, curriculum development, and other key decisions. He is also served as a Vice President, Association of Clinical Biochemistry of India and Advisory American Biographical Institute USA. Nominated as an Outstanding Scientist World Over in the year 2007 by the International Biographical Institute UK. Sir is also awarded Fellowship in Association of Clinical Biochemistry in India. Dr. Uppala Satyanarayana's research shows how diet influences oxidative stress and health. Make smart food choices focusing on nutrition to tackle diseases like heart attack, diabetes. Let us eat our way to better health. Let us welcome Dr. Uppala Satyanarayana, sir. I hope that audience will take home some message to enrich their life. Welcome you, sir.
Thank you, ma'am. The mind grows by what it feeds on. We are most honored and privileged to have a distinguished personality, Dr. U. Satyanarayanan, sir, Research and Professor, Department of Biochemistry, Dr. Pinamani Siddhartha Institute of Medical Science and Research Foundation, Andhra Pradesh, to herald the moment. With great respect, we invite sir to take the stage.
A very good morning to all of you. Respected principal of this college, Dr. Santosh Babu, Dr. Balakumaran, HOD of Biotechnology, Dr. Joseph Selvin, the Dean of Life Sciences at Pondicherry University, Dr. R. Aswani, the Organizing Secretary, Dr. Senthil Kumar, the Assistant Professor, PG Department, and we have Dr. Julian Adam, a renowned scientist from France, the faculty members, the participants, invited guests, and my dear students. At the outset, I wholeheartedly thank the organizing committee for inviting me to attend this international conference. And as you all know, biotechnology is the science of this century. And it is ultimately the biotechnology is the subject that is going to engineer our lives and give or reshape the world in the years to come. I congratulate the Department of Biotechnology of this college for organizing the 5th International Conference on Advances in Biotechnology. It is worthwhile to know a few important milestones in biotechnology, or rather life sciences, that are going to shape the future developments, the biological or medical sciences. One is CRISPR, a molecular tool, the clustered, regularly interspaced short palindromic repeats, CRISPR, a novel technique by which we can edit, delete, or modify the genes. A wonderful technique. And this has come into existence, though most of it is at the experimental stages. The developments are going to be very outstanding, and they will lay foundations for a wide range of applications, which includes gene therapy, the wide range of diseases we are encountered in the medical practice, the cancer therapy, the problems we are facing, the uncontrolled growth of the cells, and how to regulate, how to make them static, or how to eliminate the tumor cells. And then we have the treatment of the infectious diseases, which is going to be really happening in the years to come through the CRISPR technology. And then we have the agriculture and veterinary developments, the advances in the veterinary fields with regard to the different animals, the way we can breed them, the way we can have them, and also the agriculture importance, where we can think of having the plants of our choice with the nutrients of our interest. So CRISPR is going to be a boon to the molecular biologists in the years to come. And it was in 2020, the Nobel Prize was awarded to a group of scientists who relentlessly worked on CRISPR and developed the technique, such a wonderful technique. And the second technical information I want to talk is AlphaFold 2. Here I must tell you, those were the days, some years ago, when you want to know the structure, particularly the three-dimensional structure of a protein. It used to take several years. Worldwide, there are instances where not one PhD student, at least 2-3 PhD student has to work to develop a three-dimensional model for protein structure. And what surprised the world was, by use of artificial intelligence technology and through the employment of AlphaFold 2 technique, unbelievably, 200 plus million proteins, 200 plus million proteins that are there now in the globe could be modeled very precisely with respect to the three-dimensional appearance. Unimaginable. When one protein was taking some 5 to 6 years to model it, 200 plus million proteins are now available with the three-dimensional structure. And the group of the scientists were awarded Nobel Prize in 2024 for AlphaFold 2 technology. Now, with this technology, wonders can be created. I don't think many of you are aware that only because of this AlphaFold 2 technology, the COVID-19 vaccine could be developed in such a short period of an year. Otherwise impossible. Earlier, vaccine development, any given vaccine development was taking decades, later some years. But for this vaccine development of COVID-19. It didn't take years. In about a year, vaccine was developed by many firms, many government organizations throughout the world, all because of their three-dimensional structures of the proteins were well established, and that laid the foundation for the development of the vaccine in a wonderful, precise and accurate manner. Not only the vaccine development, your AlphaFold 2 will have many other applications like drug development very accurately, and this technique is going to make a platform for the curative aspect of the genetic diseases also. Well, we feel that normally whenever some research happens, you make a good concept or a good paper publication, you feel elated and you feel you have reached the top of the world. I remember one instance that happened to me 50 years ago, that was in 1976. While I was working for my PhD on amino acid metabolism, tryptophan metabolism, at the National Institute of Nutrition, during some holidays, I just went to my village. While I was walking in the fields, I came across one gentleman who was enjoying with a cigar in his mouth and looking after the cattle in the fields. He inquired me, "So what are you doing, young man, these days?" I said, "I am doing this research, that research, nutrition, your fields, all the things. We are the people who are going to control everything in life, and everything is going to be a part of the research only, and then only you people are going to survive, you people are going to live. Only because of our backbone of conducting the research, you are having so much agriculture, so much of productivity, so many diseases being cured." So all these things, I just started giving him a big lecture. Because I was young, and I thought I should impress him. And the reason was, I was a little bit elated because by then I already published one paper in International Journal, an American one, and another paper in British journal. So I was under the euphoric condition. The gentleman asked me, "Young man, cool down. I have only one question for you. I am blessed with a granddaughter 4 days ago, and she is very healthy. She is enjoying life, smiling, and I am very attracted towards her. And she takes milk from her mother through breastfeeding, and this little baby of 3 days old is capable of passing her urine, passing her stools. Now you claim you are doing a lot of research and doing everything is under your control, the world as if it is under control because of the research. You do one thing for me. You create a machine where you will feed milk and you get stools and urine outside as my granddaughter is doing." I was shocked. Such a simple question. He said, "You say you are doing such wonders in the world through your research, which is at an advanced stage, everything. I don't want anything. What I want is, yes, my baby is there, my granddaughter, she is being fed milk, and she is having some output in the form of stools and urine. You create a machine for me where this process will be conducted." Is it ever possible for us to do it? Is it ever possible to create a machine where a one day baby is capable of doing? Impossible. Then he told me, "I know a lot of things that you talk about research, although I am almost an illiterate, but what has given in the nature, you people will never be able to reach that level. What you are doing is you are just thinking of doing a 1 millionth or 1 trillionth of what God has created in nature. You are trying to understand that. The word I used, 200 plus protein, 3-dimensional structure, we came to know today. They were already there in nature. So what we are trying to do is trying to understand what is already there in the nature and try to exploit it for our benefit, either towards the health care or towards the animal care or towards the agricultural care. That's all. So I still remember, even after 50 years, many times I remember that old man. What he raised, the very relevant question is, which we will never be able to solve even decades or centuries to come. So with these few words, I only wanted to share my thought, particularly the two techniques which are quite relevant to all the life science students. And then I wish the conference a grand success. As I understand, we have eminent speakers and a large number of poster and oral presentations, and I'm sure you will have great achievements in these 2 days with respect to the intellectual feast, and I'm very confident you will go back with a very good message. And once again, I thank the organizing committee and all the students who have patience to listen to my brief talk. With this, I take leave. Once again, I thank you very much.
Thoughts that inspire actions are the lasting treasures for future generations. Thank you for your remarkable address, sir. I would request Dr. K. Nitya, Assistant Professor, PG Department of Biotechnology, to introduce the chief guest, Dr. Joseph Selvin, sir.
Good morning to one and all present here. It is an honor to introduce our chief guest, Dr. Joseph Selvin, Fellow of National Academy of Agriculture Sciences and Director of Research and Development, Pondicherry University. Dr. Joseph Selvin holds MSc degree in Environmental Biotechnology, PhD in Pathology at MS University, and his postdoctoral research in metagenomics at Environmental Research Institute, Ireland. Sir also served as ICMR International Fellow in Singapore. He has been associated with several leading international and national research institutions. He has globally recognized for pioneering work in genomics and microbial bioprospecting. Dr. Selvin has completed 16 major projects of total cost of rupees 16.3 crores, funded by European Union, DBT and Ministry of Earth and Science and Department of Science and Technology. Sir has undergone various collaborative research across 25 different countries. Dr. Joseph Selvin has made significant contribution to field of microbiology with 178 journal publications, 56 HE index, more than 600 impact factors, by books and 16 book chapters. Dr. Selvin has 2 patents and developed 2 innovative technologies. Sir has successfully guided 19 PhD scholars and 7 postdoctoral fellows. Sir has received numerous awards, including Jawaharlal Nehru Award by Indian Congress of Agricultural Research, DST Young Scientist Award, INSA Young Scientist Medal, and Outstanding Science Award in year 2020-22. We welcome you, sir. Thank you, sir.
Thank you, ma'am. It is our pleasure to invite our chief guest, Dr. Joseph Selvin, sir, to deliver the keynote address of the conference. The dais is yours, sir.
Good morning, all of you. Very respected Secretary of this institution, Sri Ashok Kumar Mundra. Sorry, he's not here. Maybe with their blessing, the conference is happening. And respected Captain Dr. Santosh Babu, principal of this institution, as well as former registrar of MS University. and the fellow speaker as well as the expert, Dr. U. Satyanarayana, Director Research from the Department of Biochemistry, as well as the organizing team, Dr. Balakumaran, Dr. Aswini ma'am, Dr. Senthil Kumar. I may be missing some of the colleagues, dear colleagues and my dear students. So I will be taking around some of the happenings, what is happening around us. After COVID-19, we thought of everything is over, not over. Now you heard about Nipah virus. You just imagine the students of biology should know what is the outbreak happening around us. See, Nipah, you just imagine, I don't know, because we have no time to interact each other. How you can imagine, we going to face the outbreaks of, we can say, see, COVID-19 have only less than 2% of mortality, but Nipah will have 50 to 70% of death rate. You can imagine, you are aware that Nipah, once, if you have a fruit, if you are taking a fruit, raw fruit, which may be contaminated with the virus, it will take 21 days, you will have the onset of the infection, means you have some symptoms. So you may be cured within a week time, maybe some fever or some symptoms, but you can imagine it has the chances of within 1 year it may return back as encephaly. So the people who have the encephalitis or encephaly, they will be in hospital, maybe they may be in ICU, they will be dying. These are all the dreadful diseases are going around. And another one you can imagine, maybe the Prime Minister Mann Ki Baat talk also given the indication about antimicrobial resistance, AMR. Did you know nowadays what are the antibiotics we are taking? Previously we will say the first generation, second generation. Now we are reached the fourth generation antimicrobial resistance, means pan-drug resistance. If you have an infection, we cannot treat with what are the antibiotics available in the market. So did you know, if you have an infection of pan-drug resistance, what is the last resort antibiotic is colistin. Colistin means, like we can say colistin cannot be prescribed, even also we also tried to get colistin for our own research. None of the pharmacy give the colistin directly. But you can imagine colistin is such a scheduled drug which has been used in India as a growth promoter in the poultry. Though it is banned, but still it is in market. So these are all, you should have the awareness about what is happening around us. This is very, very important. I'll just take you around like globally, what is the changes in the life sciences. You might have heard about it, the life. Previously, we defined the life as something is having locomotion and reproduction, then it is known as life. Now we are completely redefining the terminology life. The reason is you can, xenobot. Xenobot is nothing, you can see some of the scientists have did some like cell line. You heard about cell growth in the laboratory. They grow the cell with frog skin cell, and they mixed with what is called heart muscle cell. Heart muscle have the function of motor. So once they mix the cell, so the frog skin acts as a skin of the cell mass of what? Heart muscle. So it is amazingly, the cell is dividing, it is function as its own, it's moving. You understand me? You can imagine any life have a life node. So means there is something we can say we have Atma. So some feeling. You are saying that life has something else. You can see it is nothing. Frog skin and heart muscle cells grown and mixed, they become act exclusively. That they named as xenobots. Now the discoveries are going on. Other side, we are discussing about alien life. All the students of life sciences, you should know the 3 theories of evolution. You know Darwin's theory of evolution. We have no time to discuss. Number 2, you might have heard about it, Stephen Hawking theory of aliens. Stephen Hawking heard about it? Maybe 2018 he died. Because after Darwin, the theoretical physics means those who survived our time is Stephen Hawking. Stephen Hawking said, like one time our space mission will communicate with the extraterrestrial life, that will be the end of the universe. But I'm not part with Stephen Hawking. I'm giving another explanation. All the life on the earth is made up of 4 alphabet. What is the 4 alphabet? All the life. AGCT. Did you imagine any life on the earth without AGCT? Adenine, thymine, cytosine, guanine. All the life. That's why only we are saying tree of life. Everything is one tree. Even a single bacteria, even a single virus. The virus killing us also AGCT. Dying person also AGCT. Plants also AGCT. Animals also AGCT. Anything is AGCT. Can you say life without AGCT or something else? Any time you thought of? But your generation going to face life on the earth other than AGCT. So life on the earth other than AGCT is known as what? Aliens. So we have thought that these are all our wrong understanding. We thought that we are expecting extraterrestrial aliens will come and conquer the earth. No, we will going to develop the aliens on the earth. We going to expand the alphabet of AGCT. Means AGCT bonds, adenine, thymine, means nucleotide alphabet we expand. While you expand the alphabet, generate organism, that organism not extraterrestrial or not exist on the earth. Then you have a question, it is happening, it is already happening. While you are facing the viral outbreak with other than AGCT, that time we will understand what will be the devastation. Now, climate change. Generally we say the word climate change. Neuralink. You know the person. What is Neuralink? Heard about it? Tesla. Who is the discoverer of, who is the founder of Tesla? Elon Musk. Elon Musk have 3 important projects. The major one is Tesla. The second one is space mission. All of you should know the third important one is what? Neuralink. What is Neuralink? Neuralink is the project. He is already approved by FDA, Food and Drug Administration. They will implant a chip on the brain. So you not come to the classroom. Next 10 years of time, the chip will have everything. You have seen the robo, no, the movie. The robo, while it is helping the, what is called, the actress. She is writing the examination. It is showing all the, have you seen? No. But it is, we can say it is unimaginable. You can have the chip. You can download everything in the brain. Why you are coming back to school or the college? You understand me? So that is Neuralink. What they are claiming, Neuralink will have the support for the people in, what can I say, some neural issues or autism. That's the way they are developing. But it will change the entire world. Usually I am giving talk to I teach my students. I explain to them one way, AI. Now Rise Stars are talk, everybody indicating AI. So you can imagine artificial intelligence. Where from the intelligence? Whose intelligence it is? It is our intelligence. You can imagine your Gen Z generation. You are using the intelligence of our generation. If you are not using the brain, continuously exploit the intelligence of AI, your intelligence will be spoiled. Your kids will have, so you can imagine the addiction of what? Reels. Previously we will say this childhood, this like children have addiction and juveniles have addiction, adults have addiction, age-old fellow have addiction. But uniformly have addiction is what? Reels. Starting from the 2-year baby to 100-year fellow also. So you can imagine our intelligence only now in what? Database of artificial intelligence. All the databases are occupied by our generation or the previous generation intelligence. You are not using your brain. That is your intelligence. What will happen? Next generation will have nothing. So very careful using the AI. Try to write your own. Engineered virus. So this is actually a world political play. See, China engineered and escaped, that is COVID-19. But the scientific world entirely helping China. Even I remember one article, Nature Medicine. The COVID is happening, they publish it. They said, this virus have less impact on the, means the death rate is very less, we cannot accept it as engineered. You can imagine any virus infection is seasonal. Just general knowledge. We don't need any scientific base. General knowledge or our own tradition, we can say any virus infection is seasonal. Then I can ask you, see, 2020, the COVID was starting. All our doctors, means giving the, what can I say, interview with the media, they are saying that India is the safest country. We're going to have the biggest, what? April, May. April, May means what? The biggest summer, hottest summer. The virus cannot survive. Did you know Delta was in April, May? Highest death rate is when happened? April, May. So this virus is in Antarctica was there, Arctic was there, everything. Then what is the meaning? They try to fool us. So you are the students, be careful. You have to teach your, like we have the word microbiome. I also part of the Microbiome World Support Association. I usually ask the student, you are the ambassador of microbiome. Ambassador means I'm giving to, I don't have time, but I try to give a few small touch about microbiome also quickly. Well, just a 15 minutes talk. So you can imagine, we are the students to teach our parents, our family, our village, everybody, what is microbiome. Mammoth. So mammoth, you heard about it? What is mammoth? Ice Age movie. Have you seen? Ice Age movie, everybody seen, no? There is a Antarctic woolly mammoth. Antarctic woolly mammoth means an elephant with wool. So you have seen the movie as what? Just like pictorially or what can I say? Animation, I mean, correct? Animated movie, but it is beyond nature. It is already under creation. Recreation of mammoth and reintroducing the world, that will be the beginning of all endangered animals get back. So that's the way science is like tremendously changing. So I was, okay, this one quickly, yes, of the students. See, where we are synthetic biology. See, you can imagine synthetic biology means nothing new. Everything is start from cell biology. Cell biology transferred to genetics, Mendelian genetics. You might have heard about it. Then we have genetic engineering. Then 96, the cloning of Dolly. You heard about the Dolly, one sheep was cloned. Every world was amazing, but it was a failed. And cloning of Dolly. And 2000, we started the era of genomics and human genome. Many things are happening. And genome engineering and synthetic biology. Just one word quickly, what is the difference between genetic engineering and genome engineering? Can you express an organic molecule in a bacterial cell? Cannot possible, because many students say yes. Always cloning and expression is a protein. If you wanted to express an organic molecule, you have to transfer the entire metabolic or part of the metabolic pathway is known as genome engineering. So I'm quickly, yes, I take my time so that easy. I'll ready another 5 to 10 minutes quickly. So the entire play is on AGCT. See, you can imagine a human genome 3.2 gigabase pair. Usually I explain to the students, the unit of the genomic science and the computer science is same. Computer science we use the units, bytes, kilobytes, megabytes, gigabytes. But in the case of genomic science, we use the word base pair, kilo base pair, mega base pair, giga base pair. Now it is exa base pair. Exa means 10 power 18. Are you clear? So this, why this AGCT is the biggest playground? See, all humans, we are all here, I can say, we are all made up of 3.2 giga base pair genome. Everybody have 25,000 genes and 19,500 are clearly explained protein encoding genes. So I wanted to ask a question, why we are diverse? What's the meaning of clones? Identical. What's the meaning of clones? Identical, genomically identical. We are all genomically identical. What's the reason we are diversified? Anybody? Madam is correct, expression. But before one terminology is there, expression of genes of me and you are different. What's the reason different? Genome locus, gene locus. So you and me have the same gene, melanin secreting gene, but gene locus determine I will be black, you will be fair. You got the point. So you will be high IQ, I don't have IQ, means the gene locus is the matter. So now, sir explained, no, gene editing. Gene editing came to the place, we can change transpose zones, change the location of gene, or delete the gene, introduce the gene. So, okay, these things are, it will take a little time to explain about synthetic biology. Synthetic biology we use, anyway, I'll quickly explain. See, a genome, you should know next generation sequencing. Quickly I'll explain in a simplest way. It is known as whole genome shotgun sequencing. Whole genome, shotgun, the word shotgun cloning in biotechnology, shotgun means random. Usually the cloning is by PCR, it is known as target cloning. We know which one is we are cloning. We don't know what is we are cloning is known as randomly we are cloning. Any fragment of the DNA is known as shotgun. So whole genome shotgun sequencing, you can see how it is happening. Usually you can imagine the DNA as kilometer in size. If you divide the kilometer, you will get meter. Am I correct? You divide the meter, you will get centimeter. Divide the centimeter, you will get millimeter. This is actually happening. Means what's the meaning? Large size of DNA spliced into pieces, again further fragmented to small fragments, further small fragments. These tiny fragments undergo sequencing. Now the question is, these tiny fragments undergo sequencing. The sequencing mission give you the tiny sequence information is called as reads. The reads are like millimeter size. That is a explanation. You can small size tiny fragments undergoes the sequencing mission. It will give you the sequence information is known as reads. The reads are assembled like centimeter size is known as contigs. The contigs are again assembled into larger size is known as scaffold. The scaffold are further assembled, you will get genome. But the question is, in synthetic biology, just reverse. In genomic science, natural genomes are sequenced. In synthetic biology, designs, just reverse. You have to design the genome and synthesize it. So I will give you 2-minute story about how to create a cell factory design, a metabolic cell factory. So you can imagine, we usually given carbon to a microbial cell, the cell will produce any molecule what we are expecting. See, it can synthesize anything, even rubber. What the rubber we are getting in the plantation, we can get the rubber in a microbial cell. And anything you can imagine possible. How it is possible? So what we are doing, metabolic engineering. What is a metabolic engineering? You can imagine quickly, it is not a big thing, though big subject, I am explaining in a very, very lowest possible way. See, this is a cell. This is a cell. So this is carbon, like paddy straw. So paddy straw, you know mushroom. We are growing mushroom in the paddy straw. So mushroom is a fungi. Same way, you can imagine, we are providing carbon. The carbon is taken up into the cell. So it requires 3 genes to the final product D. So each gene encodes for enzyme 1, 2, 3. Finally, you are getting the product D. These A, B, C, these 3 are metabolic intermediates. Now, you want the product E, which cannot be synthesized in a bacteria. A bacteria cannot synthesize. I am giving a good example, then you will understand it. First, you just imagine what I am saying. So, for that, what I am doing, I am deleting the gene 3. Deleting the gene 3. So, no enzyme 3, there is no end product D. Okay. Now, I introduce a new gene 3. So, the new gene 3 encodes for another enzyme 3, which give the product metabolic intermediate D2. It is not an end product. Now, I am introducing another gene 4. Gene 4 on exclusively act on the metabolic intermediate D2, I get the product E. This is known as metabolic engineering. You got the point, no? Now I am giving the right example. You heard about it, 2015, the Nobel Prize awarded to artemisinin. Artemisinin is a molecule derived from a Chinese plant, Artemisia annua. What's the reason I'm explaining? India have plenty of medicinal plants, rarely exploited. So this one plant, China earned huge money, artemisinin discovery. You can imagine, now the question is, this is artemisinin, this is artemisinin annua. Now the question is, can you synthesize artemisinin in a bacterial cell? You got the point? Can you synthesize artemisinin? It is possible. How I explained? Same way. You can imagine, see the play is like this. Sorry. So in the plant, photosynthesis. Photosynthesis convert what? Sugar. Sugar always go through the metabolism. There is a ubiquitous metabolic pathway known as Krebs cycle. Heard about it? TCA cycle. Circle. So this TCA, from the TCA cycle, acetyl-CoA is associated with where? TCA cycle. From the TCA cycle only, secondary metabolism is happening. Farnesyl, pyrophosphate, amorphodiene. You don't need to keep in mind all these words. Only our play is what? The bacteria is having TCA cycle. The plant is having TCA cycle. Now we have to make a bacteria what is missing the pathway. Bacteria do not, TCA cycle is there, acetyl-CoA is there, all the remaining plant metabolic pathway is missing in the bacteria. So you have to synthetically design the genes. So you arrange the genes 1, 2, 3, then transfer to the bacteria. Now bacteria is synthesizing artemisinin. So the point is, any molecule we can engineer and synthesize in a bacteria is known as genome engineering. Okay? I am going to the last part. Oh, moving is little. Yeah, this is what I already explained. Mammoth. Reconstruction of genome and bring back mammoth. Okay, this I already explained. What is? Once we create the life of unnatural nucleotides, that is known as aliens. Now, the last part. See, we are all made up of, see, a human, I told you, human is made up of 3.2 gigabase pair. But did you know your gut is having 100 times higher than the human genome size? The gut is having what? Gut microbiota, everybody knows. Now the few understanding, here I stop it. See, we all born to a mother with microbiota or not? We all born to a mother with microbiota or not? No. We all born to a mother sterile. Okay, then where from microbiota we are acquiring? After birth. Now, I will explain quickly. The problem is, now I ask you the question, can you survive as sterile? So, careful. That's the reason I go and read Darwin's theory of evolution, all these things. Now, the question is, we all born to a mother sterile, can you survive as sterile? Even one day you cannot survive. Even a single day you cannot survive. Why? Got the point? So the reason is, see, while we are born to a mother, we are acquiring the first microbiota from the mother, if it is natural birth. If it is a cesarean, what will happen? So if it is a, now it is actually, government also could not allow the natural birth in the houses. So the birth always in the hospital. So if it is a cesarean baby, the first, or the hospital born baby, the baby is first handled by whom? If it is a baby is born in the house, who is first taking the baby? The grandmother, or suppose the grandmother, I am speaking about the traditional family. Okay, grandmother is taking. So who is first inoculating? You know some microbiota, you know what is inoculation? Who is first inoculating the microbiota to the baby? Those who are first hugging. Who is hugged the baby? Usually a grandmother. So grandmother microbiota is transmitted to the what? Baby. Baby is healthy. The same baby born in a hospital. Who is hugging the baby first? Nurse. So who is inoculating first? Hospital microbiota. You can imagine clearly, by general knowledge you can check it. The baby born in the house always stay in the house. The baby born in the hospital, first 3 years, the baby have a book and always go back to the hospital. Not all. Check it. You can fact check it. Therefore, only now we are discussing about Indian knowledge system, Indian traditional system. Now, see, you might feel that I'm speaking about some tradition. No, it is a well-established science in a journal called the Cell. You heard about the Cell? No, Cell journal. I'll explain you. Cell. What they are saying, it is scientifically proven. The baby naturally born is having high level of bacteria or probiotic bacteria. The baby gut is like the initial, the baby is protected by the microbiota. So the baby by cesarean, the last middle we explain later, cesarean, the baby gut full of pathobions. Because the baby first exposure is from where? Hospital. Therefore, now they come to a conclusion, what is called fecal microbiota transplantation. So the mother's fecal microbiota filtrate has to be exposed first to the baby. The baby is healthy. So this is science. What I am speaking was tradition. So this is called as Indian knowledge system. You got the point? And the last. So they put a terminology, Viksit Bharat. Sir also explained. What is Viksit Bharat? When we become Viksit Bharat, you should understand that the level of research. While you are now, the government of India not giving any funding to, there is a cry throughout the academic system, they are not giving any funding. The cry is because they are not stopped the funding for basic research. Now they expect the research of translational, not applied. So previously we will say applied research means what? We will, at the end of the research, we will say this application, that application. We will say nothing will be there. So now they are asking, you are saying this will have this application. No, then you prove it. That is called technology readiness level. If you at least you have the technology readiness, that is another talk. Sometime we will discuss. So TRL 4 or 5 only they will give you the funding. So while this is a model of China. China always translational. They are now in the. So while India also, all our research become translational, we reach the status of Viksit Bharat. So with this I conclude, wish you all good luck.
Thank you for your invaluable address, sir, by enlightening us about the synthetic genome engineering. As we conclude this memorable inauguration, it is time to express our gratitude. I invite Dr. N. Sendal Kumar, Assistant Professor and Organizing Secretary, to deliver the vote of thanks.
Jai Sri Krishna. Good morning, respected dignitaries on and off the dais. It is indeed my privilege to extend my heart of thanks on behalf of organizing committee of this 5th International Conference on Advance in Biotechnology, Sustainable Innovation towards Viksit Bharat Vision. At the outset, I express my deepest gratitude to our honorable and dynamic secretary of this prestigious college, Sri Ashok Kumar Mundraji, and our ever supportive principal, Captain Dr. S. Santosh Babu, for their constant encouragement and guidance in making this event possible. I am profoundly thankful to our Chief of Honor, Dr. U. Satyanarayana, Director, Research and Professor, Department of Biochemistry, Dr. Panamani Siddhartha, Institute of Medical Science, Andhra Pradesh, for this esteemed presence of sharing this scholarly insight during this inaugural session. My sincere appreciation goes to our chief guest, Joseph Selvin, Dean, School of Science, Pondicherry University, for gracing the occasion and delivering the inspiring address that sets a meaningful tone for the celebration succeeds. I also acknowledge with gratitude the head of the various department and the faculty members who have joined with us today, lending their presence and encouragement. I extended heartfelt thanks to Dr. M.D. Balakumaran, Head, PG Department of Biotechnology, for his continuous support and valuable guidance throughout the planning and execution in this conference. Special thanks are due to the dedicated members of the organizing committee and steering staffs who tireless effort the coordination and outreach ensure the success of this gathering. My appreciation also goes to non-teaching staff of our institution whose behind the science contributions have been indispensable and technical teams. And to the technical team of visual communication, Department of Visual Communication for their excellent coverage of today's inaugural function. The special mention to Mr. Kumbaran for this technical assistance. I would also like to place on the record our thanks data center teams for their invaluable support in the streaming conference seamlessly on the online platform, enabling participation across the border. We are honored by the presence of distinguished scholar, researcher, delegates from the across globes. Your participation, whether through the oral presentation, e-poster, or active engagement in the discussion, enriches our academic discourse. Finally, I wish to acknowledge in deep appreciation the students, volunteers. Your dedication, energy, and teamwork have truly been dividing the force behind the success of this conference. On behalf of the entire organizing committee, extend my gratitude each one of you to making this conference memorable and a meaningful success. Thank you all.
Thank you, sir. Inauguration ceremony has come to an end. Participants are requested to stay a while on their seat for further announcement.
இருளுது நனவு என்னமோ ஏதோ முட்டி முளைக்குது மனது வெட்டி எறிந்திடும் மொழியே முத்து அவிழுது கொடியே நீயும் நானும் எந்திரமாய் யாரோ செய்யும் மந்திரமா பூவே தமிழச்சி என்னமோ ஏதோ you're looking to fly மறக்க முடியலையே என் மனம் அன்று woman so lovely இப்படியே இப்ப உன்னருகில் நானும் வந்து சேரவா என்று lady looking like a Cinderella Cinderella naughty looking with a Cinderella lady looking like a Cinderella Cinderella என்னை வட்டமிடும் வெண்ணிலா lady looking like a Cinderella Cinderella naughty looking with a Cinderella lady looking like a Cinderella Cinderella என்னை வட்டமிடும் வெண்ணிலா சுத்தி சுத்தி உன்னை தேடி விழிகள் அலையும் அவசரம் ஏனும் சத்த சத்த நெரிசலில் உன் சொல் செவிகள் அறியும் அதிசயமேனும் கனா காணத்தானே பெண்ணே கண் கொண்டு வந்தேனோ வினா காண விடையும் காண கண்ணீரும் கொண்டேனோ நிழலை திருடும் மழலை நானோ ஏதோ எண்ணம் திரழுது கனவில் வண்ணம் திரழுது நினைவில் கண்கள் இருளுது நனவில் ஓஹோ ஏதோ முட்டி முளைக்குது மனதில் வெட்டி எறிந்திடும் நொடியில் ஒட்டு அவிழுது கொடியில் குவியமில்லா ஒரு காட்சி பேழையோ உருவமில்லா நாளை ஏனோ குவியமில்லா ஒரு காட்சி பேழையோ அரை மனதாய் விடியது என் காலை ஏனோ குவியமில்லா ஒரு காட்சி பேழையோ உருவமில்லா நாளை ஏனோ குவியமில்லா ஒரு காட்சி பேழை
Vice Chancellor of Research and Innovations at INTI International University, Malaysia. I would request Dr. M. Pavitra, Assistant Professor, to introduce Dr. Wong to the gathering. And before that, the main reason Dr. Wong is here with us is because of Dr. Ranjit from Santa University. Thank you, sir. Thank you so much.
Greetings to one and all present here. It is my privilege to introduce our distinguished speaker for this session, Professor Dr. Wong Shing. He obtained his PhD in Environmental Science from the National University of Malaysia in 2011. A passionate and prolific researcher, his work spans diverse areas in biosensors, bioindicators, photosynthetic microbes, agricultural biotechnology, health sciences, artificial intelligence, and renewable energy. Professor Wong has successfully completed over 25 research projects and has published more than 200 scientific articles. His outstanding research contributions have earned him several prestigious awards at international innovation platforms, and he has also been recognized with the Vice Chancellor Award and the Most Promising Young Researcher Award by Indy International University. Currently, he serves as the Pro-Vice Chancellor for Research and Innovation at Indy International University. Not only his professional stature, but also his gracious personality. With his spirit and passion, we are confident and will certainly not disappoint any of us today. So, I welcome you, sir. Thank you, sir.
I thought I'm going to start my speech.
Thank you, ma'am. I request Dr. M. Pavitra, Assistant Professor, to felicitate Dr. Wong.
I'm on the stage for the second time. What an honor to meet everyone, especially our chief guests and our honorable faculty members, professors, associate professors, and our academicians, our students. How are you? So, how many of us here postgraduates? Can you raise your hand and say yes? Postgraduates. Post. Alright. Undergraduates? Alright. Good. Okay. So, the most important, no matter you are postgraduates or undergraduates, the most important thing is actually if you must time. So we must study hard, right? So come on, give yourself a round. Right now, college is truly a great college. Okay? So you come to the right place. Let me go many places. Look at many different college, university around in India, around Malaysia. I'm from Malaysia, by the way. I went to many other countries which I don't want to mention the name. Right. And I found one truth. Ladies and gentlemen, you are at the right place. Okay? You have very good lecturers, you have very motivated our academicians. Your principal is one of the most dedicated academicians that I never ever met. See? So why not give yourself another applause? Okay. So for everyone interest, I'm from Malaysia. How many of you come from Malaysia before? Went to Malaysia, visit Malaysia before? Oh, great, great. I know, I see some hands. Okay, so not too bad. Alright. So Malaysia is 3.5 hours from here. 3 hours 3 hours 30 minutes from here. Okay, direct flight, huh? So now for all of you Indian citizen, right? Alright. So no, some I heard somebody say that actually Tamil Nadu is not a part of India. No, sorry, politics issue. We are not talking about that. Why you laugh like that? Okay. I know other part of India people when they heard this they don't not happy. Okay. Indian citizen go to Malaysia, no need visa. Okay. So you can visit Malaysia anytime. I'm from Inti International University. Alright. Inti, the word comes from Sanskrit. Aha, very familiar, right? Sanskrit means the essence. Inti. Okay, when you say Inti of something, it means the essence of something. Right? So it is a very long story. I cut it short. Okay, so at the moment we rank 509 worldwide and 122 Asian ranking institutions. Okay, so I'm happy today to bring all my experience from Malaysia to share with everyone here. Okay, so you might find some interesting things, I hope, through my sharing sessions. Okay, we all get inspired and you know what to do. Okay, on your study, you might get some ideas on your project. Okay, so please bear with me another 40 minutes. Let me know if my time is up. Yeah. Okay. Now I'm thinking of how I press the left or if I press the right. Okay, never mind. Alright, so let's start with, because, I didn't touch. I didn't touch anything. Okay. Somebody can you help me to control the slide? Okay, because it's out of control. Okay. So sometimes our life is out of control, but it's okay. We just let somebody help us, okay, to take care of it. Okay, like my wife. Alright, so ladies and gentlemen, I hope we have heard about Viksit Bharat vision. Have you? Oh, I heard about Viksit Bharat in Malaysia 2 weeks ago. You have not ever heard about it. Okay, never mind. It is a vision actually showing that India will become a developed nation by the 100 years of independence by 2047. Okay, okay. Right? So focusing on few things. Right? So I won't read everything. Right? I won't read everything. Basically, the next 2 slides will explain. Come on. The next slide, please. Alright, so then we will talk, when we talk about Viksit Bharat, right? Viksit Bharat, we talk about 4 different group of people that we want to take care of. First is the youth, right? Youth. Who are the youth? You all right? I'm also youth. Okay. I am 18 years old by today. Alright, so the next group is a woman, alright? And then the poor and the farmer. Actually we can see, when we look at, sex and success, alright? Actually it might overlap each other. Okay? So that means somebody can be a youth at the moment, at the same time a woman, at the same time not so rich, at the same time is a farmer. Correct or not? Correct or not? Okay? So it is actually focusing on certain group of people. So when we take a look on the focus, actually I really so say that our Indian government has done a good job. Why? I try to tie Viksit Bharat pillars to our sustainable development goals. You know, heard about UN SDG? Have you heard? United Nations Sustainable Development Goals. Huh? You have heard about that? Okay, look at that photo. This is how it links to each other. Okay, so when we look at it, it seems like Viksit Bharat, when we say that the focus group that we want to take care of, when we take care of their issue, at the same time we take care of sustainable development goals. Okay, so SDG, when we build that, you know, by the way, can you understand my English? Okay, good. Alright, so SDG will build on 2030, right? But I would say it will have an extension. You know why? Because by 2030, none of them, our country in the world will achieve that. No. So it will extend, right? Extend to what time? Some of the countries there might extend to 2050, right? So if India can achieve this by 2047, it will be great. Correct or not? Okay, we are not too late, right? But I will tell you an inconvenient truth. You know, inconvenient, not happy, not make anyone happy, especially me. 2047, I might not be here anymore in this world. Enjoy Viksit Bharat. Alright, cut, next slide. I know Tamil Nadu is going to have election 2 months from now. I'm not saying that the government ahead to stay around because I mentioned that. Alright? Very important. Okay. One thing that I think play a very important role for us to become a developed country. Developed, you know? We have developing country. We have underdeveloped country. We have developed country in this world. Now we want to become a developed country. Okay? So one of the points that is very important is the partnership. Okay? For the goals. Partnership. What do you mean by partnership? Alright. Dear students, I don't want to talk to you, but 2047, maybe the lecturer who got at my age will follow the karma that he right away. Just like me. Okay. So, sorry. Too much, Alright. So, you all will be here. You must partner with each other in order to be successful, in order to bring our country, to become one of the greatest country, to become one of the most developed country in the world. Okay? So, now I give we have our supervisors, we have supervisors. So, do you know, and we have our own lab, right? So think about it. Do you know what others are doing, especially the person next to if you're next door? Another lab, are they doing? Okay, so partnership means we know each other, we work with each other, alright? For the goodness of the society to make the country great, not to make everything great. No, we are in India, okay? So we want to make India great, right? So in Malaysia, I will say make Malaysia great, right? So actually, everyone should be proud and make our country a great one, correct now? Okay, so next please. Okay, so now I want to zoom in. Just now I have mentioned actually in Viksit Bharat we have 4 pillars where we take care of 4 different group of people that actually help us to handle or settle, right, some of the most pressing issues around the world which have been highlighted by sustainable development goals. Now, biotech, right, ladies and gentlemen, how biotech can help. Now I tell you how. Okay, some of my experience, some without my experience I can't share, but this one is my experience. I can show you that first, zero hunger. How many population in this world have not enough food to eat? Do you know the statistics? 1/3? Okay, you check Google. Alright, do you have Gemini, Google? You can check, right? It's huge amount. Okay, while we are sitting here, enjoy this environment, later on we will definitely know our lunch is served there in our cafeteria. We should know as well now in certain part of the world, for example in Palestine, okay, not enough food. Okay, in many parts of the world actually have not enough food. So we don't think about too far away. While we are so nicely, placed in this environment, how many people in our country actually not reachable to enough food, not accessible to them? Alright? So this is important. Okay. But one thing that actually I want to let everyone know, that actually India is one of the very few countries in this world where the food production is self-sufficed. You have enough food, yeah? You have enough food. You believe it's enough food. Okay? Just the distribution of it. Okay, now go back to the biotech. Sorry, I tend to talk a lot of politics. First, what we can do as a biotechnologist, right? First is a climate resilient crops. Okay, we can develop that, right? So when we heard about climate resilient and your brain is stuffed by all the mutants, right? Mutants, X-Men, mutant, genetic modifier. No. Not necessarily you need to do that. Okay? We can have artificial selection. Do you know what is artificial selection? Okay, read. Okay? Artificial selections. They actually need a lot of professional knowledge for it. Right? So first one, okay, we can create some climate resilient crops, yeah? Okay, and then we can do some nutritional enhancement as well, right? Like biofortification of certain staple crops, right? That one we can do, right? So not necessarily we need to put the nutrient into the food, alright? We can add it during the process, right? So this is the part of it. And also the sustainable input, right? For example, biopesticide, biofertilizers, and recycling substrate, right? For the to recycle the waste, huh? Okay, to make it a substrate, so that we can save the resources. So I put the green color there because I have samples with strong sterilization, sterilizing in the right place, so that the fungi won't take over and spread. Okay, that's the job here. But anyway, the job well done. It was successful. Alright. So all the things over there look on the. Okay, I try to press the button. Okay, don't launch the needle. No. Press. Go back. You see. Can you see the red dot? Okay. I can't see it, but anyway. Okay, penny hard. Okay, coconut hard. Palm kernel cake. Banana peel. Okay. So all these look more technology, right? Right. Okay. And sorry for the virus. They failed again. But do you think they will come back? Yes. Okay. So the question will be, we want the better of COVID. What next? Biotechnologies, we should be ready. Okay? So next one is a personalized medicine that we use genomic and genes editing and enable treatments that tailor for individual genetic profile. So just give example, how many of you know the brand called Panadol? Paracetamol. Okay? Paracetamol you know, right? Some of you know. Some of you eat it every day. Why? Because you study too hard. Okay, right? So you have it, you eat paracetamol, okay? It will help you to relieve the pain fast. But I'll tell you, some of us, we might eat one tablet, and you feel like overdose. You have allergy, okay? Symptom already. But some of us might eat up to 2 to 3 tablets, but no effect. Correct or not? Me, I immune to Panadol. You eat that tablets also, the pain is still there. Why? Maybe my genes is not good to Panadol. Gene is CGTA, right? It's like, we can't find the P. P for Panadol, and paracetamol. Okay, that's why. Okay, for me, okay, not for you. Okay, good. The third one is affordable healthcare. Like for example, biomanufacturing. Okay, using microbes and plants that can produce complex drugs like insulin and antibiotics. Okay, we are cheaper price and more sustainable price. Medicines usually is very expensive. Hormones very expensive. How to make it cheap, make it accessible for everyone. So we will have equality. Okay, everyone can get it. Biotechnology is your job. Okay, it's your job. How to mass produce these all things. Okay, the green one, actually we try something on antibiotic. Come on, next slide. Thank you. Alright, so ladies and gentlemen, a few years ago, we done a screening in Malaysia. Alright, one of my, let me see who. Walid is a master student. think what it master's doing. Okay, her job was to collect samples from the hospitals from Malaysia. Okay, you know Agnes Timbers. I had it, alright? Even though I'm already far beyond. I'm not 80 years old, okay? Just now I lied, sorry. Okay. So I just okay. So although I'm far away from 18 years old already, okay. So but I sometimes I still have pimples. Very sad, right? So it looks like the pimples now. The bacteria, there's a bacteria. See this inside. Okay, they become stronger now. Okay, because they become antibiotic resistant. Have you heard about antibiotic resistance? Okay. From your face, I know you heard about it. Alright? Okay, another word. Usually for the bacteria, okay, bacteria, not virus, bacteria. Okay, when they get antibiotic, they will die, right? They will die. You use antibiotic, they will die. But antibiotic resistance means that bacteria, you apply antibiotic, they won't die. They're still alive and singing to you. Okay, so that is about antibiotic resistance. Okay, so we found that actually from CNE sample collected from Malaysia, they are resistant to almost all these type of antibiotic. Okay, all these type of antibiotic. I have given the slides to our organizer. Maybe you can get the slide later. You can read more. Okay, on the research. Okay, so. Antibiotic resistant bacteria. Okay, so we found that antibiotic might not be the best way to kill the bacteria. So we have to work out with some certain novel or another alternative, right, to kill the bacteria. So that's why we come out with this. Okay, we develop a new alternative to deal with bacteria using physical contact. Then we use nanoparticles, right, to try to kill the bacteria here. Okay, so you might want to know more about this, but anyway, this one is done Chang Yubing. Chang Yubing was my PhD student. Okay, so he is now trying to do this. Okay, so he generate the nanoparticles. You know nanoparticles, all right? Generate green nanoparticles, and then we'll try to test it on the bacteria, and actually it have a lot of antibacterial characteristic. Okay, so you might want to do, maybe one day you want to do this. There are still a lot of things to explore, yeah, on this. Come, next one. Okay, good, thank you. All right, so another one. What our biotechnologies can do is about the clean water and sanitation. Okay, so one thing we can do is bioremediation, that I think you should know about it, that we use selected microbes or plants to detoxify polluted water. And of course, another field we can do is the biosensor, by selecting microbes that can be used in low-cost rapid tests to detect pathogens or toxic in the water supply. Okay, let's see what we can do on this. Come, next slide, please. All right, so this is some of the projects that we have done, right, by my student as well, to develop this biosensor for water quality control. The word is so small, sorry. Okay, on the left here, actually it is a set out diagram of a very highly sensitive whole cell biosensor. Whole cell means we use the living cell, live, living one. Okay, whole cell that we designed to detect the heavy metal and pesticide in drinking water. Okay, so this project was a success, and we can detect, okay, some of the heavy metals up to part per billion level, which is in line with the drinking water standard. Okay, so the next one is the highly sensitive carotenoid-based hydrophilic biosensor design for the detection of heavy metal and pesticides. Right. So if we can see, if your eyes is sharp enough, you can see there are 4 tubes here. Okay, or cuvette, yeah. Okay, or container, you can tell it. Okay, with different level of WDT here.
All right. So actually, this sensor is very simple. We just use we extract the carotenoids from the palm oil, right? And then we do the testing based on the hydrophilic. Okay. So maybe I just explain a little bit about this because it's very simple. Okay. So we know hydrophilic and hydrophobic, right? Okay. So if there is a heavy metal or ions, okay, in the water, is it good? No, may not be good. May not be good. I cannot say no good, okay? Or else later mineral water company is coming to sue me and make me bankrupt because of that. Okay, so might not be good. Okay, so when there's a ion in the water, it will increase hydrophilicity or hydrophobicity. Hydrophilicity, right? Okay, so the higher the hydrophilicity, the more the turbidity, like this. Can you see? My pointer is like this. Okay, so if the water is free from any of the heavy metal or ions or things that will increase hydrophilic, it will be like this. Can you see from here? Or like this. This is the empty one. This one. Okay, so with this set out, actually, right? So this type of biosensor, it can last for several months. We just put there for several months. It will be there. Okay, so we no need to use freezer. We no need to use a special dry place to keep it. No, we just keep it in a normal temperature in the room set out. Okay, and then when we add the water into it, it will change. Okay, based on the hydrophilicity of the water. Okay, so that is the I will call in the way that my student is extremely smart. He used he used a very simple theory to come up with something very useful. Okay, so not necessarily we need to come up with great theories, okay, never done before, then we can come up with something novel and useful. No, sometimes simple theories, okay, can be used. Okay, so can we move to the next one? All right, so one more thing is we can do is on the industrial innovation and infrastructure. Okay, so for example, we can develop bio-based manufacturing. For example, production of green material like bioplastic, bio-based chemicals, okay, and then we can do some enhancement of bioprocesses. Like for example, we use biological process as industrial catalysis. White biotech, you know, everyone understand white biotech, right? We have three, right? White biotech, red biotech, and green biotech. Okay, so this is white biotech. Okay, to make industrial process less energy extensive and reduce waste and pollutants. All right, let's go to the next slide. I wish to explain the work, the work by my student. Come next slide, please. All right, so for example, is we can use microalgae cultivation for remediation and biomass production. Two different purposes. First purpose, cultivation for remediation. We reduce the impact of the pollutant to the environment, and at the same time, we will have biomass. Biomass, we can use it for many things. Okay, so later we can explain about that. So maybe I explain here, right? Top. This one is the growth. If you can see, the green one actually is the growth of certain algae in a domestic wastewater, the green one here. And the blue one, which is lower, okay, lower growth rate. Actually, this one is the growth rate, the blue one, of the what we call commercial nutrient that we can buy. All right, like for example from Merck. Okay, we can buy commercial nutrients from there. Okay, so it looks like domestic waste. You know what is domestic waste, ladies and gentlemen? Okay, whatever they come out from our household, the wastewater, that is domestic wastewater. You can imagine how dirty is that, right? So after it go through domestic wastewater, go through the system. The student developed the system, okay, the process. There is a process. All right. So after the process, which use the natural algae, it looks like the the content of the nitrogen, the phosphorus, the next one, ammonia, yeah, have been reduced. This one is before it go through the system, all right, or the reactor. This one is after it, okay, after it. So it looks like the great reduction, okay, in ammonium, nitrogen, and phosphorus. So what is the purpose of this? If we can reduce all these three elements from the water, where the domestic wastewater flow into the river, it won't cause algae bloom, right? Algae bloom. All right. So without the algae bloom, there will be safer place for all the fish and all the living things in the water. So actually, it's a good project here. It has been done by Huang. Huang is my PhD student. Okay, so he is the one who done this. All right. So and next slide, please. Okay. Now let's move on. The next thing, our biotechnologies as a biotechnologies we can do is for the climate action and the life on land or water. Yeah. So for example, we have a carbon capture and utilization, recycling waste or converting into useful product, yeah, like building material. Later on we'll go through this. Next thing we can do is the conservation and restoration. Okay, we can use the genetic tools to help monitor biodiversity, for example. Reducing agriculture emission, for example, we develop fast fix that reduce methane emission from the livestock and crops. Yeah. So let's move on to the carbon capture utilization, which recycling waste or converting into useful products. Okay, thank you. So a group of, this one is by a faculty. This project was done by faculty members, which is our academician did it. Okay, so a group of academicians who work with Indonesia, okay, researchers, they came up with this. They use this agro waste to build composite material. All right, so usually for the building material, all right, something like this. Okay, so we will use a lot of artificial or man-made, all right, chemicals in the process in order to come out with that material, right? So one of the initiative that we have been done by us is we initiate or we develop some chemical modification on biological conformation, okay, for this agricultural waste. And then with this, we actually can replace this green material, the old chemical synthesized material with this new composite material. Okay, so actually this is how the thing's going. Okay, it can't show really very clearly here. Actually, this is a electron microscopic examination on the biocomposite material. It shows that at the usage of 30% of the agro waste, okay, it's okay. The 30% of the agro waste, it shows the best durability, yeah, for the material that generated. Okay, so thank you very much. So let's move on. Okay, so next thing for biotech, you see, biotechnology, with biotechnology we can do many things. Okay, so next thing we can do is on the responsible consumption and production, like SDG 12, right? So one is a circular economy, bioeconomy, yeah? Circular bioeconomy, we try to reduce the carbon footprint by recycle, reduce, and reuse of waste material. For example, turning food waste into useful material, yeah? Later we'll go to this. And also, alternative food and protein, like cultivation of certain food replacement and precision fermentation, aim to drastically reduce the environmental footprint of food production. Okay, so next, let's go to some of the work done by my student. Okay, next one. Okay, so Liang was my master student, graduated. Okay, Chang Yibing. Okay, this one was her master work. Okay, so one of the work that we are doing, okay, or my student done by my student, was using organic waste compound to generate green nanoparticles. We know that nanoparticles now have a high demand. Okay, so there are few ways to generate nanoparticles. Okay, of course, whether you want to say chemically synthesize it, or we can use green synthesizing. How to generate, or how to get this nanoparticle? Actually, we can Google. Yeah. Okay, there are ways. Actually, for these students, these two students, okay, they successfully turn, you know, durian, right? Do anyone know? Anyone doesn't know what is durian? Please raise your hand. Oh, you don't know what is durian? Oh, you know. Oh, okay. Thank you. Everyone should know. Durian is a very famous fruit in Malaysia. Okay, with a very distinct smell. Somebody likes it. All right. Okay, so in certain point it is very unpleasant, but in certain point it is very well accepted, right? Okay, so actually that reflect the life, okay, our life. Okay, sometimes it's positive, sometimes it's negative, but we should take everyone as a whole. That is durian. Okay, so we use the durian husk, the meat we eat. All right, the husk we grind it to become powder. Okay, and then pomegranate. Next one is pomegranate. We also grind it to become husk. A husk become powder. Okay, and then mangosteen. Mangosteen is a purple fruit, purple color. Okay, when you eat it, don't let the color, you know, stain your cloth, or it will stay there forever. All right, so these three fruits, okay, all we use the peel and the husk, we grind it, and actually we successfully synthesize green nanoparticles using this husk powder, yeah, grinded powder. Okay, so this one was done by my master student. Okay, next one. Okay, so ladies and gentlemen, I think we know what to do, and we have some ideas that I will share on what my master student had done. So today I want to show you some data, yeah, for India. This one is India data, yeah, for biotechnology. Actually, ladies and gentlemen, for biotechnology, it is a powerhouse of success to economic growth. Okay, now we see bioeconomy grow for India from 2014 to this is 2014. Okay, 10.3 billion to 2024. What is the number here? 200, 200, right? Okay, about 200 billion USD. 200 billion USD. Okay, so projection. Now we talk about Viksit Bharat 2047. Okay, yeah, Viksit Bharat 2047. Okay, so the amount of bioeconomy or revenue bioeconomy generate will be 300 billion. Okay, so if you can get 0.00001% out of it, you are a millionaire. Got the idea? Got the idea? Never mind. It is a huge number. It is a huge number, okay? That will make us never be so rich before. Okay? So it shows that we are in the right field. We are studying the right things today. Okay? So what we need to do is make our study today relevant to our national motivation or our national policy. I don't know whether you will make it policy or not. Alright, but anyway, this is a drive. Like Malaysia, we have 5 years planning. Okay, our 2020 vision failed terribly. Okay, but we cannot blame anyone but ourselves because we didn't work hard enough. Okay, but that is the truth. However, we always blame politicians. Okay, because you know, human beings like us, we never admit that we are wrong. Me too. So I also say there's the fault of politicians. So we need to change the government. We change the minister. But anyway, okay. Vision is good. Vision is good. So we must study very hard. We must work very hard to make what we are. And also we need to think how to make the things that we are doing now. Relevant, related to the vision of our country. Okay. We must do that. We must do that. Okay. So I think some of the data there is not accurate. Okay, but never mind. It is not important over here. Okay. I think it's more than 1 million jobs. Okay. Move on. Next slide, please. Okay. So another important thing. Just now we talk about the revenue. Revenue is from everywhere. Okay. Now I focus on the food grain production. Food grain is very important. Okay, many people will think that paddy is not important because rice is plain. We can't eat the rice alone. Any one of us still eat rice alone? For me, I won't choose to eat the rice alone. Okay, it is plain. It is something that you know everyone can get. I tell you no. Okay, rice is very important, very important, especially to reflect the strength of a nation. Okay, so a very strong nation or country must have the self-suffice. That means whatever that you produce is enough for the people to eat. Alright, it's important so that you won't be threatened easily by other country. Okay, Malaysia is not a food or self-sufficing country. No, we depend on many countries to get the staple food. That's why, okay, we grow. Okay, we grow, but I would say our growth is not as fast as India. You know why? Because India can produce enough food, so that you don't have to worry about food. You worry about other things, like for example, other things. Okay. Don't worry about the food. Okay, you can worry about the growth of other technologies. Okay, so now staple food. Okay, for example, the grain. Okay, 2000 here. Okay, this is the 196.8 million metric tons. It's huge. Okay, it's a huge here. Okay, that is year 2000. Okay, and until 2023, it already your India produced 330 ton metric grain, which already achieved the target of Viksit Bharat 2047. All right, and I will say in the way okay, this is a data. Okay, you can see just a graph and then the number. Okay, behind this is a lot of technologies. Behind this, a lot of pesticides, a lot of selected grain seeds, a lot of what we call irrigation. A lot of farmers work very hard for that. Okay, so ladies and gentlemen, one more very important thing. when you have enough food for your country, what can you do? You can export the food, right? Export the food. Okay, when you can export the food, it means, now very important, it means you can control others. Got a point? Got a point? Okay, so do you want India to grow to become a very influential country? Okay, yes. US also did that, but they did it by pressing the button, you know. You are not following my way, press button, you know, and then the missile launch, you kill you all. Okay, India don't do that. Okay, you won't do that, I know. Okay, all the India people, good people, we all believe in karma. We won't do that, but we can do one thing. Okay, if you are not good with me, I don't send the food to you. Get the point? You control the food, you control the people. Get the point? Okay, so who can do that? Who can do that? Biotechnologies. All of you can do that. Okay, so make yourself relevant to this. Relevant. Okay, final year thesis, master, PhD, your project, do something. Okay, come, next one. Okay, next one is about healthcare innovation and precision medicine. This is about, I found this actually in the newspaper. Okay, one thing is about the Genome India project. How many know about Genome India project? Actually, we have Genome India project has successfully sequenced 10,000 Indian genome from different part, demographic, okay, different geographical location, different ethnics. Okay, it's done already in order to know how this thing, okay, how the disease will affect population in India. This is very important. Okay, this is very important, and I will say this is a very novel project. Okay, so in future, like for example, COVID strike again, we will know better on how to deal with it in different part of India. Got the point? Okay, I can see this one, nobody have done that. Nobody. Okay, so when nobody have done that, we have done that, this is good. Okay, so ladies and gentlemen, what should we do today to make ourselves relevant to this one, to relate us to this one? Okay, the next one is the vaccine leadership. Okay, it's following the success of this one is a COWIN. I also get it from newspaper. The indigenously developed vaccine. Okay, India is building the robust biopharma sector, focusing on biosimilars, cell therapy, and gene therapy. Okay, this one I got it from here, India Times. All right, so again, our country want to make ourselves a leader in vaccines production. Okay, vaccine, not drugs, not drugs, not chemical drugs. Chemical drug is chemist. Vaccine is something related to biotech. Ladies and gentlemen, now you know about it. Again, what should we do today to make ourselves relevant to this or related to this field? Okay, next. All right, and then one more is about sustainable and net zero goals. Net zero means the carbon emission. Net zero goal. Okay, so circular bioeconomy is very important. So I think in India, I read, okay, correct me if I'm wrong. Okay, dear friends from India, I think you should know better than me on this. Bio E3 policy. Okay, you know, right? Okay, thank you. Okay, so it promotes production of material from waste, bio-based chemicals and enzymes, reducing dependency on fossil fuels and promoting sustainable circular economy. Okay, so this is in the policy of our country. Policy means we should follow. So what can we do today to make us all to follow, to make ourselves following this policy? All right, and then waste management. Okay, so this one I also read that DAO leg. How many of you know DAO leg? I didn't know until I Google it. DAL, down lake. Okay, actually this was a lake that very heavily polluted, right? But through the biotechnology, utilizing the, I think utilizing the algae, okay, that I will say the algae, okay, now the lake is clean. It's clean, very, very clean. And at the same time, they produce biomass, right, to be used in other different sector in biotechnology. All right, okay, next one. Okay, but of course, ladies and gentlemen, this one I won't read everything. I won't read everything. Okay, by looking at the, maybe you don't have to take a look at that. By looking at our environment now, biotechnology become more and more competitive, okay, or many competition now arise. Everyone is trying to fight for the high technology equipment, okay. Everyone is fighting for funding. Everyone is fighting for talents. All right, now what do we have in India now? These three. We have talents, definitely, okay. We have talents, so talent we don't have to share, okay. But two things that we need to share now. First is the high technology. The second one is funding, okay. So what I want to say is, today our university, I want to say on my university as well as INTI International University, we might not have all the high technology equipment. We might have some, but not all, right? You know, I just give you an example. How many of you know FESEM? FESEM. Okay, FESEM. FESEM, one piece is about 500,000 USD. Okay, and every year we might need another 500,000 USD. We might need another 100,000 USD for the maintenance. It's very costly. Okay, that's why I encourage everyone to explore, especially the academician here, okay, to explore what are the resources that we can share with other universities, right? So we can share, we can use it together. We don't have to buy everything, okay? One thing that INTI International University is doing now is by rental. For example, the FESEM that I mentioned just now, all right? If we rent it, all right, one sample is 150 ringgit Malaysia. So it's very, very cheap. Okay, so usually it's more worth it to rent it than to buy a new one. All right, so with that we can do more actually. Okay, so think about ourselves now. The project that we are doing is, can our university, can our college provide all the high technology equipment that we need? It might be yes, it might be no. If our university cannot provide the equipment, we might, don't worry, don't get disappointed, okay? We might use the equipment from another university to rent it, okay? Don't worry, okay? So we don't have to put everything in one university, all right? Okay, so I think that is the point that I want to raise here. Come, next one. Okay, so I want to take this opportunity to introduce a little bit about our university, the university that I come from, INTI International University. 20 minutes from KL International Airport, right? So it's very, very near. Okay, so everyone, if you come to Malaysia, you can try to contact me. Okay, if the time permit, I will maybe arrange, yeah, one tour to our university. Okay, come, next one. Okay, so this is how our campus looks like, academic block looks like. Okay, next one. Okay, so some of the labs, classroom, okay, our facilities, sport facilities. Next one, please. Okay, and I wish to highlight one thing is about the international collaboration of our university. Okay, at the moment our university have the students from 96, okay, 96 countries. It's like a mini United Nation. Okay, so you may want to visit our university to experience the diversity. Yeah. Okay, next one. Okay, so more photos. Okay, that's all. Thank you very much, ladies and gentlemen. If you have any query or you want to get clarification information, you can always contact me. Thank you.
Thank you, Dr. Wong, for your insightful talk. Thank you, sir. We are so honored to have you here. So, sir, you begin. Now Dr. Vijayalakshmi from the Department of Data Science will give a small guest of honor, a token of love to Dr. Wong. Ma'am, sir is coming. Very shortly, we'll be moving on to our plenary session 2.
Mama carry பண்ணிக்கோங்க.
Moving on to plenary session 2, we have Dr. Kain Vadakan Senior Scientific Research Officer at Amala Integrated Medical Research Department, Kerala, I would request Dr. G.K. Saravanan, Assistant Professor, to introduce Dr. Kain Vadakan.
Good afternoon to everyone. It is my privilege to introduce today's speaker, Dr. Kayan Vadakan, who is currently serving as Scientist C at the Amala Institute of Medical Science, Thrissur, Kerala. Dr. Vadakan, a biotechnologist specialist in quantum sensing, biofilm attenuation, synthesis of antimicrobial nanomaterial, and targeted drug delivery system. He completed both UG and PG in biotechnology at Administrative Management College, Bangalore University, and he earned his PhD from Tiruvallur University, and holds Doctor of Science in Biotechnology at Manipur International University and awaiting for the Vivavas examination. Sir holds multiple international patents and registered design for innovative biomedical and environmental technologies. He has authored numerous peer-reviewed publications in high impact Q1 journal and contributed book chapter with leading international scientific publishers. His contributions have been recognized with several prestigious awards, including the International Research Ratna Award for his work in microbial pathogenesis. He has demonstrated proven expertise in interdisciplinary research execution, grant acquisition, and fostering global research collaboration with a strong commitment to translating laboratory innovations into impactful healthcare and environmental solutions. Dr. Kain Vadakan sir will present his work entitled Quorum Sensing Inhibition in Nosocomial Pathogen, a novel antivirulence approach using Pseudomonas aeruginosa as a model, offering a comprehensive and mechanistic perspective on innovative strategies to combat microbial pathogenicity. May I now invite our distinguished speaker, Dr. Kain Vadakan. The session is yours, sir.
Thank you, sir. I request Dr. G.K. Saravanan, Assistant Professor, and Dr. Ashwini, Assistant Professor, to felicitate Dr. Vadakan. Thank you, sir. Thank you, ma'am and sir. Dr. Vadakan's topic is on quorum sensing inhibition in nosocomial pathogens, a novel antivirulence approach using Pseudomonas aeruginosa as a model. Kindly take the stage, Dr. Vadakan.
Good afternoon, everybody. So what is the most important thing currently in research? Can somebody tell me what is the most important thing? The most important thing currently. Sorry? Good. Anything else? But I say the most important thing in the current is finish this talk as soon as possible and let us go for lunch. Isn't it? Okay, so Dr. Ashwini here, she is my academic sister. We both did PhD from same lab and she was my junior. So she made two requests to me. First request was 3 months ago regarding this conference. Next request was 5 minutes ago regarding this conference. She told me, Kain finish in 20 minutes. We need to go for lunch. So no problem. So I will try my best to concise, because most of the time we both used to sit together in the crowd and we used to see our watches asking, why couldn't this speakers complete and why can't they understand our feeling and finish it as soon as possible. So let us consider this talk as an abstract, and hopefully next year I will come again and we'll go detail into the topic. So 1:30 I will finish it up. Is it okay? 15 minutes is fine, right? Okay, so today So today my topic is about bacterial communication. Each other. Have you heard? Okay, the bacteria have the ability to talk to each other. That is what we are going to discuss here. Before that, we should know what is bacteria. We can classify all living organisms into archaea, eukaryota and prokaryota. So bacteria is a prokaryote. So human is the most advanced organism, or bacteria is the most advanced organism? Your any comments? We are more advanced or bacteria is more advanced? Why? Okay, actually I wanted the other answer, then I can prove that bacteria is more advanced. Okay, the same way how we used the old desktop and current laptop. We need different systems for everything, where bacteria is a single microchip thing. And also humans, we originated yesterday in the evolutionary history, but bacteria are there from almost 5,000 million years ago. And bacteria faced almost 5 apocalypses. Even dinosaur died, all the big giant animals died, small animals died, but bacteria survived. And bacteria have that particular gene memory which they transferred. So bacteria is one of the most evolved organism in the entire world. Being a microbiologist, I always tell bacteria is more advanced than human, because humans can't infect bacteria, bacteria can infect us. So let us keep that in mind and let us go to the topic. So bacterial infections is, there are two things in the entire universe that is ubiquitous. One is God, another thing is bacteria. Bacteria and God are ubiquitous, everywhere it is there. Currently there is a world called space. We call it as, what to say, there is nothing in space, but the current studies state that even bacteria are there in space also. That is why we have another biology called as cosmobiology or space biology, where they study about bacteria in space. Okay, so bacteria are ubiquitous, and almost 8.5 million people die because of bacterial infection. That means it is more than the population of 34 countries in the world. Per year, we think that, okay, which disease kill maximum peoples? We tell it is cancer, it is AIDS. No, bacteria is one among them. Bacteria kill 85 lakh people, 8.5 million people per annum, which is more than the cumulative population of 34 countries. So studying bacteria is very much important, even though we will do that only in our MSc level. We will, in MSc project we will do antimicrobial activity, then we all will jump away to different topics. But studying and understanding bacteria is very important these days. Okay, how many of you are there who hasn't got a bacterial infection? Anybody is there? Because then I want your blood to study. Okay, we all have, because sometimes morning when we get up, we'll feel all of you. So first thing what we do is, we have some in chukku kapi or some hot water, gargle something, and next stage we will run to hospital, thinking that it is very safe in the hospital. But unfortunately, the studies says that hospital is the most unsafe place, because there is a saying that you cannot escape from escape. Escape is the terminology of the bacteria. We have grouped them, calling them escape bacteria, which contribute to maximum nosocomial infection. Study says that in India, 1 out of 4 people get nosocomial infection. Have you felt that you go for some leg pain to hospital, you come back with it's there for everybody. So we are facing a big challenge by these nosocomial infections. So, okay, once an infection comes, what is the primary thing we do? Antibiotics. That is the biggest mistake. Okay, we should have gone with that chukku kapi for some more time. But we go with antibiotic, and this scenario happened. We developed an antibiotic, first one in 1930s that time, and that became resistant in 5 years. And this cat and mouse games keep on going. We develop something, bacteria develop resistance. We develop something, bacteria develop resistance. And finally, we stop developing. See this diagram? It says that we have not developed any antibiotics since 1993. So there is now bacteria is getting bored, saying that I have nothing to be more resistant. But still there is a question that how, see, bacteria have lot of mechanisms to prevent our antibiotic. They use some 6 or 7 different strategies against how we against our antibiotics. But still there is a question saying that how bacteria is coordinating all those things. Bacteria is such a small thing. Still, how they are coordinating all this the resistant mechanism. Have you seen the movie Tupaki? You have seen that movie, right? A similar kind of thing is happening here. By the way, we Malayalis watch Tamil movies better than Tamil people. I guarantee you. Once you come to Kerala, you can understand that. We might not go for a Mohanlal or Mammootty movie, but we never miss Surya and Vijay movie. And we are waiting for the next movie. And we ourselves are sad that Mr. Vijay is going to stop movies. You can ask any Malayali sitting here. Okay, so bacteria communicate through a system called as quorum sensing. In this mechanism, what happens is there are some particular genes that produce a chemical signal. That signal will be passed outside the bacteria. Once it reaches a particular threshold level, it will come back. Like that way, they talk to each other. This is called quorum sensing. And how it is similar to the movie Tupaki, I will explain. So think that this is a hall. Can all of you close your eyes for 30 seconds? Can all of you close? Do you know how many people are there in this? But if one by one, if we are telling our names, we know that, okay, this many people are there. Like that, one bacteria cannot understand the presence of other bacteria inside a system. So they remain silent until it finds its friend. The same way in Tupaki, sleeper cells are waiting for a command. The same way they are waiting for a command. So bacteria are similar to bio sleeper cells. They remain in our body until they get their that's my eye pointer. Bio sleeper cells. So they remain in our body. So until they get a command. This command is happening through this quorum sensing. That's why we call it as the mechanism and the mother of all infections. So this is one quorum sensing circuit. See, I will take particularly in Pseudomonas. See, this is the quorum sensing mechanism. By this mechanism, see, it is affecting all bacterial virulent mechanism. So it is kind of main switch. That one switch that activates every virulent mechanism. So how can we combat bacteria? Okay, how many lights are here? How many lights are here? 1, 2, 3, 4, 5, 6, 7, 20, 25. It is easy. Switch off all the light or switch off the main switch. Main switch. Especially now we will shut down the entire building and go. Okay. Similarly, you don't have to fix separately all the things. Just shut down the quorum sensing gene. Then the bacteria is a silent sleeper cell till its end. So our body at time, see, we have an immune system. Still, why we are being infected with bacteria? Because of the immune system is not getting enough time. So by this, we can make the bacteria silent and our immune system will get sufficient time. That is the mechanism against this quorum sensing inhibitory drugs. See, so they are like this, they are blind until they understand. So that is the time where we can target them. So this quorum sensing basic drugs basically target the mechanism. They will not kill directly, so that bacteria cannot develop a resistance towards that. But our immune system is producing tailor-made antigens, antibodies against that. So there are 5 different methods where we can stop that. We can either stop the production of that particular signaling, or we can bind with some mechanism so that it cannot re-enter, or we can bind the receptor gene. Like that, different mechanism we use to inhibit this quorum sensing, by which we can silence the bacteria. So here what is happening means, see, this is the infection point. Usually bacteria will have a silent phase, then it go to virulence. That silent phase is this gaga phase. Then infection phase is that phase. Okay, here what is happening is the silent phase is been extended. We will only have that phase. Meantime, our immune system will get enough time to act against the bacteria. So it's a kind of a, we are not, what to say, we are not directly killing the bacteria. We are complementing our immune system to kill the bacteria. That is what in the strategy it is happening. So we have a lot of inhibiting molecules that are structurally similar to that. So I will take one case study. So in the study, we used do you know Solanum torvum? That plant, have you seen? It is very famous in Tamil Nadu. We call it as Sundanga. Okay, Solanum torvum, that devil's berry, turkey berry, green color. We make poriel with that. Yeah, I did my PhD from Tamil Nadu. I know, I used to eat. So that one. So that extract we took and find out its ability to regulate the system. We found out that it is successfully deregulating all the quorum sensing traits like biofilm formation, pyovidin formation, protease formation, etc., etc. So we took it into a larger scale. We found out what is happening in rat models. So we had some 4 different groups of rats. First one was a negative group, then was a positive control where we infected but we did not treat. Third one was an antibiotic which is commercially available. Fourth one was our own drug. We got some very interesting results that in first 3 groups the infection developed, which when we challenged, the infection developed, and in next after 7 days in the untreated group the infection was very high. Antibiotic treated group the infection was very less, and the quorum sensing inhibitory drug had, okay, it was not that much but there were infection. But on the 14th day, by antibiotic treatment and this quorum sensing inhibitor treatment, it was almost same. But one interesting factor was in this is the unchallenged thing. The challenged group was dead within 10 days because we didn't treat them, we let them die. And this is antibiotic treated cell where we cannot see any immunological action there. No inflammation, no macrophages, nothing. Whereas here we can see the residues of immune mechanism, which we understood that the own immune system of the rat started to act. So we found out that this is one safe mechanism through which we can complement the immune system. And we found out that this particular molecule which we developed have more affinity than its own signaling molecule. So suppose the signaling molecule along with our inhibitor molecule come, the bacteria will show more affinity towards the inhibitor molecule. So the inhibitor molecule will go and attach where the signaling molecule is supposed to be, and the entire mechanism is being disturbed. So we developed the pathway and we found out that all in the first diagrams I showed, it is helping lot of things. All the virulent mechanisms have been stopped. So it is kind of the main switch. This kind of targeted drug where it's using as a main switch. So this is what a brief, I should say it's a brief introduction due to time limit, the quorum sensing mechanism and how we are going to treat it. So here basically once the infection occur, we are not killing the bacteria, but we are giving time to our immune system and we are promoting perfect immunological clearance. This is the strategy where we use and where we compact antibacterial sensitivity. Thank you very much, and let's hope to meet soon. Okay, if there is any question, I think I can take 1 or 2 if there is any. Yeah, please. Okay, maybe I can sir, 1:30 what questions? Please get out from here. No, anyway, thank you very much for your time. So if there is no question, can we sum up this session? So thank you very much. Let's meet again. Bye bye.
Thank you, Dr. Kayan Vadakan, for sharing his enlightening, impactful, and visionary speech that has profoundly enriched our understanding and inspired future research. Time for lunch. Lunch is provided outside the auditorium. We will reconvene shortly after half an hour with more exciting sessions. Thank you.
ஐயோ ஆஃப் பண்ணு. ஆஃப் பண்றது எங்க இருக்கு? அங்கதான் இல்ல பட்டன் சரி என்ன பண்றது? 1 2 3
ஹலோ ப்ரோக்ராம்னு வச்சா 1:30 மணிக்குள்ளே முடிச்சிருங்கப்பா. 4:30 மணின்னு சொல்லிட்டு 5:30 மணி வரைக்கும் பாருங்க, டென்ஷன் ஆகிடும். ஹெச்ஓடி சார் எங்கே இருந்தாலும் ஆடிட்டு வரது வர வேண்டும். இப்படிக்கு பயோடெக்னாலஜி என் நெஞ்சில் குடியிருக்கும்
அந்த மலேசியன் பேசினாரு புரிஞ்சுதாங்க அவங்க ப்ரோக்ராம் அங்கே கெஸ்ட் ரூமில் ஏதோ கேட்டாரு என்ன பார்த்தேன் திரும்ப கேட்டாரு அத்தை நிமிஷமும் பார்த்தேன் எல்லாம் சீரியஸ் ஆகிட்டு சார் நம்ம அந்த நான் பண்ணி
ஹெச்ஓடி சார் எங்கே இருந்தாலும் ஆடிட்டோருக்கு வர வேண்டும். கேமராவில் பார்ப்பாங்களா? பார்க்க மாட்டாங்கண்ணா. இப்படிக்கு பயோடெக்னாலஜி மாணவர்கள் மாணவன் சச்சின் செகண்ட் யூஜி பயோடெக்னாலஜி துவாரகதாஸ் கோவர்தாஸ் வைஷ்ணவ் காலேஜ். விஐபி ரூமில் எல்லாம் சாப்பிட்டுக்கிறாங்க, யாரும் போகாதீங்கப்பா. போனால் விஐபி ரூமில் போனால் சாம்பார் வாசனை அடிக்குது, யாரும் போகாதீங்க. ஏசி யாரும் போய்
Welcome back. Our next speaker is Dr. Julian Jean Mallard Adam from IFP, Institute Francais de Puducherry. Dr. Adam will discuss வளர்ந்த நாட்டுக்காக விவசாயிகளின் பாரம்பரிய அறிவு மூலமாக நிலையான விவசாய புதுமைகள். I'd request Dr. S.N. Nisha, Assistant Professor, to introduce Dr. Adam.
Thank you, Benny. I am extremely delighted and honored to introduce the speaker of the plenary lecture session 3, Dr. Julian Mallard Adam, researcher, Institute of France, Puducherry. Hope all of you would have noticed the title of this session being given in Tamil. That is to respect his affinity towards our language. So with the same respect, I now take the liberty to continue introducing Dr. Adam in Tamil, which should be his preference as well. முனைவர் ஜூலியன் மேலட் ஆடம் ஒரு இயற்கை வேளாண்மை ஆராய்ச்சியாளர், ஆர்கானிக் ஃபார்மிங் ரிசர்ச்சர். இவர் மேக்கில் யூனிவர்சிட்டி கனடாவில் 2020ஆம் ஆண்டு PhD பட்டம் பெற்றார். தற்போது டாக்டர் ஆடம் தமிழ்நாடு வேளாண்மை பல்கலைக்கழகம் கோயம்புத்தூரில் அட்ஜங்க்ட் ப்ரொஃபஸராகவும் பிரான்ஸ் நாட்டின் வெளிநாடு மேம்பாடு ஆராய்ச்சி நிறுவனத்தில் ஆராய்ச்சியாளராகவும் பணியாற்றுகிறார். இவருடைய ஆராய்ச்சி ஆர்வம் இயற்கை வேளாண்மை, that is organic farming, பூச்சி மேலாண்மை, that is pest management, உணவு பாதுகாப்பு, which is food security, மற்றும் விவசாயிகளின் பாரம்பரிய அறிவு, that is traditional knowledge of farmers போன்ற துறைகளில் உள்ளது. முனைவர் ஆடம் நம்மிடையே இருப்பது நமக்கு மிகுந்த மகிழ்ச்சியும் ஊக்கமும் அளிக்கிறது. All of us should take his presence as a motivation to value our culture, tradition and language. முனைவர் அவர்களை பணிவுடன் வரவேற்று விரிவுரை வழங்க அழைக்கிறேன். Thank you.
Thank you, ma'am. I request you to felicitate Dr. Adam. Dr. Adam will enlighten us on sustainable agricultural innovation through farmers' traditional knowledge for a developed India. The floor is yours, Dr. Adam.
ஆரம்பலாமா? அனைவருக்கும் வணக்கம். இன்று உங்களோட மாநாட்டில் கலக்கிறதுக்கு வாய்ப்பு எனக்கு கொடுக்கறதுக்கு மிக்க நன்றி. நான் ரொம்ப மகிழ்ச்சியாக இருக்கிறேன். இன்று தமிழ் என்னோட தாய்மொழி இல்லை என்று எனக்கு தெரியும். உச்சரிப்பு கொஞ்சம் வினோதமாக இருக்கிறது. அதனால் மெதுவாக தெளிவாக பேச பார்க்குறேன். ஒருவேளை முடியவில்லை என்றால், ஏதோ புரிய முடியாது இல்லை என்றால் கண்டிப்பாக எனக்கு சொல்லுங்கள். ஆரம்பத்திலிருந்து நான் உங்களுக்காக மன்னிப்பு கேட்குறேன். என்னோட ஆராய்ச்சி நான் சில விதமான நாடுகளில் செஞ்சிருக்கிறேன். எல்லாம் வேளாண்மை சம்பந்தமானது. இது சரி. இந்த நாடு வந்து இஷிமுலே என்ற ஒரு நாடு. அந்த நாட்டுக்கு இதே நாடு மாதிரி நிறைய பேர் வச்சிருக்கிறது. இஷிமுலே ஒரு பேர், குவாத்தமாலா இன்னொரு பேர், பஷில் இன்னொரு பேர். இவங்க வந்து மேக்குஷ் ரமீரஸ் என்னோட ஒரு நல்ல நண்பர். என் கூட இந்த ஆராய்ச்சியும் செஞ்சிருக்கிறார்கள். நான் உங்களுக்கு இன்று காமிக்க போகும் ஆராய்ச்சி இவங்களுடன் செஞ்சிருக்கிறேன். அப்போ வளர்ந்த நாட்டை பற்றி, முன்னேறிய நாட்டை பற்றி பேசும் பொழுது இந்த வேளாண்மை துல்லியமான வேளாண்மை வந்து ப்ரிசிஷன் அக்ரிகல்ச்சர், இல்லை என்றால் அறிவாளி வேளாண்மை, ஸ்மார்ட் ஃபார்மிங், அந்த மாதிரி வார்த்தைகள் அதிகமாக வருகிறது. இந்த 2 வார்த்தைகள் நான் கூகுளில் போட்டு என்ன வருதுன்னு பார்த்தேன். நீங்கள் பார்க்குறீங்கன்னா இல்லை என்ன தெரியுது, கொஞ்சம் ஒரு ட்ரோன் இருக்குது, கைபேசி இருக்குது, இணையதளம் இருக்குது. யூடியூபில் தமிழ் தெரியாதவர்கள் சிலர் இருக்கட்டும் என்று சொல்லியிருக்காங்க எனக்கு. அதனால் இது மறுபடியும் ஆங்கிலத்தில் சொல்ல போகிறேன். When I look when you think of the term smart agriculture and precision agriculture that are often used when talking about agricultural sustainability or development, we often think along the lines of technology, internet, mobile phones, drones. These are the pictures that came up first when I typed the words into Google. ஆனால் அதில் காணும் என்ன கணக்கு தெரியல. அதில் ரொம்ப முக்கியமான ஒரு விஷயம், விவசாயி எங்க? So this discourse really tends very much to forget the farmers. இந்த துல்லியமான வேளாண்மை வந்து உணரி மூலம், ட்ரோன் மூலம் இந்த பண்ணை மேம்பாட்டு விவசாயியோட கையிலிருந்து எடுத்து ஏதோ ஒரு செயற்கை நுண்ணறிவுக்கோட கட்டுப்பாடுக்கு கீழே வைக்க பார்க்குறது. These terms really try to disenfranchise farmers and place decision making in the hands of an algorithm or a foreign company. அதற்கு பதிலாக இந்த நாட்டில் மிக முக்கியமான ஒரு பதில் இருக்கு. அது இயற்கை வேளாண்மை. எல்லா நாடுகளில் ஓரளவுக்கு வருகிறது. நான் சொந்த ஊர் கனடா, அங்கேயும் இயற்கை வேளாண்மை இருக்கு. நீங்கள் கடைக்கு போறீங்கன்னா, சந்தைக்கு போறீங்கன்னா ஏதோ ஒரு பகுதி எல்லாம் இயற்கை வேளாண்மை எப்படி போட்டிருப்பாங்க. ஆனால் இந்த நாடு மாதிரி பொது கொள்கை நாங்கள் அந்த அளவுக்கு கொண்டு வரல, முன்னால் கொண்டு வரல. அந்த புலத்தில் இந்த நாடு தானே ரொம்ப முதல்ல வருகிறது என்று நான் பார்த்திருக்கிறேன். அங்கே என்ன பார்க்குறோம், வந்த வெவ்வேறு செடிகள், ஒரே வழியில் வெவ்வேறு செடிகள் இருக்குது. அதில் டிராக்டர் கொண்டு வர முடியாது. அதனால் சிறு குறு விவசாயிகளுக்கு பொருத்தமான ஒரு செய்முறை இது. And these technologies are a lot more adapted towards small farmers. It's very difficult to do organic or natural farming with machinery or large areas of land. அதாவது இந்த சின்ன அளவிலான நிலத்தில் நிறைய உற்பத்தி செய்ய முடியும். ஆனால் ஒரு விவசாயியால் ஒரு சின்ன பகுதி மட்டுமே பார்க்க முடியும். அதுக்கு மாறாக செயற்கை வேளாண்மையில் ஒரு விவசாயியால் பெரிய அளவான நிலத்தை பார்க்க முடியும், பெரிய டிராக்டரில் சுற்றலாம். ஆனால் அந்த நிலத்துக்கு உற்பத்தி குறையா இருக்கும். ஏனென்றால் ஒரே ஒரு பயிர் மட்டுமே போட முடியும். இதில் கீழே கீரை, அதுக்கு மேலே தக்காளி, அதுக்கு மேலே பப்பாளி, அதுக்கு மேலே தேங்காய், அந்த மாதிரி கலந்து போடலாம். இது செயற்கை வேளாண்மையில் செய்ய கஷ்டம். அவருடன் அறிவுகள் இருக்கு. இது வந்து ஒரு ஜீவாமிர்தம் தயாரிக்கிறாங்க. ஜீவாமிர்தம், பஞ்சகாவியம், இந்த மாதிரி பூச்சி விரட்டிகள், உரங்கள், நுண்ணுயிரி ஊட்டி வந்து பயோஸ்டிமுலண்ட். அதாவது அதில் நுண்ணுயிரிகள் இருக்கும். மண்ணுக்குள்ளே இருக்கும் நுண்ணுயிரிகளுக்கு ஊக்கம் கொடுக்கும். அதாவது செயற்கை உரங்கள், செயற்கை பூச்சிக்கொல்லி தெளிக்கிறீங்கன்னா மண்ணுக்கு மேலே மட்டுமே பாதிக்காது, மண்ணுக்கு கீழேயும் பாதிக்கும். அப்போ திருப்பி இயற்கை வேளாண்மைக்கு வர விரும்புகிறீங்க என்றால் கஷ்டமாக இருக்கும். ஏனென்றால் அந்த மண் ஏற்கனவே பாதிக்கப்பட்டது. திருப்பி மகசூல் வர்றதுக்காக 2-3 வருஷம் ஆகும் என்று இருக்கலாம். நீங்கள் பணக்கார விவசாயி இருக்கீங்கன்னா பிரச்சனை இல்லை. குழந்தைகள் ஏற்கனவே ஆஸ்திரேலியாவில் படிச்சிருக்காங்க, அவங்க அப்பா அம்மாவுக்கு காசு அனுப்புகிறாங்க மாதமா 3 வருஷம் நஷ்டம் வந்தாலும் பிரச்சனை இல்லை. ஆனால் முக்கால்வாசி சிறு குறு விவசாயிகளுக்கு அந்த வசதி இல்லை. 3 வருஷம் கழித்து திருப்பி பணம் கிடைக்கும்னு சொன்னாலும் பட்டாது, அடுத்த மாதம் தானே பணம் வேண்டும். அப்போது இந்த செயற்கையிலிருந்து திருப்பி இயற்கைக்கு கொண்டு வர்றது ரொம்ப கஷ்டம். அந்த காலத்தில் இந்த மாதிரி பஞ்சகவ்ய ஜீவமிருத்த மாதிரி தொழில்நுட்பங்களை பயன்படுத்தி இந்த 3 வருஷம் குறுக்கிட்டுரும். கொஞ்சம் சில தடவை 1 வருஷம், சில தடவை 6 மாதங்களுக்குள்ள இந்த இயற்கை வேளாண்மையோட பலன்கள் நேராக விவசாயிக்கு கிடைக்குமாறு பண்ணலாம். அதுடன் செய்யும் முறைகள் மட்டுமில்லை, அதாவது மாதிரி பூச்சி விரட்டிகள், நுண்ணுயிரி ஊக்கிகள் தவிர விதை ரொம்ப முக்கியமானது. பலக்கணக்கான விதை இருக்கு. இந்த நாட்டில் பலக்கணக்கான நெல், பல சிறுதானிய விதைகள் கிடைக்கும் என்ற இது இந்த நாட்டோட முக்கியமான உணவு. நீங்கள் வேறு நாட்டுக்கு போறீங்கன்னா அங்கே அவங்களோட முக்கியமான பயிர்களோட விதை கணக்கு அதிகமாக இருக்கும். இப்போ இதுக்கு வருவேன். அதுடன் இந்த தொழில்நுட்பம் மட்டுமில்லை, அதை எப்படி பயன்படுத்தணும்னு தெரியணும். இங்கே நெல் கோட்டை ஒன்று இருக்குது. விவசாயிகள் அதுக்குள்ளே அவங்க நெல் விதை போடுறாங்க. அதில் போடுறாங்கன்னா அதுக்கு பூச்சி வராது, நோய் வராது, அதில் பூஞ்சை எதுவுமே வளராது, முழுங்காது. அடுத்த ஆண்டு அதை உடைச்சு விதை எடுத்து மறுபடியும் நட்டுவாங்க. இந்த மாதிரி அறிவுகள் சில விவசாயிகளுக்கு தெரியும். ஆனால் முக்காவாசி கடைக்கு போய் கலப்பின விதை ஹைபிரிட் சீட்ஸ் அது வாங்கிட்டு நட்டுட்டு திருப்பி அதே கடை அடுத்த ஆண்டு போவாங்க. உடல்நிலைக்கு நஷ்டம், அது மட்டுமில்லை மண்ணுக்கு நஷ்டம், அது மட்டுமில்லை பொருளாதாரத்துக்கு நஷ்டம். ஏனென்றால் விவசாயிகளுக்கு இந்த தொழில்நுட்பம் வாங்கி நிறைய செலவு அடைக்கிறாங்க. வரமாட்டேங்கிட்ட சரி. ஆயினும் உங்களுக்கு என்னை விட இந்த நாட்டை பற்றி அதிகமாக தெரியும் என்று நினைக்கிறேன். அதனால இங்கே விட நான் இன்னொரு வெளிநாட்டை பற்றி பேச போகிறேன். இஷிமுலா நாடு, அது முதல்ல சொன்னது குவாட்டமாலா நாடு. இங்கே மெக்சிகோ நாடு தெரியுமா? மெக்சிகோ. அதுக்கு தெற்கு அடுத்த நாடு இஷிமுலே குவாட்டமாலா. இந்த நாடு மக்கத்தொகை 1.5 கோடி, ரொம்ப குட்டி நாடு. ஆனால் அதற்குள்ள மக்களோட பணவகைத்தன்மை, பையனோட பணவகைத்தன்மை ரொம்ப அதிகம். நாங்கள் முறை போகும் பொழுது என்னோட பேராசிரியர், முனைவர் பட்ட பேராசிரியர், அவங்க இந்த நாடு இஷிமுலா நாடு சேர்ந்தவர். அவங்க எனக்கு என்ன சொன்னாங்க, நீ அந்த நாட்டுக்கு போகும்போ முழு பாரத நாடு சுருங்கிக்கிட்டு அப்படி ஒரு குட்டி நாட்டுக்குள்ளே போடுறேன்னா உனக்கு இஷிமுலா நாடு தான் கிடைக்கும். அங்கே மலை இருக்கிறதால ஆப்பிள் சாகுபடி செய்யும் இடத்திலிருந்து வாழைப்பழ சாகுபடி செய்யும் இடம் வரை 12 நிமிஷம் ஆகும், அவ்வளோதான். ஆனால் இந்த நாடு வளர்ந்த நாடு சொல்ல மாட்டாங்க, அது கொஞ்சம் எளிமையான நாடு அப்படி சொல்லுவாங்க. ஆனால் கொஞ்சம் யோசிச்சால் வளர்ந்த நாட்டு, முன்னேறிய நாட்டோட அர்த்தம் என்ன? நம்ம வளர்ற நாடு ஆயிரம்னு சொல்கிறோம், ஆனால் அந்த வளர்ச்சியோட அர்த்தம் என்ன? கனடா என்னோட நாடு வளர்ந்த நாடுன்னு சொல்கிறாங்க. சில தடவை சொன்ன நாடு பார்த்து அது வளர்ந்த நாடு எனக்கு தெரியல. அப்போது உலக வங்கிக்கு கேட்குறீங்கன்னா வளர்ந்த நாடு அர்த்தம் பணம், அது மட்டுமே பார்ப்பாங்க. இது முக்கியமான பகுதி இருக்கிறாரு, திருப்பி ஆங்கிலத்தில் சொல்கிறேன். When we talk about development, it's important to think about what we mean by developed. My country, Canada, is considered a developed country. Sometimes when I look at the roads or the state of other parts of the country, I don't feel it's very developed. Guatemala is not considered a developed country. So what's the definition? What development are we striving for? If you ask the World Bank, it's money, GDP. அதாவது மொத்த உள்நாட்டு உற்பத்தி. இஷிமுலா நாட்டில் மக்களுக்கு பசி அதிகம். 10 குழந்தைகளிலிருந்து 5 குழந்தைகளுக்கு சாப்பாடு கிடைக்காது, பசியுடன் தூங்குறாங்க. அது மட்டும் இல்லை நாங்கள் இருக்கும் இடத்துல 10 கூட பசியோடன் தூங்குறாங்க அங்கே. அது வளர்ந்த நாடு கிடைக்கல. ஆனால் எவ்வளோ பணம் இருக்குன்னு பார்க்குறீங்கன்னா இங்கே வந்த இஷ்யூல இந்த மஞ்சள் கோடியில் வருது. 1960 2014 வரை ஹொண்டூராஸ் நாடை விட எவ்வளோ அதிகமான பணம் சம்பாதிச்சு இருக்காங்கன்னு பாருங்க. ஆனால் ஹொண்டூராஸ் நாட்டில் பசி இஷ்யூல நாடை விட பாதி. அப்போ பணம் எவ்வளோ இருக்கு இல்லையா என்று பார்த்து வளர்ந்த நாடோ இல்லையா என்று சொல்ல முடியாது. இன்னொரு கருத்துல எதுக்கு முக்கியத்துவம் கொடுக்கணும்? மக்களுக்கு சாப்பாடு கிடைக்குதா, சத்தான உணவு கிடைக்கிறதா, மருத்துவர் கிடைக்கிறதா, கல்வி கிடைக்கிறதா, நல்லா வேலை கிடைக்கிறதா? அதையெல்லாம் கிடைச்சிட்டேன்னா அப்போ உள்நாட்டு உற்பத்தி எவ்வளோ குறையோ அதிகமோ நமக்கு என்ன? அதான் நம்ம நோக்கம் தான் இருக்க வேண்டியது. இன்னைக்கு காலையில் இந்த நோக்கங்களுக்கு விவசாயிகள், மகளிர்கள், இளைஞர்கள் ஏழ்மைக்கு முக்கியத்துவம் கொடுக்க போவாங்கன்னு பார்த்து நான் மிக்க மகிழ்ச்சி அடைச்சேன். அப்போ திருப்பி ஷிம்லா நாட்டுக்கு போனால் அங்கே வேளாண்மை சம்பந்தமான என்ன ஆராய்ச்சி பார்க்குறீங்கன்னா அங்கே ஒரு ஏரி இருக்கு. நாங்கள் இருக்கோம் இடத்துல அந்த வரப்படத்தில் பார்த்தீங்களா இல்லையான்னு எனக்கு தெரியல. அந்த ஏரி பேர் அத்திட்லான். அப்போ இந்த நாட்டுக்கு எதுக்காக இஷ்யூலே என்று பேர் போட்டு இருக்கோம் என்று கேட்டால், இந்த வார்த்தை கட்சிக்கல் மொழி வார்த்தை. அதற்கு 2 வார்த்தை பகுதி. ஒரு இஷிம் மக்காச்சோளம், உலேவ் நாடு. நாடு இல்லை என்றால் பூமி. அந்த 2 வார்த்தைகளுக்கு மண் இல்லை 3 வார்த்தைகள், மண், பூமி, நாடு. ஒரே ஒரு வார்த்தை கட்சிக்கல் மொழியில உலேவ். ஏனென்றால் மண், பூமி இல்லாத நாடு கிடைக்காது இல்லை. எதுக்கு இஷிமுல மக்காச்சோளம் நாடு சொல்றாங்கன்னா, இங்கே அரிசி, சிறுதானியங்கள் எவ்வளவு முக்கியமானது, அங்கே மக்காச்சோளம் தான் முக்கியமானது. அங்கே லட்சக்கணக்கான இல்லை ஆயிரக்கணக்கான வகையான அரிசி மக்காச்சோள விதைகள் இருக்கும். அது தினமும் 3 முறை சாப்பிட்றாங்க. அது மட்டும் இல்லை, அவங்களோட மதத்தில் ஒரு முக்கியமான பங்கு எடுத்து இருக்கிறது. அவங்க மதப்படியாக கடவுள் மனிதர்களை மக்காச்சோளத்தாலே உருவாக்கியிருக்கிறார்கள். அங்கே பல விதமான நிறம் மக்காச்சோளம் கிடைக்கும். கருப்பு, சிவப்பு, மஞ்சள், வெள்ளை. அதுவும் மனிதர்களோட நிறம் மாதிரி இருக்கிறது அப்படி சொல்றாங்க. வேளாண்மைக்கும் செய்முறைகளும் கலாச்சாரத்தையும் பிரிக்க முடியாது. இந்த துல்லியமான வேளாண்மை, அறிவிசாலி வேளாண்மைக்கு எனக்கு எந்த பிரச்சனை இருக்கு இது வேளாண்மை ஒரு தொழில் ரீதியான வேலை உருவாக்க பார்க்கிறது. ஆனால் அதுக்கும் மக்களோட அறிவுக்கும் மக்களோட கலாச்சாரத்துக்கும் சம்பந்தம் பார்க்காமலே முன்னால் போகிறது. இஷிம்லா நாட்டில் இந்த வரைபடத்தில் இத்தனை வெவ்வேறு மொழிகள் இருக்குதுன்னு பாருங்கள். மொத்தமாக 26 மொழிகள், 1.5 கோடி மக்கள் தொகை நாட்டுக்குள்ளே 26 மொழிகள். அதுடன் 3 முக்கியமான மொழி குடும்பங்கள் இல்லைங்க, 4 முக்கியமான குடும்பங்கள். அதில் மயப் என்ற ஒரு குடும்பத்தில் 3 மொழிகள் கட்சிகள் இச்சேத்துறையில் நாங்கள் வேலை செய்யும் இடத்துக்கு பக்கத்தில் பேசுவாங்க. அங்கே ஒவ்வொரு கிராமத்துக்கு சொந்த உச்சரிப்பு இருக்கும், சொந்த துணி இருக்கும். உங்களுக்கு கொஞ்சம் பழக்கம் வருதுன்னா ஒருத்தரை பார்த்துருந்தே அவங்க எந்த மொழியில் பேசுகிறாங்க, எந்த உச்சரிப்பில் பேச போகிறாங்க என்று அவங்க துணி பார்த்து தான் சொல்ல முடியும். இமேலே கிச்சே எனக்கு தெரிவரை இது கிச்சே துணி உறுதியில். இங்கே கண்டிப்பாக கட்சிக்கல் ஒரு துணி துலகியா ஊரிலிருந்து. முக்காவாசி நாடு பழங்குடி மக்கள். இதே மாதிரி ஒவ்வொரு கிராமத்தில் வேறு கலாச்சாரம் இருக்கிற மாதிரி, வேறு மாதிரி விதை, வேறு மாதிரி மண், வேறு மாதிரி வேளாண்மை செய்முறைகள் இருக்கும். அவங்க என்ன சொல்கிறாங்க, நீங்கள் 15 கிலோமீட்டர் தூரத்தில் இருந்து ஒரு விதை கொண்டு வரீங்கன்னா, இல்லைங்க 15 கிலோமீட்டரில் 15 நிமிஷ தூரத்திலிருந்து ஒரு விதை கொண்டு வரீங்கன்னா அந்த விதை இந்த மண்ணில் வளராது. அந்த அளவுக்கு வித்தியாசம் இருக்குது காலநிலையிலும் மண்ணிலும். அப்போ இங்கே நீங்கள் பார்க்குறீங்கன்னா இந்த இது மக்காச்சோளம் பயிர் பாரம்பரிய சாகுபடி செய்யும் முறை. கீழே இங்கே பருப்பு. எதுக்காக பருப்பும் மக்காச்சோளமும் சேர்ந்து வளர்த்துறாங்க? யாருக்கு தெரியுமா எதுக்கு இந்த 2 பயிர் சேர்ந்து போடுறாங்க? அவங்க தலைச்சத்து மக்காச்சோளத்துக்கு மிக்க முக்கியமான ஒரு சத்து ஆகும். மக்காச்சோளத்துக்கு நிறைய தலைச்சத்து வந்து நைட்ரஜன். ஸோ maize requires lots of nitrogen to grow. அதே மாதிரி இந்த பருப்பு அதுக்குள்ளே ஒரு சின்ன நுண்ணுயிரி இருக்கும் அந்த வேருக்குள்ளே. அந்த நுண்ணுயிரி காத்திலிருந்து சொந்த தலைச்சத்து உருவாக்கி பாதி தனக்காக வச்சு பாதி செடிக்கு கொடுக்கும். Rhizobia. So basically these the leguminous crops that you see here have nodules in their roots in which rhizobium bacteria grow and produce they fix their own nitrogen from the air, use some of it themselves and share some of it with the plant in exchange for food that the plant gives them in exchange. So by growing these two together, you can optimize fertilizer use. இதில் இயற்கை வேளாண்மை பண்ணுறாங்க, எந்த செயற்கை உரம் போடாமலே. அதுடன் இதில் சில இடங்களில் வறட்சி வந்திருக்குது வானிலை மாற்றம் காரணமாக. அப்போ மக்காச்சோளத்தோட மகசூல் ரொம்ப குறைஞ்சாலும் இந்த பருப்போட மகசூல் இன்னும் குறைகிறது. சில தடவை 100% கூட போயிடுச்சு, அதாவது அவங்க போட்ட விதை கூட திருப்பி வரல. அப்போ விவசாயிகள் என்ன பண்ணுறாங்க, அவங்க கைவிட்டுறாங்க. சரி இந்த பருப்பு வரல, வேறு மக்காச்சோளம் தான் போட போவோம் அப்படின்ட்டு. அப்போ இது என்ன விளைவு? முதல்ல உத்து சத்து, அதாவது சாப்பிடும்போ உங்களுக்கு முழுமையான புரோட்டீன் வேணும்னா மக்காச்சோளமும் சாப்பிடணும், பருப்பும் சாப்பிடணும். ஒரே ஒரு சாப்பிடுங்கன்னா முழுமையான புரோட்டீன் கிடைக்காது, வாழ முடியாது. இது 2மே தான் சாப்பிடணும். 2வது விஷயம் தலைச்சத்து இனிமேல் மக்காச்சோளத்துக்கு கிடைக்காது. அப்போ அதுக்கு செயற்கை உரம் போட ஆரம்பிக்கிறாங்க சில இடங்களில். இங்கே இன்னொரு விதை இருக்கு, அமராந்தோ. அது பழச காலத்திலிருந்து ரொம்ப உத்தசத்தான ஒரு விதை ஆகும். இந்த மக்களோட இந்த மய மக்களோட மரத்தில் அமராந்தோ அவங்க கோயில் விசேஷங்களில் ரொம்ப பெரிய முக்கியத்துவம் கொண்டுள்ள ஒரு விதை. ஐரோப்பா மக்கள் ஆட்சியை செய்ய வர்றப்போ இங்க மதம் வேண்டாம், நீங்க எங்க மதத்துக்கு வந்துடுங்க அப்படி சொல்லிச்சுட்டு அமராந்தோ உற்பத்தி செய்யக்கூடாது அந்த மாதிரி உத்தரவு பண்ணாங்க. இப்போதான் 500 வருஷங்கள் கழிச்சு இல்லை இது முக்கியமான ஒரு செடி, இதுவும் நம்ம கலாச்சாரத்துக்கு முக்கியம், உச்சசத்தான உணவுக்கு முக்கியம் அப்படி புரிஞ்சுக்கிட்டு மறுபடியும் வேளாண்மைக்கு கொண்டு வர பார்த்துட்டே இருக்கிறாங்க. பக்கத்துல இந்த கிராமத்துக்கு பக்கத்துல 3 மணி நேரம் தூரம் இன்னொரு கிராமம் இருக்கு, சோலோஹ்யா. அங்கே அவங்க அதிகமான பாரம்பரிய உற்பத்தி செய்யறதுல நீங்க இங்கே பாக்குறீங்கன்னா எல்லாம் இந்த வெங்காயம் எல்லாம் வால்மார்ட் பெரிய பெரிய கடைகளுக்கு போகுது தலைநகரத்துல. அங்கே வெங்காயம் இருக்கு, கேரட் இருக்கு, முட்டைக்கோஸ் இருக்கு, வெண்ணெய்ப்பழம் கூட இருக்கு. அதுல நிறைய 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 பூச்சிக்கொல்லி அடிக்கிறாங்க. ஏனென்றால் இந்த விதை அந்த விதைக்கு எதிர்ப்பு சக்தி இல்லை. இந்த விதை இந்த கிளப்பின ஹைபிரிட் என்ற விதைக்கு ஜாஸ்தி எதிர்ப்பு சக்தி இல்லை. பூச்சி வந்தா அதற்கு பூச்சிக்கோழி எடுக்க வேண்டிய சூழ்நிலை ஆகும். பூச்சிக்கோழி செடிக்கு மட்டுமே தெளிக்காது, அது தானாகவே மண்ணுக்குள்ளே வரும், மண்ணும் பாதிக்கும். மண்ணோட கரிம சக்தி, அதாவது இந்த சோலோகானிக் கார்பன் அதுவும் குறைஞ்சிரும். அதுக்குள்ளே இங்கே ஒரு தட்டு போட்டு இருக்கும், என்னென்ன பூச்சி அதுக்குள்ளே வளர்ந்துரும் பார்க்குறதுக்கு. அதில் ஜாஸ்தி பூச்சி இல்லை. நீ இப்போ பார்க்குறீங்கன்னா முக்கால்வாசி தீமை செய்யும் பூச்சி, சைவ பூச்சி, பைய தின்னும் பூச்சி இருக்கும். நன்மை செய்யும் பூச்சி, அசைவ பூச்சிகள் ஜாஸ்தி வரல. அதே மாதிரி மண்ணுக்கு கீழே பாதிச்சிருக்கும் என்று நினைக்கிறோம். பார்க்குறதுக்கு நமக்கு தெரியல, செய்முறை தெரியல, அந்த அரிசி நாங்கள் இனிமேல் தான் செய்யணும். ஆனால் அங்கே நிறைய செடிகளுக்கு நோய் வந்திருக்குது. நோய் வர்றதால செடிகளுக்கு ஆன்டிபயோட்டிக் கொடுக்க ஆரம்பிச்சிருக்காங்க மண்ணில் விட்டுட்டு. ஏன்றா நன்மை செய்யும் நுண்ணுயிரியல்களும் இருக்கும். அது மண்ணுக்குள்ளே இருக்கிறதுன்னா தீமை செய்யும் நுண்ணுயிர்கள் வராது. காலையில் குழந்தைகளுக்கு அப்படி சொன்னாங்க, பேத்துறனே நன்மை செய்யும் நுண்ணுயிர்கள் வருதுன்னா நோய் உண்டாக்கும் நுண்ணுயிர்கள் வராது. அதே மாதிரி செடிகளும். இது விவசாயத்துக்கு மட்டும் முக்கியம் இல்லை, நம்ம ஆரோக்கியத்துக்கு மிக்க முக்கியமானது. இந்த ஏரி பார்க்குறீங்கன்னா நீங்கள் பார்த்த வெயிலுக்கு நேரடி கீழே இருக்கு. அப்போ நீங்க என்ன தெளிச்சாலும் அது கண்டிப்பாக வயலிலிருந்து இந்த ஏரி வரை போகும். இந்த ஏரியில் மீன் இருக்குது, நண்டு இருக்குது. மீன் வாரி எல்லோரும் அங்கே மீன் நண்டு எல்லாம் பிடிச்சு அது பக்கத்தில் இருக்கும் கிராமங்களே விற்பாங்க. அதனால் என்ன விஷம் இந்த ஏரிக்குள்ளே போடுறோம்னா அது கண்டிப்பாக சாப்பிட்டுட்டு இருப்போம். அது மட்டும் இல்லை, எரியோட ஆழம் 2 கிலோமீட்டர், அவ்வளோ ஆழமான ஏரி அது. இந்த மலைகள் பார்க்குறீங்கன்னா அப்படியே அங்கேயே நிற்கல, நேராக இந்த ஏரிக்கு கீழே அதே மலை தொடர்ந்து போகிறது. அதனால நீங்கள் ஏதோ விஷம் இந்த ஏரியில் போடுறீங்கன்னா கீழே போகிறதுக்கு 100 வருஷம் தேவை, திருப்பி மேலே வர்றதுக்கு இன்னொரு 100 வருஷம் தேவை. அதாவது அதில் என்ன சுத்தம் இருந்தாலும் என்ன மாசுபாடு இருந்தாலும் நாம் அதை 200 வருஷமாக தொடர்ந்து குடிச்சிட்டே இருப்போம். இங்கே பார்க்குறீங்கன்னா இங்கே இதெல்லாம் பூச்சிக்கொல்லி கூடவை. அது இந்த கூடையில் போடுறது, கூடைக்குள்ளே போடாடுறது இன்னும் எவ்வளோ இருக்குன்னு தெரியல. அப்போ இதெல்லாம் பார்த்து நம்ம இயற்கை வேளாண்மைக்கு வரலாம் என்ற நோக்கத்தில் வேலை செய்யலாம். ஆனால் இயற்கை வேளாண்மைக்கு 2 தத்துவம் இருக்கிறது. So if you look at organic agriculture, there's really sort of 2 schools of thought. One of them tries to replace what's been created with what's being done in inorganic agriculture, so replacing toxic pesticides with organic pesticides. And one of the other philosophy instead tries not to use these external inputs and instead rely on natural ecosystems and natural soil health to protect the crops. அதாவது இந்த அங்கக வேளாண்மைக்கும் இயற்கை வேளாண்மைக்கும் முக்கியமான ஒரு வித்தியாசம். நீங்கள் சிக்கிம் மாநிலம் பற்றி கேள்விப்பட்டிருக்கீங்களா? இயற்கை வேளாண்மை பண்ணுறாங்க. அங்கே இயற்கை வேளாண்மை பண்ணுறாங்க, ஆனால் அவங்கள உணவு எனக்கு தெரியும் வரை முக்கால்வாசி செயற்கை தான் சாப்பிட்றாங்க, வேற மாநிலத்திலிருந்து எடுத்து வர்றாங்க. அது ஒரு வழி. இன்னொரு வழி வந்து நம்ம உணவு இயற்கையாக பண்ணுவோம், சந்திரனில் இல்லாமல், ஆனால் நம்மளே அது சாப்பிடுவோம், அதுக்கப்புறம் மிச்சம் இருந்தால் அது விற்பனை பண்ணுறோம் என்று இன்னொரு தத்துவம். இந்த இயற்கை வேளாண்மையில் கொஞ்சம் இந்த அங்கங்க வேளாண்மை சொல்லலாம். இந்த நோக்கத்தில் என்ன பண்ணுறாங்க வந்து இருக்கும் பூச்சிக்கொல்லி எடுத்து அதுக்கு மாறாக ஒரு இயற்கை பூச்சிக்கொல்லி போடலாம். அதாவது இங்கே வந்து போவேரியா பசியானா அது ஒரு நுண்ணுயிரி, அது வந்து பூச்சிகளை கொன்றும், பூச்சியோட நோய். அது உருவாக்கி பெரிய லெவலில் உருவாக்கி ஒரு குடவையில் போட்டு விவசாயிகளுக்கு வித்துட்டு அது தெளிப்பாங்க, பூச்சி சேர்த்துரும். இங்கே வந்து நீம், அது வந்து வேப்பிலை ஒரு சாறு, அதுவும் பூச்சிகளை கொன்றும். இந்த மாதிரி பொறிகளும் போடலாம். ஆனால் இதையெல்லாம் என்ன பார்க்குறீங்கன்னா அதையெல்லாம் விவசாயியால் வாங்க வேண்டிய பொருள். அதுதான் பல்வகைத்தன்மை இருக்காது. இதில் ஒரே ஒரு வகையான நுண்ணுயிரி இருக்கும். ஒரு நிலையான வேளாண்மைக்காக நமக்கு பல விதமான நுண்ணுயிரிகள் வேண்டும், பல விதமான பயிர்கள் வேண்டும். இதெல்லாம் கலந்து தானே தானாகவே நிலைத்தன்மை கிடைக்கும் மகசூலுக்கும் வயலுக்கும். அந்த வழியில் பார்க்குறோம்னா விவசாயிகளால் கண்டுபிடித்த பூச்சி விரட்டிகள், நுண்ணுயிரி ஊக்கிகள் ரொம்ப முக்கியமானது என்று நாங்கள் கண்டுபிடிச்சிருக்கோம் எங்களோட ஆராய்ச்சியில். இங்கே இது வந்து இஷிமுல நாடு. இங்கே எனக்கு ஒரு நண்பர் எப்படி இது தயாரிக்கிறது என்று சொல்லி கொடுக்குறாங்க. சில்தெப்போல் அதன் பேர் வந்து சிலே என்று அர்த்தம் வந்த மிளகாய், சிவப்பு மிளகாய். அப்போது அது போட்டு அதோடன் பெனித்தோ இது சொல்லி கொடுத்தாங்க. அதில் 7 வகையான பூச்சி விரட்டிகள் போட்டுட்டு அதற்கு மேலே சரி இங்கே பஞ்சகாவியில் 5 விஷயங்கள், 5 உட்பொருள்கள் மாட்டிலிருந்து வருது. அதில் மாடு சாணை, மாடு சிறுநீர் அதையெல்லாம் வருது. அங்கே மாடு கிடையாது. அந்த நாட்டில் மேற்கு பகுதி இருக்குது, கிழக்கு பகுதி நாங்கள் இருக்கிற இந்த இடத்துல மலையில் மாடு கிடையாது. அப்போ அதற்கு மாறாக என்ன போடுறாங்க? மனிதர் சிறுநீர். அங்கே 1 லிட்டருக்கு ஏதோ ஒரு 50 ரூபாய் கொடுப்பாங்க. ஏன்னா நிறைய நிலம் இருக்குது, என்ன குடும்ப சின்னதாக இருக்குது, என்ன பட்டாடு இல்லை. அப்போ சில விவசாயிகள் அதிகமாக உற்பத்தி செஞ்சு சில கம்மி பண்ணி அப்படி ஒருத்தருக்கு அப்படி விற்கிறாங்க வயலில் போடுறதுக்கு. அதையெல்லாம் கலந்து ஏதோ ஒரு 2 மாதம் அப்படி உருவாச்சு அந்த நுண்ணுயிரிகள் தானாகவே இன்னும் பெருக்கம் செஞ்சு அதுக்கப்புறம் செடிக்கு மேலே தெளிப்பாங்க. நான் இதையெல்லாம் தெளிக்கிறதுக்கப்புறம் தானே உள்ளே என்ன இருக்குன்னு எனக்கு தெளிவாக சொன்னாங்க. கொஞ்சம் ஏமாந்துட்டேன். ஆனால் நிஜமாக நல்லா வேலை செய்கிறது. அது மட்டும் இல்லை, இதன் சக்தி நேரப்படியாக அதிகிறது. ஏன்னா அதில் நுண்ணுயிர்கள் இருக்கு. அப்போ அப்படி சொல்கிறாங்க ஒரு மாதம் கழிச்சு நீங்கள் பயன்படுத்துகிறீங்கன்னா அப்போ ஒரு ஏக்கருக்கு அவ்வளோ போடணும். ஆனால் 6 மாதம் கழிச்சு பயன்படுத்துறீங்கன்னா அப்போ ஏக்கருக்கு அதுக்கு பாதி போடலாம். ஏன்றால் அதன் ஆற்றல் அதிகரிக்கிறது தானாகவே. அப்போ அங்கே இந்த மாதிரி வேளாண்மை செய்யும் வயல்கள் தெரியுது. நீங்க பார்த்த மாதிரி இந்த எல்லா செடிகள் கலந்து போட்டிருக்காங்க. அதுக்கு 2 நன்மை. ஒன்று இந்த உற்பத்தி அதிகமாக இருக்கும், மகசூல் அதிகமாக இருக்கும். ஒவ்வொரு செடிக்கு வேறு நுண்ணுயிர் வரும். வேருக்கு ஒவ்வொரு செடிக்கு வேறு மாதிரி ஒரு சத்துகள் தேவைப்படும். இந்த மாதிரி கலந்து போடுறோம்னா அப்போ ஒவ்வொரு செடி ஒருத்தர் தான் சொந்த இடம் கண்டுபிடிச்சு நல்லா வளரும். இன்னொரு விஷயம் இந்த செடியை தின்னும் ஒரு பூச்சி வருதுன்னா அப்போ பூச்சிகள் கொஞ்சம் சின்ன குழந்தை மாதிரி ஒரே உணவு மட்டுமே பிடிச்சிருக்கு, வேற உணவு கொடுத்தா சாப்பிடாது. அப்போ மக்காச்சோளம் சாப்பிடும் ஒரு பூச்சி வருதுன்னா மக்காச்சோளம் சாப்பிடும். ஆனால் அடுத்த என்ன வருது, அது மக்காச்சோளம் இல்லை, சாப்பிட முடியாது. அடுத்த மக்காச்சோள செடி தேடித்து காலமாகும். இந்த பூச்சிக்கு அலைச்சல் வரும். அப்போ சீக்கிரமா இனப்பெருக்கம் செய்ய முடியாது. இந்த பூச்சியோட எண்ணிக்கையும் கட்டுப்பாட்டில் இருக்கும். இங்கே இது ஒரே கிராமத்தில் கண்டுபிடித்த வேறு வகையான மக்காச்சோளம் தெரியும். வேறு மாதிரி சாப்பிடுவாங்க. சிலர் சப்பாத்தி செய்கிற மாதிரி பயன்படுத்துகிறாங்க, சிலர் கஞ்சி பண்ணுறாங்க. ஒவ்வொருக்கும் சொந்த பயன், சொந்த உபயோகம், பேர் எனக்கு தெரியல. அதே மாதிரி விதை நூலகம் உருவாக்குறாங்க. அதாவது இந்த விதையை எல்லாம் கண்டுபிடிச்சு ஒரு 90 நேரத்தில் எல்லாம் வச்சுட்டு விவசாயிகளுக்கு தேவைப்பட்டால் அங்கே இங்கே இந்த மாதிரி விதை வேணும்னு சொன்னால் அவங்களுக்கு தருவாங்க. இது காளான்கள் அங்கே இது வளர்த்ததுல காட்டில் கண்டுபிடிச்சது. இப்போ இது தலைக்கீழ வந்துருச்சு. சரி, பரவாயில்ல. இது இன்னொரு கிராமம், இயற்கை வேளாண்மை செய்யும் கிராமம். So this is another organic field. You can see all the different types of trees again, lots of biodiversity in the field. அதில் நாங்கள் கண்டுபிடிச்ச பூச்சிகள் பாருங்க. அதுக்கு முன்னால் செயற்கை வயல் எவ்வளோ வித்தியாசம் இருக்குன்னு பாருங்க. அதே மாதிரி மண்ணுக்கு கீழே கண்ணுக்கு தெரியாது, ஆனால் மண்ணுக்கு கீழே அதே மாதிரி வித்தியாசம் இருக்கும் நுண்ணுயிரியல்களில். இது குழம்பி என்று ஒரு உற்பத்தி, கொட்டை வடிநீர் உற்பத்தி பண்ணை. ஆனால் திருப்பி இந்த IoT ட்ரோன்ஸ் இந்த விஷயங்களுக்கு வரோம்னா இப்போ நாம பார்த்த அறிவுகள் பல்வகைத்தன்மைக்கு ஜாஸ்தி இடமே இல்லை. அவங்க விவசாயிக்கு இடம் கொடுக்காமல் நாங்களே ஏதோ ஒரு அலுவலகத்தில் முடிவுகள் எடுத்து விவசாயி எப்படி செய்யணும் என்று நாங்கள் சொல்லிக் கொடுப்போம். எப்படி சொல்லிக் கொடுப்போம்? ஏதோ ஒரு செயலி மூலம் அவங்களுக்கு குறுஞ்செய்தி அனுப்புவோம். So if compare this to the vision, or the reductivist vision I find of IoT, drones, or precision agriculture in general, of the general discourse, instead of relying on farmers' knowledge, we really tend to see a discourse where scientists or programmers propose to make optimal decisions for farmers and send them those decisions to implement through a mobile app or through SMS. கடைசியிலே விவசாயி எங்க? அது மட்டும் இல்லை, இந்த தகவல் விவசாயிகளோட தகவல்கள் டேட்டா அதெல்லாம் எங்கே போகுது? மேகத்தில் இருக்குன்னு சொல்கிறோம், இட்ஸ் இன் த கிளவுட் அப்படி சொல்கிறோம். ஆனால் மேகம் எங்கே? மேகம் எதுவுமே இல்லை. மேகம் இதுதான், டேட்டா சென்டர்ஸ், அதாவது தரவு கட்டிடங்கள். அது எங்கே இருக்கும்? அது இன்னொரு கேள்வி. என்ன நிறத்தோட கையில் இருக்கும்? நம்ம நாட்டு நிறுவனமா, வெளிநாடு நிறுவனம், எதிரி நாடு நிறுவனமா? அமேசான், கூகுள். அப்போ இது இந்த பிரச்சனை. நாங்கள் IoT இருக்குள்ள அதுக்கு மேலே அதிகமான நம்பிக்கை பண்ணுறோம்னா எல்லா தகவல்கள் நான் உங்களுக்கு வாட்ஸ்அப் மூலமாக ஏதோ அனுப்புகிறேன்னா என்னால் உங்களுக்கு அனுப்ப முடியாது. நான் வாட்ஸ்அப்புக்கு அனுப்புகிறேன் பெங்களூரில். அப்போ பெங்களூர்லேருந்து திருப்பி உங்களுக்கு இங்கே வருது. நம்ம 2 பேர் ஒரே அறையில் இருந்தாலும் இந்த தகவல்கள் ஒருத்தால இன்னொரு ஆள் வரை அனுப்ப முடியாது, இந்த நிறத்தோட கட்டுப்பாட்டுக்கு, இந்த நிறத்தோட ஆதரவு இல்லாமல். அப்போது சரி சும்மா புகைப்படங்கள் அனுப்பணும்னா செடி அனுப்பணும்னா பிரச்சனை இல்லை. ஆனால் நாட்டுக்கு முக்கியமான ஒரு விஷயம், அதாவது விவசாயிகளோட அறிவுகள், அந்த மாதிரி மிக்க முக்கியமான ஒரு விஷயம் இருந்தால் இந்த மாதிரி இந்த அளவுக்கு ஒரு தனி வெளிநாடு நிறுவனத்துக்கு மேலே ஆதரவு பண்றோம் என்றால் அது நாட்டோட இறையாண்மைக்காக ஒரு ஒரு ஆபத்து என்று நம்புகிறேன். அப்போ இங்க ஆராய்ச்சியில என்ன பண்ணிருக்கோம் கடைசியில இந்த மாதிரி சர்வர் இல்லாமல், அதாவது இந்த கூகுள், அமேசான் இந்த நிறுவனங்கள் இல்லாமல் ஏதோ ஒரு தகவல் அமைப்பு உருவாக்கலாமா என்று பார்த்தோம். அதன் பேர் சம உரிமை பகிர்வு பிணையம். சம உரிமை எல்லோரும் சமம், எந்த சர்வர் கிளையண்ட் ஒன்றும் கிடையாது, எல்லோரும் சமம், என்னோட கைபேசி உங்க கணினி எல்லாம் ஒரே மாதிரி தான், நேராக ஒருத்தருக்கு பேசுறதுக்கு. பகிர்வு என்ன தகவல் பகிர போவோம். பிணையம் ஒரு இணையம், ஒரு வலை. ஆங்கிலத்தில் இதுக்கு பியர் டு பியர் சொல்றாங்க. So in English it's called the peer-to-peer networks. It's a network where you can share data without depending on a server, which usually belongs to a large company like Google, Amazon, Facebook and so forth. So this allows you to share data directly between users without any dependence on a foreign company. இன்று இணையம் எல்லாம் இந்த மாதிரி கூகுள் பயன்படுறீங்கன்னா முகநூல், ஃபேஸ்புக், வாட்ஸ்அப் என்ன இருந்தாலும் இதே மாதிரி இவங்களுக்கு பேசணும்னா பண்ண முடியாது. திருப்பி இங்கே பண்ணி திருப்பி இங்கே வரணும். இங்கே ஏதோ ஆகுறதுன்னா இணையம் பிரச்சனை இருக்கிறதுனா அப்போ இதெல்லாம் வேலை செய்யாமல் போயிடும். நாங்கள் அதிலிருந்து ஒரு முழுமையான பகிரப்பட்ட அமைப்புக்கு மாற பார்க்குறோம். அதாவது விவசாயிகள் நேராக ஒருத்தருக்கு தகவல்கள் அனுப்புற மாதிரி ஒரு அமைப்பு அமைக்க பார்க்குறோம். அதை எப்படி பண்ணுறோம் பிடிஎஃப்ல இருக்கிற இது கொஞ்சம் தெரியல, ஆனால் இந்த சர்வர் நாங்கள் கழுத்துட்டு நேராக விவசாயியோட கைபேசியிலிருந்து கைபேசி வரை தகவல் அனுப்புற மாதிரி ஒரு மென்பொருள் உருவாக்கி கொண்டிருக்கிறோம். இதன் பேர் விண்மீன், எல்லாம் கட்டற்ற மென்பொருள். So this is our open source software. It's called Vinmean or Constellation in English, Constellation in French, the language in which it's coded. And you can access it all online if anybody is interested, if anybody is into coding. இதெல்லாம் இணையதளத்தில் இலவசமாக எல்லோருக்கும் கிடைக்கும், இல்லாமல் நீங்கள் பார்க்குறீங்கன்னா அதில் மக்களால், விவசாயிகளால், உங்களால், ஆட்சியாளர்களால் சொந்த புகைப்படம் பேரையெல்லாம் உள்ளிட்டு அதுக்கப்புறம் உங்கள் தகவல்கள் அதில் உள்ளிடலாம். தகவலில் படங்கள், புகைப்படங்கள், காணொளி, ஒலி, தகவல்கள், தேதி, எண் என்ன வேணாலும் அதுக்குள்ளே போடலாம். அதே மாதிரி அதற்கு மேலே வெவ்வேறு செயலிகள் உருவாக்க முடியும். That means you can use our software, our Constellation software to build your own apps for your own projects if you like as well. இங்கே உதாரணமாக கம்போடியா நாட்டில் அதுக்கு தான் கம்போடியா மொழியில் தெரியுது. இதே விண்மீனுக்கு மேலே அங்கே நிலத்தடி நீரோட உப்பு தன்மை தெரிகிறதுக்கு ஒரு செயலி உருவாக்கி கொண்டிருக்கிறார்கள். அப்போ நான் முடிவுகளுக்கு வர்றேன்னா ஏதோ ஒரு 3 விஷயங்கள் நீங்கள் இன்னைக்கு மறக்கக்கூடாதுன்னு சொல்கிறேன்னா, முதல்ல அறிவாளியான இல்லைன்னா துல்லியமான வேளாண்மை, அதுக்கு அடிப்படை கண்டிப்பாக விவசாயிகளோட அறிவு தான் ஆகணும். ஏதோ ஒரு அலுவலகத்தில் ஏதோ ஒரு ஆராய்ச்சி நேரத்தில் உட்காந்து ஒரு ஆளால் விவசாயிகள் விட நல்லா வேலை செய்ய முடியாது. ஆனால் அவங்களோட தேவையான ஆதரவுகள் கண்டிப்பாக செய்ய முடியும். நீங்கள் உங்களோட ஆராய்ச்சியில் ஒரு வேளாண்மைக்காக, விவசாயிகளுக்காக அதிகமான உற்பத்தி மகசூலுக்காக தொழில்நுட்பங்கள் கண்டுபிடிக்க போவீங்க. ஆனால் இந்த விஷயம் மட்டுமே மறக்காதீங்க, முதல்ல விவசாயிகளுக்கு போயிட்டு அவங்களுக்கு என்ன தேவை, அது அவங்களோட கையில் இருக்குமா, அவங்களால அதை பயன்படுத்த முடியுமா, தானாகவே நீண்ட காலமாக அவங்களால அதை பயன்படுத்த முடியுமா, இல்லை என்றால் உங்களுக்கு மேலே, இல்லை என்றால் உங்கள் கண்டுபிடிப்பை கைப்பற்றும் நிறுவனத்துக்கு மேலே ஆதரவு ஆகுமா, அந்த விஷயம் தலையிலுக்குள்ளே வைக்கணும். ரெண்டாவது என்ன கண்டுபிடிச்சாலும் அது விவசாயிகளோட அறிவு செயல்முறைகள் தெளிச்சு போகக்கூடாது. அதுக்கு மாறாக விவசாயிகளோட ஆதரவு கொடுக்கணும், அவங்களோட அறிவுகளுக்கு ஆதரவு கொடுக்கணும். ஆனால் அந்த அறிவுகளை அழிக்க பார்க்கக்கூடாது. கடைசியில் வந்து அறிவாளி வேளாண்மை வந்து விவசாயிகளோட அறிவு இல்லாமல் சாத்தியமில்லை. என்ன பண்ணாலும் அது நம்மோட அடிப்படை இருக்க வேண்டியது. So I'll just repeat for non-Tamil speakers who might be online. There's 3 main takeaways I'd like to leave with you from this talk. The first is that you can't have smart agriculture, intelligent agriculture, without using farmers' intelligence as well. If you're going to develop a technology that aims to help farmers, that advances precision farming or smart agriculture, it should aim to augment or to support farmers' knowledge and not try to displace it or to take decision making out of their hands. Because in the end, you can't have a smart agriculture without farmers' intelligence to back it up. அவ்வளவுதான், உங்களுக்கு நன்றி சொல்றேன். இங்கே என்னோட தொடர்பு இருக்கு, நீங்க வேணுமென்றால் தொடர்பு கொள்ளுங்கள்.
Thank you, Dr. Adam. Your talk has truly enriched our understanding, and we are grateful for your valuable insights on leveraging traditional knowledge for sustainable agriculture. Thank you.
அடுத்த வீரமா முனிவர் பிறப்பெடுத்து தமிழகத்துக்கு வந்துள்ளார் என்று எண்ணுகிறேன். அவர் தமிழுக்காக தன் வாழ்வை செலவிட்டார். ஆனால் தாங்கள் தமிழ் மற்றும் விவசாயத்தையும் சேர்த்து வளர்க்கிறீர்கள். உங்கள் விளக்கக்காட்சியினை நாங்கள் கேட்க கிடைத்தது எங்களின் மகிழ்ச்சியையும் பெருமையும் ஆகும். நீங்கள் எங்கள் மாநாட்டிற்கு வந்தமைக்கு எங்கள் மனமார்ந்த நன்றிகள்.
Each plenary session has enriched our understanding, broadened our perspective, and motivated us to explore new dimensions in research, innovation, and application. We sincerely thank all the speakers for generously sharing their valuable knowledge, experience, and time with us. Your contribution have truly added great value to this conference. Let us give a big round of applause for all the respected plenary speakers. And now it's time for online and oral poster presentations, along with offline poster presentations will be conducted as per scheduled. We also invite participants to showcase their work through online and offline poster presentations. So participants of poster presentation, make your way to the designated areas, and there will be a Google link will be posted in the group. So kindly you can upload in that, and further information will be uploaded in the group over there. So kindly join the given respective groups. So I would call upon Princey to take over the panel's discussion.
இது இன்னொன்று வேணும். வாட்டர் பாட்டில் 2 மாதம் இங்கே ஒரு சேர்
Thank you, Dr. Kalyani. Today's panel session is from Shazia, panelist. முடிச்சுட்டு இப்போ கூப்பிடலாமா? இது முடிச்சதும் இங்கே தான் இருக்கா ரெண்டும்.
I request the participant to occupy the front seat. I repeat, I request the participant to occupy the front seat. First UG students are requested to occupy the front seat. I repeat, first UG students are requested to occupy the front seat. Good evening, participant and esteemed guests. Welcome to ICAB SIVB 2026. I am Princey Julia and I am honored to be your anchor for this exciting panel discussion on translation, biotechnology, converting academic research into industrial venture. Coming together is the beginning, keeping together is progress, and working together is success. With that spirit, we are gathered here to explore the dynamic aspect of translation biotechnology. Today's discussion aims to bring to light varied aspects associated with translation biotechnology, sparkling thoughtful discussion and exploring the journey of idea to impact, the challenges faced by researchers, funding opportunity, industry collaboration, intellectual property and commercialization strategy. To navigate this topic, we have an exceptional panel of experts with us. Our first distinguished panelist is Dr. D.J. Mukesh Kumar, Associate Professor, Research, Department of Biochemistry, Savitha Medical College and Hospital, Chennai. With over 20 years of academic and research experience, he has published 190 plus research papers and holds 13 product patents and secured multiple funded research grants. His expertise includes nanotechnology, biomaterials, bioenzyme, sustainable biotechnology, and product development. He leads the Green and Eco Lab, focusing on eco-friendly biosynthesis and sustainable innovation, and actively mentors startups through consultancy in patent and funding support. We are delighted to welcome Dr. D.J. Mukesh Kumar to our panel. Our next distinguished panel is Dr. Janani Dakshinamurthy, a leading clinical scientist and entrepreneur and the founder of Gene Aura Private Limited. She is an IIT Madras alumni, certified genetic counselor and internationally trained in aging from United Kingdom. Dr. Janani actively practices across major hospitals and made an impactful contribution in women's reproductive health, cancer genetic and PCOS research. She is a DST recognized women scientist and founder of the Women Endocrine Health Foundation and PCOS 360 community promoting healthcare innovation and awareness. We are pleased to welcome Dr. Janani Dakshinamurthy to our panel. We now welcome the next panelist, Mr. Anuj Raj, founder and director of the Travancore Institute of Biosciences Research, Kerala. He is an accomplished biotechnology researcher and entrepreneur with strong expertise in nanomedicine and carbon-based nanomaterials with multi-peer-reviewed publication to his credit. His works beautifully bridge science, healthcare and innovation. We are honored to have you with us today, sir. We are delighted to welcome Dr. Naveen Kumar Venkatesan, a scientist turned entrepreneur and the founder and the CEO of Humogenics Bioscience, Chennai. He is a renowned expert in molecular microbiology, bioinformatics, immunology, and antimicrobial resistant research. Dr. Naveen has pioneered the development of rapid point-of-care diagnostic device for infectious disease and AMR detection with patent currently under process. His excellence has been recognized through prestigious honors such as AstraZeneca Grand Challenge Award and the Louis Pasteur Award. In addition, he serves as the Vice President of Tamil Nadu Association of Biosafety and Biosecurity and actively contributing as the academic board member and adjunct faculty at a reputed institution. Please join me in giving a warm welcome to Dr. Naveen Kumar Venkatesan for joining the panel. Our next panelist, Ms. Pavitra K., founder and managing director of FSG Foodworks, a seasoned food safety expert and entrepreneur whose work has supported hundreds of food business across India and earned national recognition for excellence and innovation. She has been recognized nationally with Food Safety Gene being selected among India's top 50 women startups by IIM Bangalore and Goldman Sachs. She is also author of Certified Safe, a practical guide on food safety and hygiene for small food business, and actively contribute to industry knowledge sharing initiative focused on building safer, more sustainable food system. We welcome you, ma'am, for joining the panel. We are pleased to welcome Mr. Dinesh Bhupalan, next panelist, a dedicated researcher and biotechnologist currently working at Senji International Bengaluru. He brings strong hand-on expertise in cell line engineering, CRISPR gene editing, antibody development, protein analysis and bioprocess technology. He has actively contributed to biopharmaceutical researcher, vaccine production initiative and the development of advanced laboratory platform. A published author in the field of stem cell regenerative research, Mr. Dinesh combines academic depth with strong industry-oriented translation experience. And it is a privilege to introduce Dr. M. Nitya Kalyani, a distinguished scientist, academic leader and entrepreneur. She is the chief scientist at Rajiv Gandhi Government Central Hospital and Madras Medical College, Chennai. She was associated with Apollo Group of Hospital and the founder and director of Cell to Cure Therapies and IGN Biotech Private Limited, Chennai. She has received several national and international grants, hold multi-patent and has led landmark initiative including the Human Pancreatic Islet Transplantation Program in India. With extensive research collaboration and numerous publications, her contribution to medical biotechnology and innovation are truly remarkable. Her unique ability to integrate scientific rigor with creativity and leadership has earned numerous awards for innovation, education and women's leadership. We are honored to have Dr. Nitya Kalyani as the moderator for this panel discussion. We have an action-packed 60-minute session. We will start with some prepared focus question followed by the deeper dive into the challenge.
We want to have a very focused approach. It's not that we want to put everything together and then we confused, even our teammate would be confused because they don't know what to focus at one time. So it is segregated.
So the same question I would like you to answer in your own way. How do the children find out that whatever I'm doing is going to be useful and it is having the market need? Because market need is our Sona Madri, we can have 100 products, at last not even a single thing which you are not able to sell, it won't, and of course the food products in a second, nowadays there's a lot of need, but for a product like yours, I know where she is working on the genetics and all, precision medicine, it is not necessary at all when there is so many things existing. So how will you stand alone, totally different and prove to the world that yes, it's a need? Or how will are you creating a need for it?
So, நம்ம இது வந்து two category of students எடுப்போம். One category of students ரொம்ப தெளிவா அப்பவே ஸ்டார்ட் பண்ணும்போதே they know this is what they want to do. This is a very very niche. இப்போ current scenarioல maybe நம்மளோட Gen Z அடுத்த வர generation இப்பவே தெளிவா இருக்காங்க. All the 5th standard 6th standard students a little more இன்னும் அப்பவே தெளிவாயிடுறாங்க. I want to do this, I want to do that அப்படின்ட்டு. பட் நம்ம இப்போ கரண்ட் நம்ம பசங்க கிட்ட வருவோம். இங்கே you're already halfway through and இதை நீங்கள் வந்து you've chosen this not with a plan of an entrepreneurship or market analysisலாம் பண்ணி இந்த ஃபீல்டுக்கு வந்து மேபி எஸ் இட்ஸ் அ க்ரோயிங் ஃபீல்ட், அது நீங்கள் அனாலிசிஸ் பண்ணியிருப்பீங்க. இட்ஸ் வெரி 12th வரும்போது பேரண்ட்ஸ் அண்ட் ஸ்டூடெண்ட்ஸ் எவ்ரிபடி தே ரிசர்ச் எந்த ஃபீல்டு ரொம்ப க்ரோ ஆகும், எந்த ஃபீல்டுக்கு நிறைய ஆப்பர்ச்சுனிட்டிஸ் இருக்கும் அப்படின்ற அந்த ரிசர்ச் பண்ணிட்டு வருவீங்க. பட் யூ டோன்ட் கம் வித் அன் ஐடியா ஆஃப் அ ப்ராடக்ட் ஆர் சம்திங் அண்ட் இங்கே ஆன் யூஜி லெவல் யூ லேர்ன் எவ்ரிதிங், எல்லாமே கற்றுப்பீங்க. இட்ஸ் அ மிக்சட் subjects over there and PG போறப்போ கொஞ்சம் ஸ்பெசிஃபிக்காக இருக்கும். பட் you have to start thinking at least when you're in PG, not whatever you learn. So whatever you learn from the college வந்து it's a skill தான். அங்கேயே வந்து ப்ராடக்ட் சொல்லி கொடுக்கறது இல்லை, they teach you skills and அந்த skills-ஐ வந்து how you can repurpose it for the society is what you will have to start thinking at least during your final year's project. அந்த project time-ல வந்து you should start focusing there because இப்போ நீங்கள் ஒரு கம்பெனிக்கு internship போகணும் அப்படின்னு நினைக்கிறீங்க to learn about the product and the market-னா as a student the company will openly welcome you. The minute you finish your project, graduate ஆயிட்டீங்கன்னா எல்லா கம்பெனியும் ஏன் நமக்கு வரான், why does he want to come here அப்படின்ற question comes in. They don't want to take you in it is not very easy to get an internship or or trying and testing out opportunity after you finish your college. It is very, very difficult at that time. And the PG mudikra varaikum da the liberty. Any company you can walk in, tell you are a student, you want to learn what they are doing and you want to explore ideas and options na, and the PG varaikum da mudiyum. Even a PhD scholar, you will not have time for that. Even if you have time, they keep asking, because you're already starting a research, they will think who's your professor yaru, guide yaru, department enna, avanga background enna, products பண்றாங்களா, இங்க இந்த ஐடியாஸ் எடுத்துப்பாங்களா, lot of things, even a company has to protect their அவங்களோட ப்ராடக்ட்ஸ் ஐடியாஸ் எல்லாமே ப்ரொடெக்ட் பண்ணணும். ஒரு உள்ள வரும்போது ஒரு இதுக்கு பிரேக் இருக்கும் அதுக்கு. ஸோ PG is the best time when you can actually learn from the world outside. ஒரு மார்க்கெட் அனாலிசிஸ் அங்கே பண்ண முடியாது, பட் யூ கேன் எக்ஸ்ப்ளோர் எங்கே வேர் தேர்ஸ் அ லாக்குனே. ஸோ இஃப் நான் இப்போ the field or whatever I'm doing is called genetic counseling. எவ்வளோ பேருக்கு தெரியும்? என் கிளாஸ் அட்டென்ட் பண்ணீங்களா டிஜி வைஷ்ணவில் முன்னாடி? ஸோ அவுட் ஆஃப் த ஹோல் பாப்புலேஷன் 2 பேர் தான் கை தூக்கியிருக்காங்க. there is an Indian board, that we have a board of genetic counseling of India who qualifies students முடிச்சு practice முடிச்சிட்டு they qualify you to give genetic counseling.
So you can just advise what is it needed if they have to get that done.
So if to get qualified, so nobody knows about this niche. Ana a student from a life science background da idu panna mudiyum. We don't have a lot of medical boards who are doing this. MBBS பண்ணவங்க ரொம்ப கம்மி தான் இதை பண்ணுறது. But people from a biotech background or a life science background, they enter into this field. This awareness itself is not there. So like sir said, awareness illana, it's most probably that you've not searched enough for it. So idu vandu I did genetic counseling and I found that there is so the whole Indian population ku, any wild guess how many certified genetic counselors are there? Random number சொல்லுங்கப்பா. Whole Indian population ku, the population size, the number of dentists we have, number of doctors we have, number of nurses we have, number of physiotherapists we have, number of nutritionists we have. Any guess how many genetic counselors are there? Only 300. Only 3 for the whole Indian population, and India is a prevalent country with genetic disorders. Sir சொல்றாரு 16.2 injection. So we have trial sites in the country where we are trying to get it for free from the government and everything for the student, for the patients. It's a very ரொம்ப necessary-ஆன ஒரு field, but we were just discussing about thalassemia கொஞ்ச நேரத்துக்கு முன்னாடி. So thalassemia, it's very again common da. So இது மாதிரி எல்லாமே genetic counselors தான் can actually sit with the patient and decide what can be done for them, என்ன testing பண்ணணும் அப்படின்ட்டு. And to get into this path, you cannot randomly get in. உங்களோட education முடிச்சிட்டு you will have to work under a certified genetic counselor for a year to gather your basic course and ஒரு log book submit பண்ணாதான் entrance exam கே வந்து you are eligible. You need a clinical experience. ஏன்னா நம்ம வந்து இங்க doctors மாதிரி hospital work பண்ணல, we are studying with classrooms and boards. You don't have patient rotation is mandatory. நம்ம countryல மட்டும் இல்ல, there is an American board, there is an European board, there is a Middle East board, there is a Southeast Asian board. எல்லாத்துக்குமே for a student to get qualified, 1 year clinical rotation is mandatory for them.
Just 1 second, Dr. Janani. He wants to make a small announcement.
போஸ்டர் ப்ரெசன்டேஷன் பார்ட்டிசிபென்ட்ஸ் யாராவது இங்கே இருந்தீங்கன்னா பேக் ஸ்டேஜுக்கு போயிருங்க, அங்கே நடந்துட்டு இருக்கு போஸ்டர் ப்ரெசன்டேஷன்.
Yeah, I'll continue the same thing. So now I wanted to understand how can as he is also into the food industry, I'm very much sure that genetics and how many of you all are aware about what are the applications of genetics? 100% I think you all should know by this time. Can anyone just summarize what are the applications of genetics? இவங்க ப்ரொஃபைல் கூட படிச்சாங்க, அதுல நம்மளுக்கு பாதி பிட்டு கிடைச்சிருக்கும். Yeah, please. One, yeah, good. Another, anyone else? Very good, very good. Anyone? நிறைய இருக்கு பேர், அழகா அப்படி சொல்லிட்டே போலாம். ஆன்கோ ஜீனோமிக்ஸ், டிஸ்ஜீனோமிக்ஸ், ஃபிட்னஸ் ஜீனோமிக்ஸ், இப்போ புதுசா வந்திருக்கிற ஒரு ட்ரெண்ட். You can keep adding on to it. There are lots and lots of application. In that way, how can you connect the This is to Mr. Anuj as well as to Dr. Janaki. How can you connect the nutrigenomics with the genetics part?
First of all, my name is Anuj. I am the Managing Director of Travancore Institute for Bioscience Research. I did my PG in Biotechnology integrated from EIT University. Then I did PhD in nanomedicine. Then I have shifted to food testing and water testing. So we have a lab in Trivandrum. We are focusing on food development, nutrition profiling, shelf life, and we are doing R&D for the food companies and beverage companies. So now the research is going on in probiotic beverages, as well as ma'am told, we are helping the food companies for doing the shelf life, nutrition profiling, like that. So ma'am சொன்ன மாதிரி அவ்வளோ டீட்டெயிலாக நமக்கு சொல்ல வராது, ஏன்னா we are purely focused on wet lab. ஸோ எப்படி லேபில் பண்ணணும் அந்த மாதிரி டீட்டெயில்ஸ் தான் தெரியும். ஸோ ரெகுலேட்டரி சைட் வந்து அவங்க சொல்கிற மாதிரி that comes under a food auditor or something. இந்த கொஸ்டின் மேம் முதல்ல கேட்டிருக்கணும், இதில் எத்தனை பேருக்கு வந்து என்டர்பிரினர் ஆகிறதுக்கு மைண்ட் இருக்கு? அப்போ இவ்வளோ நேரம் பேசின சும்மாவா? கொஞ்சம் முன்னாடி வர முடியுமா?
Can you both please get up? Who wants to be an entrepreneur? Can you please get up? Oh wow. Yeah, wonderful. Yeah, nice to know at least two are. What's the reason others aren't interested? Do you think it is very tough and you cannot get through it? Can we just get open towards the audience so that if at all you have questions? Yeah, what is the problem you are facing and how can we, help with respect to that? By answering to his question, I think Dr. Janaki also will be able to address towards this genetics and food. I think that most of the things are connected towards the food. Be it the gut bacterium or whatever it is. And there is a small research which when I was with Apollo Group of Hospitals, we did it. Obesity is very, very common in our population, especially the childhood obesity is very, very common. So most of the things that are genes are responsible for it. There is different genes for responsible, be it for the increased meal portion size, ipanala binge eating pattern. Sit down, you can sit down. Oh yeah. You have to come and sit forward because the session is for you. You cannot be so hesitant if you wanted to be an entrepreneur. You should not have any shyness. First and foremost thing is that. Oh wow. So I think, yeah, great. So the first thing is, as I said, for the obesity or for anything, everything is connected to the genes. A small study which was on the binge eating as well as for the obesity, I had a lack for leptin receptor. So that's the reason I had a constant binge eating pattern. And the nutrition specifically, the cruciferous vegetables and the omega-3 fatty acids was advised to me. And once I started improving with respect to that, and the binge eating, epaparu mixture சாப்பிடுறது, கடலை சாப்பிடுறது, that pattern slowly it started reducing. Is it what we are trying to analyze out of this? Is it right, or what is your thought process on it?
உங்களுக்கு எல்லாருக்கும் ஒரு question. Two brothers, two friends, two sisters, or two just friends here. ரெண்டு பேருமே சேம் ஹேபிட். அதாச்சும் நானும் ஒரு பிரியாணி வாங்குறேன், அவரும் ஒரு பிரியாணி வாங்குறேன், நாங்கள் சாப்பிட்றோம். அவன் மட்டும் வந்து ஒல்லியாகவே இருக்கான், நான் வெயிட் போட்டுகிட்டே இருக்கேன். எவ்வளோ பேர் இது மாதிரி ஃபீல் பண்ணியிருக்கீங்க? நாங்கள் எல்லாம் ரெண்டு பேரும் சேர்ந்து குரூப் ஸ்டடி ஒன்றா தான் பண்ணுறோம். அவன் மட்டும் மார்க் வாங்குறான், நான் வாங்க மாட்டேங்கிறேன். எனிபடி? நிறைய கை இருக்கு இல்லையா? எவ்ரிபடி ஹேஸ் திஸ் எக்ஸாம்பிள்ஸ். நோ, அவன் மட்டும் சிஸ்டர்ஸ், அவன் மட்டும் அழகாக இருக்கா, நல்ல ட்ரெஸ் கிடைக்குது, நான் மட்டும் இப்படி இருக்கேன். திஸ் இஸ் அ வெரி காமன் திங் விச் ஐ சி. அண்ட் இது வந்து கொஞ்சம் ஐ டேக் a little more deeper. ஒரு சீரியஸ் சென்ஸ் போகும்போது, two girls from the same house, சேம் parents, same environment, same in utero environment also. ஒரே அம்மாட்டன் வந்திருக்காங்க. பட் one girl has irregular cycles, the other girl has proper. Same with boys, one might have early diabetes occurrence இருக்கும், இன்னொருத்தவங்களுக்கு லேட் diabetes வந்து onset இருக்காது. This is a little more serious issue. கொஞ்சம் திங்க் பண்ணா, this is all decided by their genes, you know. So like நான் படிப்பு பற்றி கூட சொன்னேன், மார்க்ஸ், even your memory, the way you can memorize something, the way you can remember something, short term memory, long term memory, இதெல்லாமே it is all again your genes only. So we have a very separate field, wellness genetics அப்படின்ட்டு இருக்குது. இதில் வந்து we can understand your sleep pattern. சில for some people ஒரு 3 hours 4 hours தூங்கினாலே போதும், they are rejuvenated, body can survive. But for some people they need to sleep for that 8 hours. அந்த 8 மணி நேரம் எல்லாருக்குமே தேவையில்லை, சில பேருக்கு தான் தேவை. அது யாருன்றதை நம்ம ஐடென்டிஃபை பண்ணலாம். Similarly நம்ம இப்போ ஜிம் போகிறப்போ we have seen a lot of cardiac arrests on the gym floor. எக்ஸசைஸ் பண்ணிகிட்டே இருக்கிறப்போ we have seen a lot of fatal accidents happening over there. We should understand யாருக்கு எந்த threshold வந்து their body is capable of, and you know அதை பேஸ் பண்ணி தான் ட்ரெயின் பண்ணணும். This is how you know fitness genetics work. And on your eating nutrition, so இந்த micronutrients வந்து vitamins and minerals, நான் யூஸ்வலாக பேஷண்ட் கிட்டயோ கிளைண்ட் கிட்டயோ பேசுகிறப்போ இது உங்கள் பாடியில் இருக்க நட்டு போல்ட் மாதிரின்னு சொல்லுவேன். எவ்வளோதான் ஸ்ட்ரக்சர் நல்லா இருந்தாலும் அந்த நட்டு போல்ட் லேசாக துரும் பிடிச்சிச்சுன்னா அந்த ஸ்ட்ரக்சர் பிரேக் டவுன் இல்லையா, அந்த மாதிரி ஈவன் இஃப் யுவர் மைக்ரோ நியூட்ரியன்ஸ் ஆர் நாட் கேட்டட் டு, இதை பார்த்தீங்கன்னா நம்ம லாட் ஆஃப் அஸ் இந்தியன்ஸ் வி வில் ஹாவ் ஹீமோக்ளோபின் கவுண்ட் கம்மியாக இருக்கும், ஹேர் ஃபால் அதிகமாக இருக்கும். கேர்ள்ஸ் அண்ட் பாய்ஸுக்குமே, கேர்ள்ஸோட பாய்ஸ் தான் ரொம்ப ஃபீல் பண்ணுறாங்க ஹேர் ஃபால் அதிகமாக இருக்குது அப்படின்ட்டு. ஸோ ஹேர் ஃபால் இருக்கும். ஸோ அண்ட் ஆல் ஆஃப் திஸ் வந்து பார்த்திங்கன்னா வி ஹாவ் சம்திங் கால்ட் MTHFR மியூட்டேஷன், வெரி காமன் நம்மளோட கண்ட்ரியில் அது. அண்ட் தென் விட்டமின் டி ஆல்மோஸ்ட் இப்போ இங்கே இருக்கிற ஒரு 50 மெம்பர்ஸ் இருக்கீங்கன்னா 45 மெம்பர்ஸ்க்கு வந்து விட்டமின் டி டெஃபிஷியண்ட்டாக தான் இருப்பீங்க. அண்ட் இதுக்காக நம்ம வந்து வெயிலில் போய் நில்லுன்னுலாம் சொல்லுவாங்க, நம்ம வெயில் பார்க்காத நாளே கிடையாது. It's not like Canada or something. We are every day we are with the sun only still. So all this your genes has to process இல்லையா. So இதை பேஸ் பண்ணி we give recommendations to clients அவங்களோட நியூட்ரிஷனுக்கு எல்லாத்துக்குமே. This is called nutrigenetics. இதுதான் nutrigenomics அப்படின்றது, a very upcoming field again on this. And ma'am, it's a second question.
Currently it is booming a lot. And there are very less nutrigenomics counselors also which are available in the hospitals. That's the reason I wanted to bring. Yeah, exactly. There are very few. This is also very much needed, and that's one of the reason. Yeah.
This is more of nutrition, right? There's something known as food tech, biotech. That's the future trend where it is all about artificial meats, artificial proteins, where you take the strain not from your animals and your plants, but you take the strains from your microbes and develop meat, which is a booming industry in Singapore and other parts of the countries. The other one is you tweak the genes in the crops that are resilient to droughts, which can already predict if the crop is going to survive or not. So biotech and food tech can be used in all these manners.
Perfectly said.
And also there's something known as functional foods, prebiotics, probiotics, fermentation, which is also a booming industry and considered as a future trends. Yeah, food process.
Now I would like to move on to Dr. Naveen and Dinesh, Mr. Dinesh. So as you both are for the people, how many of you are all interested in cloning, cell culture, things? No. Why? Oh, yeah, of course. Please lift your hands. Yeah, thank you so much. So that the questions are for you, that's the reason. So I just wanted to know you are into that. When you spoke about, the crisp editing and all, what is the future of it? In which way, things can be applied? That is one. The same thing goes on to you also, because you are also into the cloning. So how do you think that they have to specifically focus on that to get into that, or like I'm pretty sure innovation is something which happens all of a sudden, that is a different thing, but how can they channelize with respect to this?
Yeah, so here I would like to take like இது வந்து ரொம்ப சயின்டிஃபிக்காக கொண்டு போகாமல் I want to bring it like more like idea point of view. எவ்வளோ பேருக்கு பிரச்சனை இருக்குது, ப்ராப்ளம், உடனே கை தூக்குறவங்க எல்லாருக்குமே பிரச்சனை இருக்குது. யா, we have problem. So what is the first problem? Like நம்ம மார்னிங் எழுந்த உடனே ஃபஸ்ட்டு ப்ராப்ளம் என்ன பண்ணணும்? ஏன்டா எழுந்துக்கணும் அதுவே பிரச்சனையா? குட், ஆமாவா? ஸோ வாட் இஸ் த ஃபர்ஸ்ட் ப்ராப்ளம் யூ ஃபேஸ் வென் யூ வேக் அப்? ஓகே, எழுந்தாச்சு, அம்மா எழுப்பி விட்டாங்க. வாட் இஸ் த ஃபர்ஸ்ட் ப்ராப்ளம் யூ ஃபேஸ்? என்ன பண்ணுவீங்க? எஸ், என்ன பண்ணுவோம்? ப்ரஷ் பண்ணுவோம், ரைட்? ஓ, மொபைல் பார்க்குறது ஐ டோன்ட் நோ, லைக் தட்ஸ் நாட் அ ப்ராப்ளம் ஐ திங்க். ஸோ வித் if you're not brushing, bad breath and a problem இருக்கும், microbes, right? So to address the problem, somebody has developed a toothpaste and brush. So there is a problem when you wake up. இப்போ நீங்கள் லிஸ்ட் பண்ணீங்க அப்படின்னா மார்னிங் டு ரீச் காலேஜ் யூ நீட் டு transport's a solution. Problem is உங்களால் நடந்து வர முடியாது. Okay, first thing you need to identify to want to become an entrepreneur. Okay, so we don't need to teach you like techniques எல்லாம் சொல்லணும்னு அவசியமே கிடையாது. நீங்கள் entrepreneur ஆகணும். First of all, who is an entrepreneur? Anybody an idea? entrepreneurனா யாரு தெரியலையே. Very good. Problem ku solution kondu varanga. Very good. So I thought like somebody would say somebody who's going to make money.
That's the ultimate goal too.
No, I would differ here because many people think that entrepreneur is someone who's make money. But
most of us don't.
So what I generally believe is that, or general perception is like that. Okay, entrepreneur you money, they want to make money. But the thing is, even take Saravana stores, they make money. அப்படிதான் நம்ம நினைப்போம். But thing is, entrepreneur யாரு அப்படின்னா someone's problem solve பண்ணக்கூடியவங்க தான் entrepreneur. Okay, so someone who solves your problem is an entrepreneur. It's not that someone is going to make money, but by solving the problem they're going to make money. அவ்வளவுதான். இப்போ you are going to Swiggy, right? Swiggy தெரியும்ல எல்லாருக்குமே. Swiggy is solving a simple problem. என்ன ப்ராப்ளம் சொல்லுங்க பார்க்கலாம்? Food delivery. More than the AI, Swiggy என்ன பண்றாங்க? Zomato, Swiggy எல்லாமே. ஏன் பண்ணணும்? நம்ம வாங்கிக்கலாம்ல. More than that, convenience, more than that, comfort, அதுக்கும் தாண்டி time, because now I'm so busy I cannot go out. எனக்கு நான் மீட்டிங்ல இருக்கேன், எனக்கு டோர் ஸ்டெப்புக்கு வரணும். So that problem is Swiggy solving. ஒண்ணுமே இல்லை, just connecting. So science is very simple. நீங்கள் ஒரு ப்ராப்ளம் எடுத்து சால்வ் பண்ணீங்கன்னா then you become an entrepreneur. So when Dr. Nithya asked about like gene editing, இது எப்படி நாங்கள் ஸ்டார்ட் பண்ணோம் அப்படின்னா we always look at real problem. When I travel everywhere I see around, I see a problem. Okay, I'll always tell, you give me a problem, I would be ready to solve. So இது என்ன ஆச்சு அப்படின்னா one of my relative kids who had a disease where like அவருக்கு என்னன்னா he has a platelet disorder. Anything happens, he will bleed like anything. அவரோட நோஸ்ல பார்த்தீங்கன்னா டேப்ல வாட்டர் வர மாதிரி வரும். I've seen him in front of me, I was shocked. Okay, completely they would close it. So அவர் என்ன breathe பண்ணுவார்னா through mouth only he will breathe. When I went to the hospital, I saw not only him, there are more than 10-15 kids having the same problem. Someone in front of my eyes saw like literally fighting for their life, right? If you are there, நீங்க அதை பார்க்க கூட மாட்டீங்க, அந்த அளவுக்கு he was suffering. So then I went to it and then I spoke with the doctors. We did lot of research. Then we came up with an idea is that there is no treatment for that matter. Okay, அதுக்கு ட்ரீட்மெண்ட்டே கிடையாது. Then what is the solution? We did a lot of research. Then the solution it could be is one of the things is gene editing. Okay, you can do like a stem cell transfer பண்ண முடியும், but there is only other possible ways. There is a lot of complications. We talked to all big experts in Chennai, எல்லாமே பண்ணோம், but we came up with a solution of gene editing. இந்தியாவில் அதெல்லாம் இல்லவே கிடையாது. நம்ம என்ன பண்ணுவோம், பயோடெக்னாலே always say right, there is no scope. But நம்ம என்னெல்லாம் பயோடெக்ல யூஸ் பண்ணுறோமோ everything comes from US, China, Europe தான். கரெக்ட் தானே? You can start, all of you, like I could see like not only 3 of you, all of you have a equal potential, I would say. Only thing it's all mindset. Entrepreneur வந்து நாங்கள் இப்போ எல்லாருமே பேசினாலுமே you will not become an entrepreneur. அது வந்து it's not like an assignment. நான் நாளைக்கு போயிட்டு you become an entrepreneur, so you want to do an assignment an entrepreneur, it cannot be done on demand. So it has to come within you. You have to feel. I've seen so many people who are not from biotech done so much of discovery because அவங்களுடைய பசங்க வந்து have suffered. Delhi I saw a lady, who are from MBA background, அவங்க பையனுக்கு epilepsy, anytime she would get fits, he would get fits. She developed a wearable, and the wearable பார்த்தீங்கன்னா எங்க fits வந்தாலுமே she would get alert, physician would get alert, go and save him.
So the more is observer becomes an entrepreneur.
Absolutely, I would say, right? You need to observe more, find out more problem இல்லாத மனுஷனே கிடையாது.
Thank you so
Right?
much on that. I would like to get an answer from you also, sir, with respect to this.
Good evening. Good evening, everyone. I'm Dinesh actually. I'm from a CRO company. I'm working a full-time researcher. So, இப்போ நான் இங்கே மேம் அவரோட கொஷனுக்கு ஆன்சர் பண்ணேன். ஸோ he coined the term gene editing. இங்கே எத்தனை பேருக்கு தெரியும் ஜீன் எடிட்டிங்ன்றது வெறும் வார்த்தை கிடையாது. இட்ஸ் எவல்யூஷன். லைக் ஜெனிஃபர்னு ஒரு சயின்டிஸ்ட்டு கிறிஸ்பர்ன்ற ஒரு டெக்னாலஜி கண்டுபிடிக்கலன்னா இன்னைக்கு எல்லாரும் வந்து லைக் வாட் எவர் செட் பீட்டா தாலசீமியா they have cured it with CAR T cell therapy and CRISPR editing. So இது எல்லாமே வந்து சயின்ஸோட எவல்யூஷனாக நான் பார்க்குறேன். when I started from college background 5 years before I'm a student, now I'm a working professional. எங்கே நான் டிஃப்ரென்ஸ் பார்த்தேன்னா I applied all the corporate companies, small research hubs, startup companies, everywhere. எல்லா இடத்துலையும் எனக்கு ரிஜெக்ஷன் வந்தாலும் I didn't lose my hope. I'm like trying to understand what is going on in the world. Like what is the term called stem cell therapy, gene therapy na enna, stem cell therapy na enna, what is cell therapy, like there are different terms. So adu vandu na understand panna aarambichen. When you understand it, you will get a clear idea. So நம்ம ஏன் இதெல்லாம் பண்ணுறோங்க, ஏன் people are doing that. If you get that clear picture, then எல்லாமே உங்களுடைய ப்ராப்ளம்ஸ் ஐடென்டிஃபை பண்ணி my ideology is very simple. I'll target one ஆர்கன்லேருந்து டிசீஸ்லேருந்து ஜீன்லேருந்து அப்படியே போய்கிட்டே இருக்கும். உங்களுக்கு அப்ஸ்ட்ரீம்லேருந்து டவுன் ஸ்ட்ரீம் எல்லாமே அப்படியே கன்சிக்யூட்டிவாக போயிட்டே இருக்கும். ஸோ யூ டார்கெட் as they said ப்ராப்ளமை கண்டுபிடிங்க. ப்ராப்ளம் அது எப்படி வந்து பீப்புள் அஃபெக்ட் பண்ணுது அண்ட் பீப்புள் எத்தனை மக்களுக்கு அஃபெக்ட் பண்ணுது. இட்ஸ் நாட் லைக் ஸ்மால் பாப்புலேஷன். 10 or 20 பீப்புளுக்கு நீங்கள் க்யூர் பண்ணி எதுவும் பண்ண முடியாது. ஸோ அவுட் ஆஃப் பாப்புலேஷன் மில்லியன்ஸ் அண்ட் ட்ரில்லியன்ஸ் உங்களுடைய டிசீஸ் நீங்கள் பண்ணக்கூடிய மாலிகுலர் டிசீஸ் வந்து எந்த அளவுக்கு அஃபெக்ட் ஆகிருக்கு, அதுலேருந்து நீங்கள் எப்படி யூ கேன் மேக் அ வே, அதில் ஒரு ஐடியாலேருந்து நீங்கள் எப்படி கன்வெர்ட் பண்ணலாம் மாலிகுலாக, ரைட்டிங் அ தீசிஸ், சப்மிட்டிங் டு யுவர் ஃபேக்கல்ட்டி, அவங்கக்கிட்டேருந்து ஒரு சஜஷன் வாங்கி யூ மேக் அ கம்யூனிகேஷன். நம்ம வேலை கேட்குறதுக்கு தான் இமெயில் பண்ணணுன்னு கிடையாது. யூ கேன் கம்யூனிகேட் டு எனி ஒன். நான் வந்து அனுஜ் கிட்ட பேசலாம், சார் கிட்ட பேசலாம், எனி ஒன் ஐ கேன் கம்யூனிகேட் பண்ணுறதுனால உங்களுக்கு என்ன ஒரு அட்வான்டேஜ்னா யூ வில் கெட் இன் டு த பாயிண்ட் வேர் யூ வில் கெட் ஆன்சர் ஃபார் யுவர் கொஷின்ஸ். ஸோ இஃப் யூ ஆர் interested in animal cell culture, go and talk to people who is expert in that. So you know what you are going to do in next 5 years. So you keep on ask the question to yourself, like what you going to do after 5 to 10 years. So இப்போ gene editing entrepreneur சார் பண்ணுறாருல, ஸோ இது வந்து நெக்ஸ்ட் 15 இயர்ஸ்க்கு எங்கே கொண்டு போகுதுன்னா already we are making a stable cell line for each and every knockout or knock in or knockdown. We are making a reporter cell line, we are making a overexpress cell line. அது ஏன்னா we are very niche to it. Like requirement வந்து ரொம்ப சிம்பிளாக இருக்குது. இப்போ as you said, simplify your problem. அதுலேருந்து will set up a goal or this molecule we have to do a knockout, this gene we have to do a knockout. So we focus on that. We use CRISPR technology, design a guide RNA, delete the gene, then அதோட கான்சிக்வன்சஸை நாங்கள் ஸ்டடி பண்ணுவோம், ஸ்டெபிலிட்டி ஸ்டடின்னு நாங்கள் சொல்லுவோம். ஸோ அதில் வந்து யூல் ஃபைண்ட் இட். தட்ஸ் ஹவ் ரிசர்ச் வில் கோ. இட்ஸ் நாட் ஒன்லி பிகாஸ் ஆஃப் entrepreneur, it's also a major part to the researcher.
Thank you so much. Wonderful answer. Than being an observer, constantly find a solution towards it in a right time and in a right way is this answer. So next I would like to, is there any other question which you want to ask us rather than sleeping? I can find one happily, one soul peacefully sleeping, but it's okay. So if at all you have any questions, we are here for another 20 minutes, so we want to address and think over it so that we can make it really productive. And before that, I have a question with Dr. Mukesh, as he is from Savita Medical College. ஆண்டர்பிரினர்ஷிப்னு பேசுகிறோம், நிறைய பேசுகிறோம் சார். ப்ராப்ளம் கண்டுபிடிச்சிடுறாங்க, சொல்யூஷன் இருக்குது. இது எல்லாமே எங்கே போய் பாட்டில் நெக், தெர் இஸ் சம்திங் கால்ட் அஸ் ஃபார் ஆல் த பயோடெக் பீப்புள் தெர் இஸ் அ பாட்டில் நெக், clinical trial where they have to break their head on things. So how do you see that? Apart from, this is a question apart from the funding side, as you are working with a hospital setup, I want you to answer on this.
Actually, being a faculty of Savita, we can access all the way 360 angle. We have all the facilities. When it comes for clinical trials, it's not a big deal. Okay, and we have very extensive team, especially for clinical trials, and a lot more clinical trial works already ongoing, especially for cancer. We have pharma companies there, we have collaborate with them, and we have a very good team, both clinicians, researcher, and data management. So all together we are working. It depends on the product, because when it comes for the food, okay, even last year we have come up with a doctor.
I perfectly understand. With respect to the food, the bottlenecks are lesser. So culture or stem cells, you will be facing it in the future. So you need to have an idea on how you have to make it work. So that's the reason this question is all about.
That's அது வந்து ஒரு ஃபார்முலேஷன் மாதிரி கொண்டு வந்தாங்க. பட் நான் சொன்னேன், இதை ட்ரக்காக கொண்டு போனால் நிறைய இருக்கும், இது சப்ளிமெண்ட்டாக கொண்டு போயிடலாம். ஸோ வென் இட் கம்ஸ் சப்ளிமெண்ட், ஓகே, எஃப்எஸ்எஸ் சர்டிஃபிகேஷன் இருந்தால் போதும், அண்ட் அதர் ப்ரொசீஜர்ஸ்லாம் வேறு மாதிரி இருக்கும். ஸோ தட்ஸ் வாட் கிளினிக்கல் பீப்புள், அவங்களுக்கு தெரியும். நம்ம ப்ராடக்டாக சொல்லும்போது அதனோட எஸ்ஓபிஸு ப்ரொடக்ஷன் பார்க்கணும், ஏகப்பட்ட இருக்கும். பட் சப்ளிமெண்ட்டுன்னு வரும்போது அதனுடைய மார்க்கெட் வேறு மாதிரி இருக்கும். பட் ஈஸிலி பிகாஸ் most of the EDI எனக்கு வந்த ப்ரொபோசல்ஸ் எல்லாமே சப்ளிமெண்ட்ஸ் தான். ஃபார் எக்ஸாம்பிள் ரீசெண்ட்லி jamun seed, okay, which is having a very good anti-diabetic property. So a girl for pharma branch la irundhu vandanga, she formulated a diabis biscuit. Okay, so adu idu panni EDI la in fact solla pona panelist la irukravanga kitta kuduthu saapida vechaanga. She is that much confident. சாப்பிட்டு நல்லா இருக்குன்னு சொல்லிட்டு வந்து பார்த்தோம் அப்படின்னா, ஸோ அந்த பாட்டில் நெக் எங்கே அப்படின்னா இடிஐயில் வந்து ஐவிபிஏ வந்து பார்த்தீங்க அப்படின்னா, வாட் எவர் த வேலிடேஷன்ஸ், எனக்கு இந்த ப்ராடக்டாக இதை கமர்ஷியலைஸ் பண்ணுறதுக்கு என்னென்ன ஹடில்ஸ் இருக்கோ அதுக்கு அவங்க ஃபண்டு கொடுத்துருவாங்க. ஒன்ஸ் அது ப்ராடக்டாக ரெடி ஆகிடுச்சுன்னா செகண்ட் ஃபேஸ் ஆஃப் ஃபண்ட் இருக்குது. மார்க்கெட் பண்ணுறதுக்கு என்னென்ன சப்போர்ட்டுமோ, ஓகே, அந்த சப்போர்ட்டை வந்து அந்த ஸ்டார்ட் அப்க்கு வந்து தே ஆர் கண்டக்டிங் பூட் கேம்ப்ஸ், ஓகே, கம்பெனி ரெஜிஸ்டர்ஸ், லீகல் அட்வைசர்ஸ் எல்லாமே கொடுத்து. ஸோ ஆஸ் ஐ சார், இந்த 21 ஆண்டர்பிரினர்ஸ் டிஃப்ரெண்ட் டொமைன்ஸில் இருக்காங்க. ஒன்று எலக்ட்ரானிக்கில் இருக்கான், ஹோம் சேஃப்டியில் ஒர்க் பண்ணிகிட்டு இருக்கான். ஸோ அவனுக்கு ஏற்ற மாதிரி என்னென்ன சப்போர்ட்ஸ் இருக்கும், வி ஹேவ் வெரி குட் நெட்வொர்க். ஸோ த்ரூ தட் தே ஆர் கெட்டிங் அண்ட் தே ஆர் க்ரோவிங், பிகாஸ் தே ஆர் நாட் ஜஸ்ட் அன் ஆண்டர்பிரினர், தே ஆர் கெட்டிங் மனி, தே கேன் see the liquids in their hand. So that's making them more fuel to go more on. Okay.
So, எத்தனை பேர் கிளினிக்கல் ட்ரையல்னா என்னன்னு தெரியும்? What is clinical trial? Could you give me example? What is clinical trial? Subjects, or I would say in a real scenario. இப்போ நான் கார் டெவலப் பண்ணுறேன் அப்படின்னா காரை இங்கே நான் ஓட்ட முடியாது, இங்கே டெவலப் பண்ண முடியும், பட் ஆன் த ரோட் ஐ ஷுட் டெஸ்ட். ஓகே, if I'm developing a car for Indian roads, I should test in Indian roads, not on US or Germany. அங்கே ரொம்ப ஸ்லிப்பரியாக இருக்கும், பிகாஸ் யூ ஹேவ் ஐசஸ், ரைட்? ஸோ அதுக்கேற்ற மாதிரி அங்கே போகும். ஸோ கிளினிக்கல் ட்ரையல்லாம் என்னென்னா, ஃபர்ஸ்ட் ஆஃப் ஆல் யூ நீட் டு அண்டர்ஸ்டாண்ட், நான் ஏதோ ஒரு டிவைஸ் கொண்டு வரேன், அந்த டிவைஸ் வந்து நான் என்னோடய இன்டெண்டட் யூஸ் அப்படின்னு சொல்லுவாங்க. இன்டெண்டட் யூஸ்னால் என்னென்னா, எங்கே என்ன நான் பண்ணணும்னு நினச்சனோ அது கரெக்டாக பெர்ஃபார்ம் பண்ணுதா. இப்போ நான் கார் ரெடி பண்ணியிருக்கேன் அப்படின்னா கார் கரெக்டாக ஓடணும்னா அது ஓடுதா, இல்லை மொபைல் பண்ணியிருக்கேன்னா அந்த ஃபீச்சர்ஸ் எல்லாம் கரெக்டாக ஒர்க் ஆகுதா. தட்ஸ் கால் ட்ரையல். அது ரியலாக டெஸ்ட் பண்ணுவோம், பேஷண்ட் சாம்பிள் எடுத்து செக் பண்ணுவாங்க. So I caution you everyone, don't carried away by the word called entrepreneur. So I've been in this field for last 10 years. I caution you, it's a deep tech. Word kelvipattirupinga, deep tech. It's not that நீங்க immediate start பண்ணி உடனே you can get the fruits. And people always hang for low hanging fruits. It's separate. But if you want to be really serious entrepreneur on biotech, please prepare your mind. Mind prepare பண்ணிக்கோங்க. Immediately you won't get benefit. you have to have the persistence. நீங்கள் வந்து என்ன நினைப்பீங்க, ஸ்மூத்தாக இருக்கும் ரோடு, ரொம்ப மோசமாக இருக்கும். I would say, I caution you. I give an example. We did a clinical trial for one kit we developed, a respiratory panel. It took us 1 year and we spent almost more than 50 lakhs just for doing the clinical trials. This is very simple. நான் ரொம்ப கம்மியாக சொல்கிறேன். When we go for gene editing and gene therapy, it's too much. That's why it is called 16.2 crores. ஏன் ஒரு இன்ஜெக்ஷனோட விலை அவ்வளோ இருக்குது அப்படின்னா, they spent already so much money and the value of that is too high. நான் ஒரு பிளட் குரூப்பை கொண்டு போயிட்டு 16.2 crores பண்ண முடியுமா? 50 rupees அது நீங்களே சொல்லுவீங்க. Because how much value will create. So clinical trial அப்படின்றது ரொம்ப ரொம்ப complicated one. Again I would caution, don't develop a product and go for clinical trial. First understand that and come to your develop a product. Okay, what kind of samples is mostly physician is looking for? It's a urine or saliva, blood தான். நீங்கள் ஒரு கிட் டெவலப் பண்ணிடுவீங்க, ஃபைனலாக அவங்களுக்கு தேவை வந்து யூரினில் வேணும், பட் யூ டெவலப் ஃபார் blood. ஸோ நீங்கள் என்டையராக ஸ்பெண்ட் பண்ண 5 yearsம் வேஸ்ட். ஸோ again I say you start from the backward and develop the innovation later. உங்களோட innovation வந்து I would say only 5%. The rest is the regulation, clinical trial, everything. So
wonderfully added.
Don't carry it away by the
yeah, thank you.
clinicals.on GOV. So you will get all the clinical trials. Our SARS on number, all clinical trial like stem cell, beta thalassemia, you will take a disease, you will list out some 10 to 15 which is ongoing. Clinical trial stage 4 stages இருக்குல்ல, அந்த first stage, the second stage, எத்தனை பேர் research பண்ணிட்டு இருக்காங்க, who are the scientists involved, you will get all the information from that website.
Yeah, currently everything is available also online, so wherein you can learn it up. The process is actually easier than 10 years. Huh? Yeah. So it is actually, I mean, I would say it's in a positive way, it is also equally good, but because like the way we struggled, I don't think the current generation have to undergo all those issues.
வந்து ஒரு இன்சிலிக்கோ கிளினிக்கல் ட்ரையல்ஸை பண்ணி பாசிபிளாக பாசிபிள் இல்லையான்னு சொல்லிடும். ஆமாம், ஒரு ட்ரையல் மாதிரி நல்ல நோ it's like sir says no, and the end பார்த்துட்டு you can to trace it back, maybe you can take it, but you cannot rely on it.
Yeah, so it is really a 60-minute packed session, that is what was mentioned. So the last 10 minutes I open up to the floor. I want you to be benefited out of it. நாங்க வந்து உட்கார்ந்து பேசிட்டு போனோம், நீங்க தூங்கி நீங்க கதை கேட்டீங்கங்கிறது is not necessary. I want you to open up so that we can answer the real questions rather than நாங்களே பேசி ஒரு பிரயோஜனம் கிடையாது. Please open up. Do you all want the mic?
ஆக்சுவலி எனக்கு ஒரு 2 minutes தான் பேச முடிஞ்சது. மெயின் பாயிண்ட்ஸு கொஞ்சம் நான் பார்த்து வச்சுருந்தேன், மிக்ஸ் ஆகிடுச்சு. இப்போ எல்லாருக்குமே வந்து என்டர்ப்ரனர் ஆகணும்னு பிளான் இருக்காது. ஸோ ஜாப் எப்படி தேடணும் அதை பற்றி ஏதாச்சும் ஐடியா இருக்குதா? எத்தனை பேருக்கு லிங்க்டின் அக்கௌண்ட் இருக்கு? எல்லாம் ஆக்டிவாக தான் இருக்கீங்க. ஸோ எல்லாத்துலேயும் வந்து ஃபீட்ஸில் பார்ப்பீங்களா இங்கெல்லாம் வேக்கன்சி இருக்குது அப்படின்னலாம். அப்போது எந்த மாதிரி ஸ்கில்ஸ் எல்லாம் தேவைப்படும் அதுக்கெல்லாம்? ஏன்னா தனித்தனி ஜாப் டிஸ்கிரிப்ஷனுக்கு தனித்தனியாக வந்து ஸ்கில்ஸ் தேவைப்படும். மாலிகுலரில் என்னெல்லாம் தேவைப்படும்? இல்லை, உங்களுக்கு ஒரு ஐடியா இருக்கும்ல எந்த ஃபீல்டில் போகணும், எனக்கு ஃபுட்டில் இன்ட்ரெஸ்ட் இருக்குது, மாலிகுலர் ஃபீல்டு போகணும், கேன்சர், அதுக்கு என்னெல்லாம் ஸ்கில் வேணும்னு எல்லாம் தெரியுமா? அதுக்கு எங்கெல்லாம் ட்ரைனிங்கு இன்டர்ன்ஷிப்ஸ் எல்லாம் கிடைக்கும்னு தெரியுமா? Networking is also important, ma'am. Then they should have an idea where to go and in which company, whether it's in pharma or food company.
Depending on the skill set, you should choose the place.
No, ma'am. As per the place, they have to develop their skills, I am saying.
Exactly. Yes. Yeah. First know your interest, then know the place. That is what.
Because if they are planning in pharma, means there will be analytical instrumentation like GCMS, HPLC, like that. They should go for some training or something like that, I say. Okay.
Core interest is all about.
Okay.
Yeah.
Okay. So, sir, at the first he was telling that he is a last bencher, I think. ஸோ அவர் சொன்னதில் ஒரு விஷயம் என்னென்னா, இப்போ உங்களுக்கு வந்து ஒரு ப்ராடக்ட் டெவலப் பண்ணோன்னு ஒரு ஐடியா இருந்தால் ஸ்ட்ரைட்டாக வந்து ஃபண்டிங்குக்கு போக தேவையில்லை. யூ மேக் யுவர் ஓன் ஒர்க் பிளான் ஆர் ப்ரோட்டோடைப் அண்ட் யூ கேன் contact or communicate with the labs like us. We are ready to give you all assistance for free. So what the thing means, we are help நாங்கள் இன்டர்ன்ஷிப்னு சொல்ல மாட்டோம். Any students who are sending the proposal or something, like if they are having any idea, we will take them for free. So அவங்க ஸ்கில்ஸும் டெவலப் ஆகும், அவங்க போகிற டைமில் ஒரு ப்ராடக்ட் or something, they will be also happy.
Well said, well said, well said.
And if you are anyone interested in making any innovation or something, contact Dr. Ashwini ma'am. I will take care of that for free.
Wonderful.
That's my offer for you.
So I would quickly move on to the questions. Can you all please come up and do you all want the mic? Or do you have the mic? Yeah.
I hear that colon cancer identification is very difficult. Why is it? Why it is very difficult to identify the colon cancer?
It is invasive. Invasive in the sense, see, the organ is very interior. Okay, at least colon cancer is at least colonoscopy is little more less invasive, but we don't, usually the symptoms overlap with other common symptoms like gastric issues, constipations and all that. அதனால first சும்மா gastric issues, constipation நம்ம colon cancer யோசிக்க மாட்டோம் இல்லையா. So usually when they come to us, they're already in stage 3, stage 2. It's an advanced stage, So that is why they say it's a little tough. But when you have a family member with genetics, you can actually predict. நம்மளால predict பண்ணிக்க முடியும் who is at a predisposition for a colon cancer or at predisposition for other cancers also. And NCCN or body, they have framed guidelines on preventive measures also for them. இது எப்படி வந்து stage 0 stage 1 identify பண்ணணும், what are the preventive measures you have to take அப்படின்றது there is possibility. So if anybody has a family history, it is an advice that you can reach out to a genetic counselor in the city and get in touch for your screening for that.
Next question. Please come up with as many questions as possible in the last 5-10 minutes. Yeah. A girl who wanted to become an entrepreneur. Please, come on. Yeah.
Okay, first of all, I intro myself. I am Sri Vashni from second MSc Biotechnology. First of all, I thank you all. In spite of your valuable time you are spending for here, I am greatly thankful to you guys. And my question is all about that idea to product is all about your telling is I'm very approachable and very insightful. But my doubt is, if a problem, the solution is fine. That solution we have to be beneficial by all. So how guys are playing a role by reaching that particular person who need the solution to the product?
It depends. You need to find your niche market.
Okay.
So I come from, I'm into food safety consultancy and food safety audits and training. So my niche market is only food establishments. That is your restaurants, hotels, manufacturing units. So now when the service கொடுக்குறது on the particular niche market. I cannot go to him, can I do food safety audit and all that? It's completely different, right? So what is your plan? Future plan. Just come up with one product or service. Randomly tell me.
Say what is your interest, and இதுதான் என் மைண்டில் இருக்குது. இந்த ஸ்பெசிஃபிக் டிசீஸாக இருக்கலாம், இல்லை ஒரு காமனாக ஆட்டுக்கால் சூப்பாக கூட இருக்கலாம். ஸோ யா யூ சே தட் அதுக்கு தகுந்த மாதிரி நீ யார்கிட்ட அப்ரோச் பண்ணணும், they would be able to guide you.
Okay. Basically my plan is up to biotech based products like in a sustainable goals and sort of saving environment condition for helping agriculture.
Okay, eco-friendly products.
Yes.
Your focus is on eco-friendly products. Not farmers exactly. For example, from the waste, creating the biotech related eco-friendly products. Okay.
See, eco-friendly products is a very broad spectrum. It can vary anywhere from sanitary pads to petroleum. Okay? So it is a different market. But to put a very common, you get a common question or common answer kudukanona, you, like she said, you identify in the group ku ungalku use agum அப்படினு. அந்த group first round as a student நம்ம என்ன பண்ணுவோம்னா நம்ம சுத்தி இருக்கவங்களுக்கு அது எவ்வளவு பேருக்கு யூஸ் ஆகுதுன்றதை பார்ப்போம். Okay, so that is what he said. Identify problem. And the problem நம்மள சுத்தி எத்தனை பேருக்கு அந்த problem இருக்கு, அந்த problem இருக்கிற ஒரு 10 பேர் நீங்க ஐடென்டிஃபை பண்ணிட்டீங்களா through survey questionnaires, because you have groups. காலேஜ்ல இருக்கும்போது நிறைய groups இருக்கும். ஒரு survey questionnaire கொடுக்கலாம். Give it to more friends. LinkedInல போடலாம். இந்த questionnaire கொடுத்துட்டு அவங்களுக்கு இந்த மாதிரி ஒரு சொல்யூஷன் கொடுத்தா யூஸ் ஆகுமா? That's a very small, the first do a market analysis பண்றதுக்கு.
I would like to add on. This point was already told by one of the speakers. Know your neighbor. So once you know your neighbor, you will be able to know what the need is also all about, and accordingly you can target it out. Once you get into the core, you will be able to channelize it through. Can I go more? Hope it has answered. Okay. Or you have one more question?
One more.
Yeah, please.
இன்னும் நம்ம மார்க்கெட்டிங் அப்படின்னு வந்துட்டோம்னா இப்போ நம்ம ப்ராடக்ட்டை வந்து எவ்வளோ பேருக்கு கொண்டு போய் சேர்க்குறோன்னு ஒன்று இருக்குல்ல, பிகாஸ் அதில் இருக்க சேலஞ்சஸ் ஏதாச்சும் கண்டிப்பாக சேலஞ்சஸ் இருக்கும். ஸோ இப்போ நம்ம பிரச்சனைக்கு இருக்கிறவங்களுக்கு ஒரு தீர்வு நம்ம கொடுக்குறோன்னா, இப்போ எனக்கு வந்து இப்போ இவங்களுக்கு உடம்பு சரினா என் மருந்து வந்து எங்களுக்கு ஹெல்ப் பண்ணும், கண்கூட நான் பார்க்குறேன். இப்போ அது மாதிரி கணக்கு தெரியாதவங்க நிறைய பேர் இருப்பாங்க. நம்ம ப்ராடக்ட் நமக்கு அவங்களுக்கு ரீச் ஆகுது அப்படின்ற போது நம்ம எப்படி இது பண்ணலாம், ஓகே எக்ஸ்பேண்ட் அவர் பிஸ்னஸ், அதாவது நம்ம ப்ராடக்ட்ஸை வந்து இன்னும் வாஸ் இதில் எப்படி ரீச் பண்ணுறதுக்கு எப்படி ஓகே தேங்க்ஸ் சொல்லுங்கள். எப்படி அந்த சேலஞ்சஸ் எப்படி இருக்கும், எப்படி அதெல்லாம் தாண்டி how we can reach our product as a number one. That's my
உங்கள் செகண்ட் கொஸ்டின் தான் கரெக்டான கொஸ்டின். ஏன்னா the first question you asked, right, again you are going the same way. நான் ப்ராடக்ட் டெவலப் பண்ணிட்டேன், யார் வாங்குவான்னு நான் எப்போ பார்ப்பேன், அது பண்ணவே கூடாது. The first thing is first go to the audience. யார் பிரச்சனை வச்சுருக்காங்க, முதல்ல you have to identify them. அதுக்கப்புறம் தான் you should go to develop a product. அது பார்க்காம ப்ராடக்ட் டெவலப் பண்ணவே பண்ணாது, இட்ஸ் வேஸ்ட். அது எப்படி தெரியுமா, நம்ம ஒரு லைக் ஐ டெவலப் திஸ் அண்ட் ஐ கோ டு ஈச் ஒன் ஆஃப் யூ, கேன் யூ பை திஸ், கேன் யூ பை திஸ், கேன் யூ பை திஸ், தென் சே ஐ எம் நாட் இன்ட்ரெஸ்டட். ஸோ நீங்கள் கொடுக்கும்போது தே ஷுட் லைக் ஓடி வந்து பிடிச்சி எடுத்துகிட்டு போகிற மாதிரி இருக்கும், ரைட்? அந்த பெயின் பாயிண்ட் வில் சே கால்ட் அஸ் பெயின் பாயிண்ட். அந்த கஸ்டமருக்கு இருக்கிற பெயின் பாயிண்ட் வந்து ஹியூஜ். ஸ்விக்கி அவனால் எழுந்து போக முடியாது, மீட்டிங் மேனேஜர் திட்டுவார், ஆன்லைனில் இருக்கணும். ஸோ கிளிக் த பட்டன், த ஃபுட் கம்ஸ், த பெயின் இஸ் டூ மச். ஸோ என்ன பண்ணுங்கன்னா ஃபர்ஸ்ட் ஐடென்டிஃபை யாருக்கு அந்த பிரச்சனை, ஹூ ஹேஸ் அ ப்ராப்ளம். வி கால் இட் அஸ் கஸ்டமர் பர்சோனா அப்படின்னு சொல்லுவாங்க. ஷீ டாக்ட் அபவுட் கஸ்டமர் டிஸ்கவரி, ரைட்? யாருக்கு அந்த பிரச்சனை இருக்குது முதல்ல அண்டர்ஸ்டாண்ட் பண்ணுங்கள். அண்ட் தென் எவ்வளோ பேருக்கு இருக்குது, 2 பேருக்கு தான் இருக்குது அப்படின்னா தயவுசெய்து ஸ்டார்ட்டே பண்ணாதீங்க. 2 million people have the problem, then it's worth solving the problem. Okay, so first identify the problem, who has it, when they have it, what they want, why they will buy from my product. Okay, அது ரொம்ப இம்பார்ட்டன்ட். மார்க்கெட்டிங் என்னோட ஸ்ட்ராட்டஜி உங்களுக்கு யூஸ் ஆகாது, உங்களோட ஸ்ட்ராட்டஜி எனக்கு யூஸ் ஆகாது. ஒரே ஒரு ஆப்ஷன் என்ன அப்படின்னா, give a quality product and talk about your benefits. We call it as value proposition. Okay, value proposition. ஒரே விஷயம், how many of you know like the last Vijay movie which was banned? என்ன படம் எத்தனை பேருக்கு தெரியும்? இதுக்கும் தெரியாத, கை தூக்க மாட்டீங்களா? ஓகே, அதுதான் ஷார்ட் வைல் பேன் பண்ணப்பா. விஜய் ஃபேன் நினைக்கிறேன் நீங்க, கரெக்டா? ஓகே, போஸ்ட் பண்ண. ஓகே, வெரி குட். How many of you know Bio E3? Bio E3, very good. 3, you know, as a student you can apply for Bio E3. Recently DBT called for just an idea. நீ சொல்றீங்கல்ல, அந்த ஒரு ஐடியா கொடுத்தீங்கன்னா they give you 25 lakhs. 25 lakhs யார் கொடுப்பா as a grant? I'm just giving a very simple example, but you can go ahead and many of my friends develop like 5 crores funding வாங்கிருக்காங்க. There are a lot of huge funding இல்ல, அதனால நான் ஸ்டார்ட் பண்ணல அப்படின்னா I will not accept. Yes, yeah, that's what Bio E3 is like that. இது வந்து 6th standardல இருந்தே ஸ்டார்ட் பண்றாங்க அவங்க. When I saw the list I was shocked. Oh my god, 6th to 12th க்கு வந்து CRISPR diagnostics, monoclonal antibody, stem cell therapy. அதான் நீங்க சொன்னீங்கல்ல, My daughter also, very clear. Yeah. Just last one. Recently, I went to, like a couple of days before, I was in Gujarat, attending a synthetic biology. They talked about a lot of things. Right? Synthetic biology is a biotech or a periya vishayam. Soon you will see like synthetic meat, synthetic milk. Therima, yeast will produce milk, cow milk. Adalam pandranga. Amazing. 8th grade student presenting a paper in the conference. I was paneling the session. I was shocked. I was thinking he was like very short fellow. I've been understand. 8th abdinna. Shocked. He's in the India Book of Records. Youngest person to publish in a high impact journal in India. So as he said, there is no age limits. You can go. The limits are very, very.
Yeah. Moving on to the next question quickly. Yes. He has, and then. No, you please continue.
I just have a simple question, ma'am. So recently there has been this topic going on about gut immune axis and systemic immunity. And when I say that topic, it's actually quite staggering because it directly relates the intestinal system with the brain, with that of the brain and the other functional body parts. My question is, why aren't people emphasizing on the idea of postbiotics? You said that you said the topic of prebiotics and probiotics, but the act of postbiotics actually poses more additional information, as it also involves the idea of resistant starch, inulin and other things. So why not encourage the society to impart on these things? Because they could just simply store the sweet potatoes in the refrigerator for a few hours just to get the ultimate benefit from it by supporting the specific organisms, Faecalibacterium, Proschnitzia or something, some microbes that reside in the large intestine. So why is this not being encouraged? Is it because of the uprising of it, or has there not been?
See, it is not about being encouraged. அது இப்போதான் ஐடென்டிஃபை பண்ணியிருக்காங்க. ஓகேவா? One thing, எவ்வளோ பேர் வீட்டு சாப்பாடு சாப்பிடுவீங்க, இட்லி சாப்பிடுவீங்க, தோசை சாப்பிடுவீங்க, fermented food at home. That's the best postbiotics you can have, you know. So I do gut microbiome tests in my clinic. Pandro, it's a routine for us. Postbiotics are already listed out in that also.
But the problem is, postbiotics aren't actually the problem. We don't necessarily have the microbes to support such process that happens within our large intestine because of all the junks that we get to digest every single day. It gets to disrupt the immune system.
Yeah, so see, we've gone one step ahead. Naanga gut microbiome test pannite, we're also giving ultra personalized probiotics. So your gut might be different, your brother's gut might be different. உங்க ரெண்டு பேருக்குமே உங்க பேர் போட்டு பாட்டில்ல வரும். And this is again FSSAI and FDA approved drugs.
But doesn't it cost a lot? Because it's being specifically
phone வச்சிருக்கீங்களா? உங்க phone வச்சிருக்கீங்களா? என்ன phone வச்சிருக்கீங்க?
A touch phone. Yes.
எவ்வளவு ஆச்சு phoneக்கு? 20. Yes. Gut microbiome test will cost you 10. Probiotics will cost you 5.
But people don't necessarily have the awareness to consider this as a widely
awareness illa. That is the problem there. Okay, so it is not about being cost. I will not agree when people come and tell me genetic tests are costly. அவங்க iPhone ஓட தான் அந்த உட்கார்ந்திருப்பாங்க முன்னாடி. Okay, this is my routine patients. Every day this is the one tell. I'll tell 25,000 test. Phone எவ்வளவு சார் அப்படின்னு கேட்டா health விட phone முக்கியமா? Okay, so this is one question. Cost is not an issue. Awareness is the issue here.
Switch on. So ma'am, basically zeolites, when it comes to zeolites, it's been extensively studied material, and there are lots of research paper on it, and it has a potential to even treat cancer and stuff. And when it comes to that, there are also people studying it to use it in pills and use for cancer treatment, but then there is no particular implementation of it. Why is that so?
See, actually when it comes for how recently we are working in non-invasive. Okay, so ஒரு ப்ராடக்ட் வந்து for example, how to say alkaline phosphatase. Okay, those who are working with molecular biology, they know ஒரு சின்ன wire cost more. If you go to CLRI, alkaline phosphatase மூட்டையாக வச்சுருப்பாங்க to dehair the cattle skin. So நம்ம purpose of product வந்து காமனாக தான் இருக்கும், but where we apply, okay, that it costs matter. Okay, so ஏன் அங்கே அப்ளிகேஷன் இல்லை அப்படின்னு கிடையாது. As sir said, அந்த காஸ்ட்டு தான் அது இருக்கணுமே. And moreover, in India, okay, so last year my publication is 75 papers per year, okay, so it's not about how much you publish, okay, so நம்ம அதை எப்படி வந்து பார்த்தீங்க அப்படின்னா ரிசர்ச் கம்யூனிட்டியில் ஓகே நெக்ஸ்ட் லெவலுக்கு மூவ் பண்ணி எடுத்துகிட்டு போகிறோம் அப்படிங்கிறது தான் இருக்குது. ஸோ இட் டசன்ட் மீன் தட் த மோர் பேப்பர்ஸ் ஆர் தேர் இன் பர்டிகுலர் டாபிக், ஓகே, இட் இஸ் மோர் வயபிள், ஓகே. இன்ஃபேக்ட் நோபல் லாரியட்ஸ்லாம் வந்து பார்த்தீங்கன்னா 2-3 பேப்பர்ஸ் தான் பப்ளிஷ் பண்ணியிருக்காங்களே. ஸோ அதனால் யூ see research, but where it is எக்ஸாக்டாக அப்ளை பண்ணுறாங்கன்னு பாருங்கள். அண்ட் மோர் ஓவர் வென்
quote this particular thing. For example, diabetes பற்றி நம்ம எல்லாருக்குமே தெரியும். The load is humongous. Type 1 diabetes இருக்கட்டும், type 2 இருக்கட்டும். நம்ம ஊரில் நிறைய brain dead cases வருது. எல்லாமே pancreases has been thrown out into the bin. But when I wanted to use the pancreas for the purpose of research, I got a kind of memo or something from the Commissioner of Police. The law, which prevents also the usage at times of certain radioactive elements, the usage of certain things, whether it is going as per the law, that also matters. Apart from that, how far you will be safe, how far your regulatory body will be allowing to use for the cancer patients are already they are they are struggling a lot. Adding on to this, we should not create a pressure onto them. That is one of the reasons proton therapy is also kind of becoming a hit model in certain cases. Did I answer? Yeah.
Yes, ma'am. In that case, if we modify this idea that is already existing, will it work out?
Sure, for sure. And that is what, as I said, we have been working on to it. With respect to the pancreas and diabetes, we have been working on to it to make it viable to the society. Yes, that is the innovation. That itself will be a biggest innovation altogether. Any other questions? Yeah.
Ma'am, both of us வந்து interested to become a doctor, but financial engineering இருப்போ நீங்கள் வந்து genetic counseling சொன்னீங்களே, இப்போது ஹேஸ் அ பயோடெக் பேக்ரவுண்டாக இருக்கிறப்போ ஜெனட்டிக் இன்ஜினியரிங்னு ஃபுல்லாக ஸ்பெஷலைஸ்டாக படிக்காமல் டாக்டர்ஸ் ஹாஸ்பிட்டல் ஃபீல்டில் வந்து ஒர்க் பண்ணணும்னா என்னெல்லாம் டெவலப் பண்ணிக்கணும் ஸ்கில்ஸ்?
Genetic counseling is one option which I give. That's why that 1 year clinical rotation is important, because you learn genetics over there. You have other ways for allied health sciences also, like nutrition. You can go on for a clinical nutrition, but it's a degree again. You have to do a nutrition degree. Achuma edukka mudiyadhu. And there are other we have nutrition. These two are the major things which is coming up greatly right now. I mean, enakku therinja varaikum there might be others. You can just go through them or talk to. You have something.
I can add on to your point. So உங்களுக்கு clinical work பண்ணணும், but you should not have any doctor degree. That's what you are asking, right?
I wish to work on hospitals.
Yes, hospital and clinical aspects. So there is a course called embryology. அது நீங்க பண்ணீங்கன்னா you can become an embryologist. You can work on human patients, like human gametes கூட work பண்ணி you can do IVF, in vitro fertilization. Yes, stem cells will also get work. So நீங்க வந்து ஜெனட்டிக் கவுன்சிலருக்கு ஏதோ ஒன்று நீங்கள் போய் சர்ச் பண்ணால் உங்களுக்கு தெரியும் எதெல்லாம் இருக்குன்னு. தே ஆர் ஸ்பெசிஃபிக் கோர்சஸ் ஃபார் தேட். நீங்கள் அது பார்த்தீங்கன்னா இன்டர்நெட்டில் கிடைக்கும். எம்ப்ரியாலஜி பற்றி நீங்கள் பாருங்கள். ஐ திங்க் தட் சூட்ஸ் யூ.
தேங்க்யூ சார். Sir,
in fact, the way he does, clinical CROs, they do a lot of clinical trials also. So they also work closely with patients, because trials for drug monitoring ellame pandrappo, they are also from the life science background.
ஸோ உங்களை மாதிரியே தான் எல்லாருமே வந்து when they start பயோடெக்கில் பாதி பேர் இங்கே லேண்ட் ஆனது காரணமே they would try to become an MBBS. ஓகே, ஃபைனலி எனக்கு வந்து அந்த ஃபீல்டுக்குள்ளே போகணும் அப்படின்னால பயோடெக் வந்திருப்பீங்க. பட் ஐ டெல் யூ ஒன் திங், லைக் ஈவன் நானுமே அப்படி தான் ட்ரை பண்ணேன். ஓகே, ஸோ thank God I'm not become an MBBS or a physician. The reason is, I'll tell you the benefit. I always tell to, like, I mean, many of the students know already why. நீங்கள் இப்போ MBBS ஆனீங்க அப்படின்னா you could treat patients like உங்கள் லைஃப் டைமில் ஒரு 5 lakhs people you can treat, but you have more potential in biotech. உங்களோட one discovery or one innovation can reach millions of people at one time. அது ரொம்ப பவர்ஃபுல். ஸோ யூ லைக் நான் MBBS ஆனால் என்ன சொல்லுவீங்க, I want to do serve the people அப்படின்னு சொல்லுவீங்கல்ல. this is also serving people, right? So as you rightly said, IVF, stem cells, there are a lot of areas where patient need more benefits. Adu physicians alone cannot do it. So as a vaccine, for example, COVID appa, how many of you like took vaccine? Vaccine ellarum worried padanga. That's again innovation of biotech. So you have a huge potential. So adanal don't feel bad that I didn't become an MBBS.
In the considerate of one last question,
Ma'am,
and a quick answer for yes, you can.
Ma'am,
Yes.
can I ask another question I have?
Yeah.
Sorry.
Yeah, no problem.
So, இப்போ organ on chip devices for personalized ஏதாவது ஒரு diseasesக்குலாம் வந்து demand வந்து ஜாஸ்தியாக தான் இருக்குது. But the product scale up வந்து பண்ண முடிய மாட்டேங்குது. இப்போ பர்டிகுலராக liver diseasesனால் நிறைய chronic liver diseasesக்குலாம் முன்னாடியே early diagnosis இருந்தால் treatment will be easier சொல்லி சொல்கிறாங்க. அதுக்கு வேரபிள்லாம் clinical trials இல்லாமல் one stage pass பண்ணுது. But they are not able to scale up. But the demand and the people suffering with liver diseases are increasing over the years. But அப்படி டிமாண்ட் இருந்தும் ஏன் ஸ்கேல் அப் பண்ண முடிய மாட்டேங்குது அந்த மாதிரி ப்ராடக்ட்ஸ்க்குலாம்?
See, there are certain products, yes, as I have worked with the liver transplantation, I'm just trying to answer this particular question. They have been doing it. It is not that there were certain lag. We also got few products when I was with Apollo. We've had few products which we got it, as you said, the variables and everything, but it was not standardized. It was varying. What the blood parameters reported, what the other thing reported, it wasn't right. So blind eye, it actually certain things fail in the clinical trial, and that's the reason it is not out. It is not out. Things are, of course, diabetes care, there are few drugs which are out, and it has been into the I think phase 2 or phase 3, I'm not all that sure. Phase 3, I believe. So things have been working out in that way. Okay. Yeah. Can I have a quick question?
Good afternoon, respected dignitaries. Thank you for being here. I would like to ask only one thing that I wanted to ask you to share one of the experience.
You can just speak out directly without what. Okay, I think that has already been answered by him. I think that was already answered. Anyways, I would just give the mic to him. Very quick one-liner, sir.
ஆக்சுவலி என்னோட ஹெச்ஓடி யூஜி ஹெச்ஓடி வந்து உன்னை தப்பாக இந்த டிபார்ட்மெண்ட்டில் செலக்ட் பண்ணிட்டேன்னு கழுத்தை பிடிச்சி வெளியே தள்ளினார். ஐ கேம் அப் வித் 6 years யூஜி கம்ப்ளீட் பண்ணும்போது. ஓகே, ஆமாம். ஸோ எம்எஸ்சி நானும் பாலாவும் வந்து கிளாஸ்மேட்டு. ஸோ எம்எஸ்சி வந்து பார்த்திங்கன்னா பாலா இங்கே ஐபிஎம்எஸ் போயிட்டார், நான் வந்து பாரதாசன் போயிட்டேன், ஆசான். ஸோ எம்எஸ்சி முடிச்சுட்டு பிஹெச்டி ஜாயின் பண்ணியாச்சு. ஜாயின் பண்ணிவிட்டு கலைச்செல்வன் கிட்ட ஜாயின் பண்ணிவிட்டு, ஸோ அங்கே எம்எஸ்சியில் வந்து பார்த்திங்க அப்படின்னா ஒரு ஃபேக்கல்ட்டி வந்து பாரதாசனில் 400 கழுதைங்க இருக்குது அப்படின்னு வந்து சும்மா ஏதோ பேசும்போது நான் இங்கே பிஎஸ்சி ஜாயின் பண்ணிவிட்டு ஃபோன் போட்டு 400 கழுதையில் ஒரு கழுதை இங்கே ஜாயின் பண்ணியிருக்கு அப்படின்னு. பிகாஸ் அண்ட் இப்போது சவிதாவில் ஜாயின் பண்ணும்போது கூட ஓகே அங்கே இருக்கிற ஒரு ஃபேக்கல்ட்டி உன்னால் இங்கே 3 மாதம் தாக்கு பிடிக்க முடியாது நாங்கள். ஓகே 2 years மேலே போச்சிருக்கு. ஓகே ஸோ மோர் தென் 150 பப்ளிகேஷன்ஸ் இருக்குது. ஸோ வேர் ஐ ஸ்டார்டட்னா எனக்கு எங்கே என்ன முடியாதுன்னு சொல்கிறாங்களோ, ஓகே உன்னால் இது முடியாது, யூ ஆர் ஒர்த்லெஸ். ஓகே இன்ஃபேக்ட் சொல்ல போனேன்னா ஒரு கிராண்ட்டுக்கு வந்து இன்டர்வியூக்கு போகிறேன் நான். அங்கே என் ஒய்ஃபு நீ போகிற காரியம் உனக்கு உருப்படாது, சண்டை போட்டுட்டு வந்துட்டேன். ஆமாம் சண்டை போட்டு வந்துட்டேன். ஓகே அது வரைக்கும் கூட அந்த கிராண்ட் எனக்கு கிடைக்கணும்னு நான் முடிவு எடுத்ததே கிடையாது. ஓகே ப்ரொபோசல் சாங்ஷன் ஆகிடுச்சு. ஃபஸ்ட்டு கால், தேங்க்ஸ் a lot for your motivation. ஓகே
Yeah, so the problem which I saw is like when I was doing my PhD, full of a patient based தான். இன்னைக்கு பார்ப்போம், next day the patient won't be there. ரொம்ப யங் பேஷண்ட்டாக இருப்பாங்க. It was disturbing. ஒரு 1 hourக்கே நமக்கு இவ்வளோ டிஸ்டர்பாக இருக்குது. பட் வாட் ஹேப்பன்ஸ் லைக் அவங்க மதர் ஃபாதர் தம்பிலாம் எவ்வளோ சஃபர் ஆவாங்க. ஸோ that made us to come up with quick diagnostics and many other in the terms of editing and everything.
I just wanted to stand out from the crowd. So most of my colleagues were already into nutrition and product development. But when I was during my childhood, my parents suffered a very bad food poisoning. So since that time, I wanted to pursue a career in food safety, and I built my own career.
So just like sir said, masters mudichittu I was working in IVF clinic as an embryologist. Most difficult case was PCOS engalukku. They don't respond to medicine and everything. And then I wanted to do something over there. That I did my PhD journey appadhaan panna. I wrote a proposal to the Department of Science and Technology, and I got a grant, a Women Scientist Award for the proposal and a grant, and that's how I started Angalda. And from there, then it was a decision that I had to take this to the Indian community. And obviously after that I had opportunities outside, but I wanted to bring it to the public. Indians benefit avano அப்படின்றதால I put my startup here. It was a very difficult situation da, but I still put it up here. Yeah.
So Spark and pathina, I read one article. It's a generation of iPSC by Dr. Yamanaka. So anga paathu da inspiration ana the stem cell patti idallam. So I go for it.
ஆக்சுவலி சார் வந்து 6 அரியர்னு தான் சொன்னாங்க. அஸ்வினி மேமுக்கு தெரியும், நான் விஐடியில் முடிக்கும்போது எனக்கு 13 அரியர் இருந்தது. அதுக்கப்புறம் மேம் எல்லாம் 5 இயர்ஸில் முடிச்சுட்டு போனாங்க, எனக்கு 6 இயர் ஆச்சு. அதுக்கப்புறம் வந்து ட்ரிவாண்ட்ரத்தில் வந்து அனந்தபுரி ஹாஸ்பிட்டல்னு ஒரு ஹாஸ்பிட்டல் இருக்குது, அங்கே வந்து கிளினிக்கல் பயோகெமிஸ்டாக ஜாயின் பண்ணேன். நான் பார்க்கும்போது எம்எல்டிக்கு வந்து ஆல்மோஸ்ட் 15 to 18 சேலரி, பயோடெக்குங்கிறதுனால 5 to 6. சரி இங்கே சரிப்பட்டு வராதுன்னு ஸ்ட்ரைட்டாக போய் பிஹெச்டி ஜாயின் பண்ணிட்டோம். அது வந்து நூருல் இஸ்லாம் யூனிவர்சிட்டின்னு சொல்லிக்கிட்டு குமாரகோவிலில் இருக்காங்க. ஓகே, மற்றவங்களுக்கு எம்டெக்குங்கிறதுனால 8,000 ஸ்டைஃபண்டு, எனக்கு 5,000. சரி இது செட் ஆகாது அப்படின்னு ஒரு 3 இயர்ஸ் ஓட்டிட்டோம். அதுக்கப்புறம் கோவிட் வந்தது, சரி வேலைக்கு ஆகாது. சரி அதுக்கப்புறம் பயோ இன்ஃபர்மேட்டிக்ஸ் அந்த மாதிரி எல்லாம் வந்து லேப்டாப் வச்சு ஸ்டார்ட் பண்ணோம். கம்பெனி ஸ்டார்ட் பண்ணும்போது இன்வெஸ்ட்மெண்ட் வந்து 20,000 தான். இப்போ கம்பெனி ஒர்த்து ஆல்மோஸ்ட் 4 crores இருக்குது வித்தவுட் நோ ஃபண்டிங். போதல்ல.
Thank you so much. Lessons taken, அதுக்காண்டி அரியர்ஸ் வைக்கணுங்கிறது கிடையாது. They have proved that they are successful now. You can always take a good lesson from it. And இல்லைனா உங்கள் டீச்சர்ஸ் அண்ட் lecturers, I think they will scold us for it. என்ன வந்து இன்ஸ்பயர் பண்ண அரியர்ஸ் வைக்க சொல்கிறீங்கன்னு. So take a positive lesson, and to become an entrepreneur or to be successful, first know the research, what you want. As she rightly mentioned, start right from the basics, know the interest of what he said, okay, and from there try to move on. What are the legal aspects with respect to it, or research panna what are the legal aspects, from there know the know-hows of it, and then slowly try to move towards that particular, and this will really surely help you both for your MSc or for your PhD, or if at all you wanted to become an entrepreneur also. So I think together all the best to all of you, and you are free to reach out to us, and through Dr. Ashwini, she'll be able to help you out. So any questions, any queries further on, you can just reach out to us. Thank you so much. Thank you one and all. Thank you so much to all the speakers.
Thank you ma'am, thank you sir. So it was very nice having you all with us today. So being a professor, being a faculty, ஸோ அவங்க பேசினது நீங்கள் என்ன பண்ணணும், எப்படி திங்க் பண்ணணும் அப்படிங்கிறதான் தாண்டி ஒரு ஹிடன் மெசேஜ் எல்லாமே they started within them. நான் இதை பண்ணும்போது எனக்கு இது தோணுச்சு, நான் இதை பண்ணும்போது நான் இதை ஃபேஸ் பண்ணேன். ஸோ whenever you are doing something, எல்லாமே எல்லாருக்காக பண்ணியிருக்காங்க, not only for them. Of course மேம் கரெக்ட்லி செட் மணி இஸ் ஆல்சோ மை கோல். ஆனால் அதையும் தாண்டி ஒரு காஸ் இருக்குது அவங்க எல்லாத்துலேயுமே, ஒருத்தரோட சஃபரிங் இருக்குது. ஸோ வென் யூ திங்க் தட் அவர் பெயின் பாயிண்ட்டுன்னு சார் சொன்னார் இல்லையா, அந்த பெயின் பாயிண்ட் அவர் வந்து ஆண்டர்ப்ரனர்ஷிப் இதிலேருந்து சொன்னார். பட் வென் we see someone suffering, ஏன் இப்படி நடக்குது அப்படின்ட்டு ஒரு கொஷின் கேட்கும்போது, அது உங்களை ரொம்ப புஷ் பண்ணும்போது அல்டிமேட்லி யூ கேன் டூ வண்டர்ஸ். ஸோ இட் ஸ்டார்ட்ஸ் வித்தின் யூ. அதை வந்து அவங்க ரொம்ப அழகாக எல்லாருமே ஒவ்வொரு பாயிண்டில் எக்ஸ்பிளைன் பண்ணாங்க. ஐ ரியலி தேங்க் யூ ஆல் ஃபார் பீயிங் வித் அஸ். இட்ஸ் டெஃபினெட்லி அ பிக் டே ஃபார் மீ, ஃபார் த பீப்புள். தேங்க் யூ ஸோ மச்.
What an extraordinary and intellectually enriching session this has been. Thank you, Dr. M. Nitya Kalyani and esteemed panelists for an enlightening and thought-provoking session. Today's panel discussion on translation biotechnology, converting academic research into industrial venture, has not only dependent our understanding but has also ignited a powerful vision, a vision where ideas evolve into innovation, laboratory transforming into launchpad, and research become a real-world solution that serve humanity. The wisdom shared by our distinguished panelists reminds us that every breakthrough begins with curiosity, courage, and collaboration. This session has truly empowered us to think beyond boundaries, bridge academic with industry, and build a future driven by science and entrepreneurship. A huge thank you to all the panelists for their wisdom and time, and to all of you for being a great audience. Let's give our panelists a final resounding sound of applause. I request Dr. Balakumaran, Head, Department of Biotechnology, to felicitate the panel member. Please, sir. Thank you, sir. Thank you, ma'am. With this, we officially conclude the day one of the conference. We're looking forward to welcoming you all tomorrow for another exciting day of learning and discovery. Refreshment are available at the outside of the auditorium.
Please glow in a line.
Hello. Hey, sorry. Hello, hello. Hello, hello. This. Hello, hello. லைனில் போங்கம்மா எல்லாரும். பாய்ப்பா பாய்ப்பா. சொல்கிறேன்ப்பா சொல்கிறேன்ப்பா. பார்த்து பொறுமையாக போங்க. நாளைக்கு எல்லோரும் வந்துடுங்க. மறக்காமல் எல்லாம் நாளைக்கு வந்துடுங்க. டீ பிஸ்கட் சாப்பிட்டு வராமல் போயிடுவாங்க.